IMMUNOCON 2024. We hope that you have enjoyed this brilliant minds so far. In the forthcoming day, the talks lined up are exciting and intriguing and bound to keep you curious about the latest advancements and research in the field of immunology. We will now proceed to our 12th plenary lecture with Professor Rafi Ahmed, who is also the recipient of the IIS Golden Jubilee Oration Award 2024 and is the director at Emory Vaccine Center. I request Professor D.N. Rao from AIMS and Professor Satish K. Gupta from NII to lead the session.
Good morning. Welcome to the 12th plenary lecture and the Indian Immunology Society Golden Jubilee Oration Award for 2024. This plenary lecture and the oration will be delivered by Professor Rafi Ahmed, who is the director of the Emory Vaccine Center, who is well known to the Indian scientific community. He has made seminal contribution in understanding the mechanism of immunological memory, which has helped us in developing new and more effective vaccine. In addition to that, he has also made significant contribution in our understanding of the mechanisms of T-cell exhaustion and which will help in developing more effective immunotherapy and treatment of cancer. Professor Ahmed, floor is for you.
Thank you. Thank you. I want to thank the organizers for inviting me and for their kind. Okay, can you all hear me? Yeah. And for your kind hospitality. It's always a pleasure to come back home, even though I left India 54 years ago. Whenever I come back, it still is home. Before I give my talk, I want to say something about immunology. It's actually wonderful to see the immunology meetings in India increasing, the number of people participating, getting better and also younger. So it's wonderful to see so many young people interested in immunology. I may be biased, but I think immunology now is probably the most exciting discipline in biomedical sciences. The reach of immunology goes, pretty much touches everything. Touches infectious disease, touches autoimmunity, touches cancer, also touches things like cardiology, because there's inflammation. Wherever there's inflammation, there's a role for immunology. And many diseases are due to inflammation. So my message to the young people here is fantastic. You have made a great choice, entering a field which will allow you to uncover exciting fundamental mechanisms. But more importantly, there's a fantastic opportunity for translation of your findings into therapies for both chronic infections as well as acute infections and all these other diseases. And I didn't mention vaccines, and of course, vaccines are incredibly important, and the fundamental basis of vaccinology really is immunology. Okay, all right. So with that little bit of message about immunology, let me tell you what I'll discuss today. What I'm going to do in my talk today is, for the first half of it, I will describe to you our current understanding of what T-cell exhaustion really is. And what do we mean when we say that the T-cells are exhausted? And most of these findings that I'll describe in the first part, which will be mostly in cartoon, a little bit of data, but mostly cartoon summarizing our work and other work, have come from the mouse model of chronic LCMV infection. I will then shift to some work on human cancer and tell you about our recent work on immune responses in HPV positive head and neck cancer patients. So basically there are two parts to my talk. First part is really fundamentals of what T-cell exhaustion is. And the second part will be about HPV positive head and neck cancer. So what is T-cell exhaustion? I think what T-cell exhaustion really is, it's an ongoing immune response to chronic antigen stimulation. And the story I'll tell you today is, how do CD8 T cells adapt to this continuous chronic antigen stimulation? The immune system isn't really designed for that. In particular, CD8 T cells are not designed to be continuously stimulated. Their real function is You get an acute infection, you generate a very potent CD8 T cell response. It eliminates the virally infected cells. The effector cells, then most of them undergo apoptosis and die. Subset of them turn into long-lived memory cells, and these memory cells can live for the rest of the life of the individual and play a role upon re-exposure. But when you have a chronic antigen stimulation, then there has to really be a different adaptation of the T cell response. And a major breakthrough in this came from recent studies which defined really what kind of states, what are the different CD8 T cell states during chronic infection. And Vijay also covered some of this in his excellent lecture yesterday. So the major breakthrough in understanding really how T cells adapt to chronic infection was the discovery of these, I should put on my glasses, came from the discovery of these, what I will refer to as either stem-like CD8 T cells. Many people in the field call them TPEX, precursors of exhausted cells. The term I prefer the most is chronic resource cell, and I'll tell you why I like that term. But I'll be going back and forth between stem-like CD8 T cells or chronic resource cell. So this cell was discovered and identified by Se-Jin Im, who was a postdoc in my lab. And at the same time, there was two other papers, a paper by He from China, also in Nature, same issue, and a paper from Dietmar's lab in immunity. These cells are quite unusual, and actually my talk is mostly focused on these cells. So I'll go through this part quickly because I'll be telling you more about them. There are two very unique characteristics of these cells. First is their location. So these cells are usually found in lymphoid tissues, and within lymphoid tissues, they're found in the T cell zones. They are not found really where the major infection is, but they create a niche and are in this niche. Think of it as kind of staying in a bunker. The war is going on, but these cells are in a bunker. And what they're doing is they're providing then effector cells coming from them. So this, the other important point for these cells is that they are quiescent cells, okay, and do not express effector molecules. I'll keep coming back to this. They do not express any effector molecules. These cells are continuously giving rise at a slow rate to these transitory effector cells. Okay. Let me take a step back. So if you ask the question, people use two markers very often to define exhausted cells, PD-1 and TOX. What I want to emphasize is that all three subsets, which are there, the three states, the stem-like cell, transitory effector cell, and the more differentiated cells, which truly are the real exhausted, all of them express PD-1 and TOX1. So PD-1 and TOX1 on its own do not necessarily define an exhausted cell. It is the chronic infection program that brings up TOX and brings up PD-1. So these cells, which are PD-1 positive, TOX positive, but they also have TCF1. So among the three subsets of T cells you get under conditional chronic infection, only cell that expresses TCF1 is the stem-like or the chronic resource cell. These cells also express BCL6, which is very interesting because as you all know, BCL6 is really associated with CD4 T follicular helper cells. But this very interesting chronic resource cell has kind of hijacked certain aspects of the CD4 TFH program. So it also has BCL6. And that is the reason it does not have effector molecules. BCL6 is anti-effector. Okay? So very unique population, CD8 T cells, PD-1 positive, TOX positive, but also TCF1, BCL6, no effector molecules, and expresses co-stimulatory molecule. When they are slowly differentiating, the first step in this differentiation is downregulation of TCF1. And as Vijay pointed out very elegantly, there's this interbalance between TCF1 and TIM3. So TIM3 comes up when TCF1 goes down. But at the same time, also TBET comes up. These cells start proliferating. They express effector molecules, and they come out into the circulation, and then will go to sites of infection. These cells then eventually get what we refer to as the truly exhausted cells, which can be defined by unique signatures. And again, as Vijay pointed out, these cells express this. The program is driven by Blink1. So basically what exhaustion is, it isn't T cells lying in a coma. It's an ongoing immune response. It's an ongoing, and it's highly regulated. It's highly regulated by several inhibitory receptors, regulated by the microenvironment, and in particular, regulated by PD-1, because as far as the CD8 T cells are concerned, PD-1 is the major regulator of this chronic state. Okay? And the key cell that's maintaining this response is the PD-1 positive, TCF1 positive, TOX positive chronic resource cell. So what happens when you do PD-1 blockade? When you do PD-1 blockade, you greatly increase this differentiation. So if this was happening at 1x, this now happens at 5x. Okay? And I want you to get a better idea of how the quantitative differences are between the three subsets. So a simple cartoon to show that. So when you look within the lymphoid tissue, in this case, you'll be looking at the spleens of LCMV chronically infected mice, where there are infected parts and non-infected parts. Typically, you have all three subsets. About 10 to 20% of the cells are the stem-like cells. About 10 to 20% of the cells are these transitory effector cells, and these cells are functional. They can be improved, they can become better effectors, but they are functional cells, and so are these. Okay? And these are our truly exhausted cells. So the ratio typically is 10 to 20%, 10 to 20%, 60 to 70%. So basically, most of the cells there are exhausted, but you've got this transitory effector cells coming out, and you have the stem cells which have pluripotency and are functional. When you do PD-1 blockade, you get now a big increase in the blue cells. Okay? So that's why it works, because what it does is that it promotes the differentiation of more effector cells from the stem-like cells, and then you get more effectors, at least for a short window or transiently you have that. And this is why you get better control of the infection or better control of the tumor. I'll now give you an example in the LCMV model where the stem-like cells are actually located in the spleen. So again, within the spleen, you have both infected areas and then you have the non-infected areas. So here is, so just focus first on this. This is a section of a chronically infected mouse, and we are staining with the LCMV antibodies. So the red is where the infection is. You see that there's a lot of infected cells. The green here is defining these marginal zone macrophages. So and inside that is the T and B cell area. So you'll see that major infection is of the red pulp. And most of these are F480 positive macrophages. And there is some infection here. Those of you who can look, you'll see a little, or antigen there. You see a little bit of small red dots, but this is where the major infection is. Now you ask the question by using intravascular labeling. You can also do this by microscopy, and we've done both, but I'll give you an example here of intravascular labeling. And you see, this is a technique that allows you to identify if the cells are in the white pulp B cell area, T cell zones or B cell zones, or if they are in the red pulp, which is much more accessible to blood. So you see that the stem-like cells don't stain with this, i.e. they are in this area. Whereas the transitory effector cells are mostly in the red pulp, and the exhausted cells are also in the red pulp. Because again, these cells are sitting in the white pulp, surrounded in an interesting niche, and then they are, and as they differentiate, these cells go out where the infection is. The next question I want to address is, are the stem-like cells, and this is an important concept and important for understanding what's going on. Are the stem cells, which are not where the major infection is, are they getting TCR signals? And I'll give you the answer and then show you the data. Yes, they are. All three subsets are getting TCR signals. And we showed this by using a mouse where we can actually, NUR77 is one of the downstream markers that goes up upon TCR stimulation. So we're using NUR77 reporter T cells. And what you see in this slide is this is staining with the MHC class I tetramer to look at LCMV-specific cells. And this is, on this axis is PD-1. This is a control. This is a mouse that cleared the infection. So there's no antigen there. This is a LCMV immune mouse having memory cells. So you see that memory cells, of course, don't express any PD-1, whereas during chronic infection, all express PD-1. And now you ask, are these cells getting TCR signals? This is our negative control. In a antigen-free environment, these memory cells are neither expressing PD-1, nor are they getting any, nor are they NUR77 positive. But if you now go to this chronically infected mouse and look at their antigen-specific cells, and then you, as you said, that you see that all of them are NUR77 positive, and of course, all of them are PD-1 positive. You then ask the question, if you divide them into the 3 subsets, are they all positive? And the answer is yes. If you divide them into the 3 subsets, this is the stem-like, this is the transitory effectors, and these are the more exhausted, differentiated cells. As I told you, these are always the majority population. These are smaller. Okay. You now see that the memory cells, of course, have no NUR77, but all 3 populations express NUR77. And then this leads to one more interesting thing. They are getting TCR signals, but they are not proliferating. So here you look at these 3 cells, all of them getting TCR signals based on NUR77. And then you ask, are these cells at that time proliferating? The stem-like cells are not proliferating. They are very quiescent, and I'll come back to this in a second. The transitory effector cells, i.e. the recent progeny coming from the stem-like cells, are proliferating quite a bit. The exhausted cells are not proliferating. These are truly exhausted in terms of their proliferative capacity. So this exhausted population, which comprises 50 to 80% of the cells, whether you do PD-1 blockade, they don't increase in number. You can throw any cytokine on them. We have in vitro and in vivo, IL-2, IL-15, IL-7, they don't. The stem-like cells will respond to all of these. But when they are in their niche, their quiescence is maintained. And this is one of the very interesting things to explore in the field. You know, how is this quiet? They're not exhausted, but they're being kept quiescent within the tumor within, in this case, the chronic environment. And you see the same thing in the tumor system. They are always quiescent. So I think that this is an area that people are beginning to explore. And this basically is, I think, talk between the T cells and the myeloid cells, which are kind of keeping these cells quiescent. But the important point at the same time here is that maintaining the stem-like state is an active program. The TCR stimulation is there, and that allows them to maintain all these their transcriptional profile and then and remain quiescent at the same time. But then they will respond if you remove PD-1 break or provide appropriate cytokines. And then here is the other part. Look at this. These cells, the transitory effector cells, have high levels of granzyme B. The terminally differentiated cells, even though they have no proliferative capacity and are also not very efficient killing, do have a granzyme B, but these cells don't. And again, this is their unique program with both BCL6 there and with the TCF1 there, and all of these things is keeping them not effective molecules. Okay. How much time do I have? 10 minutes, okay. I'll go through the next part quickly because it'll flow quite nicely. Okay, so now that I've told you about this unique population, then the question becomes, do you find cells like this in human cancer? And the answer is yes. Many people have shown this both in tumor models and preclinical mouse tumor models, but more importantly, in humans. So human cancer is being shown in pretty much any cancer which there is an ongoing immune response. These cells have been identified. Okay? Many people have shown it. We showed this in lung cancer. And then a beautiful paper came out more recently from Neerha Cohen's lab showing the same thing. And he also shows that the presence of these stem-like cells in these niches, and they usually are in the T cell zones. They are not in the B cell zones. So when you look within the lymphoid part, where are they? Are they close to T cells or B cells? They are not close to B cells, they're close to T cells. That's what we showed in our original paper by Sajan, and then Neeraj Cohen very nicely shows this in lung cancer, that they're again in the T cell areas. T cell areas next to the tumor. Now I want to switch, and I'll go through this part quickly, is we wanted to study a viral-mediated cancer so that we could look at antigen-specific cells using the appropriate MHC class I tetramers. Very often when you're looking at neoantigens, it's difficult to know exactly which neoantigen is there. So it's done usually on total T cells, identifying them very appropriately, but not being able to be 100% sure of antigen specificity. But with a viral-mediated cancer, we thought that we could succeed in this and get more information. So we looked at, just to remind everyone, HPV contains the major oncogenes for HPV are E6 and E7. So we included T cell epitopes from E6, E7, but we also included two other proteins, E5, which is also an oncogene in certain cases, and E2, which is a larger protein and is needed for maintaining the HPV genome in episomal form. And our study design was as followed. I'll just explain the study design and walk through the real data quickly. So what we did was we collaborated with oncologists or with surgeons who were doing surgery on people who had HPV positive head and neck cancer. These were treatment naive individuals. They had not had any treatment yet. They had been identified and diagnosed as having HPV positive head and neck cancer, and were going in for surgery. So what we were able to do is that the patients provided us with the tumor sample and also provided us with a metastatic lymph node. This is not a draining lymph node, so we had two sites of the tumor. Primary tumor, which is in the tonsils, and a metastatic tumor, which happens to be in the lymph node. So we got the primary tumor and the metastatic lymph node. At the same time, we got the blood. So our first goal was to identify which HPV epitopes from the E6, E7, E2, and E5 that we were using, which T cell epitopes were being recognized in these individuals. So what we first did is we took their blood and we expanded their blood for 2 weeks in the presence of that pool of peptides. And then we looked to see if there was any cells that expanded and were producing interferon gamma by ELISpot. We then did ICS assay. And then once we got this response, it was a good response, we then went one step further, identified the precise epitope and the tetramer. Here's an example of where you get a good response after 2 weeks of expansion. Very nice interferon gamma producing cells. And then by ICS, we show that they're CD8 T cells, and they also make TNF alpha for this expansion. And here is the tetramer staining. This is a double tetramer. You see this clean population. What's interesting is that it was a small study. We only had about 20 patients, but all of them were positive. There was none of them that did not show some T cells in their blood upon this stimulation and expansion. And then the ones where there was a higher number, we were able to identify, and we identified a bunch of tetramers. And I'll come back to this again. Note that E2 and E5, especially E2, will keep coming up as a major response or a major antigen that's targeted. So we used these tetramers and then looked. And when we did this analysis, so this is now taking the tetramer that we know these individuals are responding to, I don't see, and this is direct staining of the TILs. No expansion, nothing. You get the tumor samples you get the TILs from the tumor and you stain. And this was shocking, and we never expected that there would be such a high frequency of HPV-specific CD8 T cells in the tumor of these individuals. And you can see that in this instance, 3% in the primary tumor, 1% here. I'll come back to the PBMC in a second. Here's another one. Here's a summary ranging from 0.1% and even up to 10%. in some cases up to 10% of the CD8 TILs were recognizing one tumor. And you ask, why is the blood so low? Because that's the way it is. Because the stem-like cells are resident in the tumor, in the T cell zones. The differentiated cells, the exhausted cells, are also resident at the site of the tumor. Only few cells that come out are the recently emerging effector cells. So even when there is such a high frequency of antigen-specific cells in the tumor, cells in the blood are very low. It's not negative because that's where we expanded them from, okay? But the frequency is low. I point this out because when people are looking in cancer patients who are not having any therapy, just steady state and looking for them in the blood, they are there, but they're going to be a very, very low frequency. Okay, then we did the usual stuff. We did the single-cell RNA-seq on the tetramer-sorted cells. I'll go through this quickly. Again, we find the three subsets, the stem-like, the exhausted, and then the transitory, and just focus on this green cluster. Again, the only cell that has TCF1. All of them were PD-1 positive, and all of them were tox positive, okay, but only one with TCF1. There's an interesting chemokine called XCL1, and that again is a characteristic of these cells. We showed that in the mouse model. This attracts the CDC1. So again, this is how they create the niche around them. And again, no granzyme B, no effector molecules. Okay? And then the other ones I've told you about. And here is the several patients that we looked at. You find all three subsets in all of these individuals, in these tetramers. Okay? And then we ask, are these cells functional? So I don't have time to go over the data in detail, but I tell you what we did. We sorted from the tumor the stem-like population and the more differentiated population, and then ex vivo, we stimulated them with the cognate peptide. This was now very easy for us. We could now, because we knew the specificity, so we can now stimulate with the peptide. And what you see here, and this is you labeled with CTV, so you can follow the division. And what you see is without stimulation, and tetramer on this axis, this is stimulation with peptide and other things in the cocktail for 5 days only. And then you see that there's, without providing any peptide, here's the tetramer, no dilution of the dye, no stimulation, no proliferation, same with the terminally differentiated cells. But look at this now. The stem-like population, when you provide the peptide, proliferates beautifully, i.e. it has the pluripotency, has the ability to proliferate under the right conditions, whereas these cells have no capacity to proliferate. So here's a summary of the many experiments we did. Stem-like cells always proliferate. The more differentiated cells don't proliferate. And what happens when they proliferate? Do they express effector molecules? Yes, they do. Look at this. Now granzyme B comes up beautifully. Yeah. CD25 comes up beautifully. These cells already had CD28 and they retained it. So basically these cells are functional. You can take them out, provide the antigen, cognate antigen, they will proliferate, differentiate to give you effector cells. So what this suggested was several things. And let me summarize this and this is important. So we asked, where are these cells within the tumor? And as I told you before, the stem-like cells stay away from the tumor. So this is the section of the tumor. The blue means that this is staining for cytokeratin. This is where the tumor is. There are a lot of CD8 T cells in this thing. It's a hot tumor. Yeah. And in fact, all of them are hot tumors. Okay. And all of them express PD-1. But now you ask which of these PD-1 positive CD8 T cells in the tumor express TCF1. It's only the cells here which are not where the tumor is. There again, within the tumor, there is a area of T cells and B cells, but mostly these cells now are found in this T cell zone. So let me conclude this part and then show you a couple more slides to end up. So what we have shown, that I think there are three important points here. The first one is that the cellular machinery, and I put the box around it, that the cellular machinery, i.e. the PD-1 positive stem-like CD8 T cells that are required for responsiveness to PD-1 and also for other therapeutic intervention, these exist in these treatment-naive individuals who are going in for surgery. Okay? And that PD-1 adjuvant therapy could be an option or as a potentially effective treatment. The other interesting finding was, and I didn't show you all the data, but we found that HPV E2 and E5, in particular E2, is the major antigen that's seen. And after our paper came out, 2 more studies have confirmed it, looking in HPV positive head and neck cancer patients, that again, it's E2, which is the dominant antigen in HPV positive head and neck cancer patients. And why is that important? It's important because until now, almost all of the therapeutic approaches, these involve therapeutic vaccines for HPV and also involves T-cell therapy for HPV, have focused on almost exclusively on E6 and E7. And this makes sense because E6 and E7 are the main oncogenes. Every cell expresses it. So it's not that one should not include E6, E7, but here we're identifying which really the dominant target is. So I think it's really critical to include E2 in the therapeutic vaccination strategies and also consider T-cell therapy. If you're considering that, is to target E2. Okay, so I will end in the next 2 slides. So what we are now doing is we have started a small clinical trial at Emory, and this is ongoing now, where it's small in scale, but I think it will tell us something important. So what we have done is, or I should say our oncologists have done, is they're recruiting 20 patients who are going in for surgery. And before they have their surgery, they will get 2 shots of PD-1 blockade. In this case, it's a collaboration with Roche, so they have with Genentech. So they've given us their anti-PD-L1. And we have so far enrolled 7 patients in the study that's ongoing. We hope to enroll 20. And we have data from the first 2 patients. Both of these patients, very small, but the data are quite encouraging. It's only 2 out of 2, and all they've gotten is 2 shots of anti-PD-L1, so essentially 1 month of PD-1 therapy. And both of these have shown a significant reduction in the tumor in this short window, which is what we would predict. If you have the cells there, they're begging for PD-1 blockade. The cells are ready, they're there, and if the right therapy is given, they will show an effect. Now, this doesn't mean that they should not have surgery, but if this shows up, so in this case, they all will get it. But if the results are promising, we'll go to a larger study, and I think there'll be some potential options for these individuals. And then this is my last slide, because what we are now doing is we want to address the issue of cervical cancer, which is a major problem in India. And again, cervical cancer, there have been a lot of studies done. Almost all of them, again, the focus has been E6, E7. The vaccines that have been tested for cancer have been E6, E7. Not much is known about the T-cell immunology of cervical cancer. So as a first step, we are just starting a collaboration with the OBGYN department at AIIMS. And this will be a collaboration between AIIMS Hospital and with our lab at ICGB that Murali Kaja and Anmol Chandali run. And the Gates Foundation has expressed an interest in funding the study for us. And actually, I'm going to Delhi tomorrow, and we'll be meeting with the OBGYN group at AIIMS on Tuesday, I think. And hopefully, we'll get this started. It will be very small beginnings, but we'll start analyzing T-cell responses in 3 different groups of patients. Basically, HPV positive, but asymptomatic. Okay. Second group will be HPV positive and with SIN, different stages of pre-invasive. And then the last group, the third group, will be HPV positive with cervical cancer. What we hope to do is measure both T and B cell responses to HPV antigens, and then move forward and hopefully try and do what we did with the head and neck cancer. And we'll get information about what are the major antigens being targeted and what correlates with HPV clearance versus non-clearance, and what correlates with progression versus non-progression. So let me end here and thank, I'm going to skip. I just want to highlight, I've mentioned the names of my people, but I want to highlight our collaborators for the cancer studies. And in particular, I want to collaborate Nabil Saba and Mihir Patel for the HPV studies that I showed to you. Thank you very much.
Thank you, Professor Rafi. An excellent talk, and which has a translation value, really excellent. Few questions, please, from the audience. So many hands.
So, Rafi, that was superb. I wanted to touch on some of the fundamentals. And one of them is that what was surprising to me is that these stem cells are seemingly vigorously signaling through their T cell receptor, at least as manifested by the expression of NR4A1, NR77 in the mouse model. Yet, of course, PD-1 signaling presumably is active and not impeding at least that signaling aspect of the TCR. And I wonder how you
that's a great question, and I forgot to mention. So PD-1, as we showed, and many people have shown, Tasuku Honjo showed this many years ago. PD-1 is a beautiful molecule because its expression is regulated by TCR signaling. So in fact, if a cell is expressing PD-1, it means it's getting TCR signals. But at the same time, then it's regulating it. So it's like a rheostat. It's a beautiful molecule that's upregulated by TCR signaling, and its purpose in life is then to regulate excessive TCR signaling. So we knew that PD-1, we have always known it, but I think the new 77 convinces more people because it's a direct evidence. So if in human cancer and TILs always have PD-1, so that means they are getting TCR signals. For us, that's how we look at, if we want to identify tumor-specific T cells, we only look at PD-1 positive T cells. But those are the ones that will be tumor-specific.
Right, I guess just to follow up on that, but there must be some molecular indication of PD-1 signaling in these cells. And do you have that? If you
I mean, this is the PD-1 signaling has been described very well, not by us, by many other people. You know, what it recruits SHIP2. Yeah. It recruits SHIP2, and then that will dephosphorylate TCR, but more importantly, also dephosphorylates CD28 signaling, costimulatory molecule. That has been worked out by many other people very elegantly. I should include that slide. I didn't include the slide, which is kind of textbook knowledge by the work done by other people. Yeah.
So, Saurabh,
always excellent talk. One question was about the non-responsive immunotherapy, non-responsive tumors. You think the problem is lack of tumor antigen-specific T cells, or different ratios of the stem-like versus exhausted population?
Yeah, excellent. Yeah, I think when you are getting complete unresponsiveness, and there are many instances where you don't get a response, I think it's just you don't have enough cells. PD-1 blockade does not create a new cell. It will only give rise to increase from that stem-like cell. So I think one is not enough cells. Second one would be not enough stem-like cells. In that Hakohen paper that I referred to, he shows that the presence of the stem-like cells in these niches correlates with better responsiveness to immunotherapy.
Yeah. So over here. So when the stem cells become transitory cells, do you think there's further T cell receptor gene rearrangement happening?
Sorry, what was that?
T cell receptor gene rearrangement.
No. The T cells remain the same. There's no change. In fact, we showed many times, and also in the HPV, the TCRs are the same. They're conserved.
Alpha beta.
So they're alpha beta, and you'll find the same chronotype, same TCR in all three subsets. Okay? The stem, the transitory, and the more exhausted, it's the same TCR.
You want something? You raise the hand first. Yeah. The last question. Please give it to him.
Yeah, it's an exciting talk. So my question is that, so all these three population of cells, like the stem cell and the effector and the terminally exhausted, all are tumor antigen specific. So what determines their fate? It is already like when they are why these stemless stem cell like cells are maintaining this TCF1, and so what is actually determining their fate? It is
Yeah,
predetermined.
it's their program. So again, I didn't have time to go into it, but the fate decision is made very early. So you generate a more effector-like cell, and you generate this. So everything initially comes from a naive cell. A naive cell recognizes a viral antigen or recognizes a new antigen, i.e. a tumor antigen, and then it differentiates. The initial naive cell differentiate will give you a more effector-like cell, okay, which will have short-lived, okay, but then also amazingly generates the stem-like population. Okay? And then this population keeps the engine running.
Okay.
So the reason it has that is because it has TCF1, has BCL6, okay, has LEF1. Basically, that program plus the epigenetic structure is totally distinct. So the generation of this resource cell is critical for maintaining the response. We and others show that if you knock out TCF1, you don't get that population, the system crashes. Within a few weeks, the T cell response is totally gone.
So the process is random, or it's
Okay, I think you continue.
Yeah,
I will
okay.
go to the very last
we can ask
question,
later.
Taruna.
Yes, okay.
Yeah. Taruna, give the mic to Taruna, please.
We can discuss later.
Be brief, Taruna.
Thank you. And my question is, how are you going to have the inclusion criteria for the clinical trial that you are thinking for these HPV positive cervical cancer patients?
Oh, we are not considering any clinical trial at the moment. Just and this is our collaborator is Nilanchali, who's a OBGYN. At this stage, no clinical trial will be done, just observation, just collecting the blood sample, mucosal samples, looking at immune responses. The clinical trial we're doing with the PD-1 is in Atlanta. Okay.
Yeah,
and there, of course, all the appropriate things have been done. Yeah.
Thank you.
Yeah.
Dr. Rafi.
Okay, Rafi, come, don't go.
So I have
We
a quick
have
question.
another pleasant responsibility. May I invite the President and the Secretary of the Indian Immunology Society to please come on stage to honor him with the Golden Jubilee Oration Award. Lovely talk. Always. Come. Please come. Taruna, come.
Along with this.
There is something, some money. Thank you, Rafi, again. Thank you very much. Yeah.
Thank you, everyone, and congratulations, Professor Rafi, sir. This is Dr. Sanjeev Gupta. We run a small consulting company in India, which is helping translation of medtech innovations to the market, especially for medical devices and diagnostics. And through your talk, I realized that there is a possibility of companion diagnostics development because you're talking about therapeutics. We would be interested to collaborate with you. Thank you. Congratulations once again.
Thank you, Professor Rafi, for a wonderful talk and the chairs for this session. Now we move on to our next plenary lecture by Professor Clive Gray from Biomedical Research Institute, Stellenbosch University, Cape Town, South Africa. I request Professor Satya Das from AIMS and Professor Uday Ranga from JNCSR to now take over and guide us through this session.
So this is you want to introduce.
You introduce.
The pointer. Good morning. It's my great privilege to introduce Dr. Clive Gray to this audience. Dr. Gray is a professor of immunology in the Division of Immunology at Stellenbosch University. He has published over 140 papers in various aspects of HIV and reproductive immunology. His research focuses on inflammatory mechanisms involved in precipitating preterm birth and adverse birth outcomes by investigating cellular and molecular events in placenta. He leads a research group to identify how immunological tolerance at the maternal-fetal interface is broken due to maternal HIV now, and how this affects the ontogeny of infant immunity in HIV-exposed uninfected children. He is also the chair of the Education Committee in the International Union of Immunological Societies and director of the Federation of Clinical Immunology Societies Center of Excellence at Stellenbosch University. Welcome, Dr. Gray.
Great, thank you very much, and thank you very much for the invite, and I'm greatly honored to be here in Bangalore. It's my first time in Bangalore, my fifth time in India, and each time it gets better and better, so thank you very much. So I've slightly changed my title to how HIV-infected maternal HIV infection impacts on placental macrophage function and vascular development. So I have a research group in South Africa called the Reproductive Immunology Research Consortium in Africa. This is my group. We're standing in front of the statue of Nelson Mandela, and this was the prison that he was released from in 1991. So it's a great landmark in Cape Town. So I'm going to talk about the HIV prevalence in South Africa, and as was mentioned by the chair, the HIV-exposed uninfected child, preterm birth and a potential plasma biomarker, and the impact of antiretroviral treatment. And of course, there is universal treatment, as I believe there is here in India, which has mitigated to a large degree maternal fetal or maternal child transmission, but it's not the end of the story. And now I want to home in on placental macrophages, which are the Hofbayer cells, and then search for a potential mechanism. So this is beautiful Cape Town, and those of you who came, we had the third largest delegation last year in Cape Town from India. 95 of you came, and this is what you would have seen at some point in Cape Town. But that's one side of the mountain. On the other side of the mountain lies a township called Khayelitsha, where most people in Cape Town live, and this is the real Cape Town. And this is where we have our research sites, and we are human immunologists as opposed to mouse, and we recruit cohorts of HIV-infected maternal women and collect their babies. And we have two sites, one in Guguletu, one in Khayelitsha. So just to paint the picture, in terms of HIV prevalence, there are 9 provinces in South Africa. Cape Town is down here, that peninsula. The range of HIV prevalence is enormous, ranging from 21% in the east of the country down to 8% where our research labs are. But if you look at the breakdown between male and female in terms of HIV prevalence, if you look at the childbearing age of between 20 and 39 years old, especially the 35 to 39, there's about 1/3 of infected women who would be on antiretroviral treatment are giving birth to children. And here what I'm showing you on the right is a level of viral suppression, the level of people on antiretroviral treatment in terms of the last published data that was in 2022. So what this means is it gives rise to a very large proportion of children exposed in utero to HIV at some stage. Treatment can be either prior to conception, especially if it's somebody who already knows they're HIV infected, or it could be during gestation if it's for the first time that they know, and they may be presenting themselves at antenatal clinics at around 22 weeks of gestation. And what I'm showing you here is within Africa, South Africa has the largest prevalence or proportion of HIV-exposed uninfected and the highest numbers relative to the other African countries. But why is this important? Why would this be important to measure? HIV is not there, they're exposed. Well, these children, these infants are not healthy. They succumb more to bacterial, more to viral infections. They have altered innate and adaptive immune landscapes. They have significant associations with neurological abnormalities and poor growth. And ARV treatment itself has been shown to not reduce those levels of abnormalities, but actually to maintain them. So what is the issue? Is it HIV exposure or is it antiretroviral exposure? And there is published data to show that ARV treatment either before conception or during gestation can result in very similar outcomes in those exposed infants as if there was no ARV, and that was reported before the universal treatment was rolled out. So I'm going to show you one study very briefly that we were involved in, and this was to look at the difference between ARV prior to conception, and women were enrolled. There were 261 women enrolled in this arm here, and the treatment at that time was a non-nucleotide reverse transcriptase inhibitor, an efavirenz-based treatment. And then the second arm was individuals who were treated for the first time because they did not know perhaps they were HIV infected before they came to the antenatal clinic. So we nested ourselves within this study, and we collected 53 placentas from those who were what we call stable, i.e. initiating ARV prior to conception, and initiating initiating those who started ARV during gestation, and we looked at 77 of those. So the outcomes were preterm birth, low birth weight, small for gestational age. So preterm birth is anything, for those who don't know, it's anything less than 37 weeks of gestation. So to cut a long story short, this is what we published a few years ago, is that women who started ARVs or initiated ARVs at some point prior to conception had a twofold risk of maternal vascular malperfusion, which I'll explain in a moment. And this was the only factor that was related to prior to starting antiretroviral treatment prior to gestation. So what is MVM? MVM is the inability of the maternal vasculature to penetrate into the fetal tissue. And there's lots of changes within the placenta, and you get placental insufficiency. So what we found, and it doesn't take too much effort to imagine that this would be the case, is that MVM was significantly associated with low birth weight and also with preterm birth. And this has been previously described before in another scenario where MVM was associated with spontaneous preterm birth. So the hypothesis that we had is that the association between MVM and preterm delivery, the placenta is the link between what has been reported between ARV and poor birth outcomes. So what's going on inside the placenta? So for those who don't know the placenta, so MVM is the inability of the maternal arteries to be remodeled to penetrate into the fetal tissue. So the placenta is a fetal organ, and within the fetal tissue or the villus tissue are these very specialized placental macrophages, which incidentally have the same origin as microglial cells that you heard about yesterday through the yolk sac. So there's a very close association between what happens in the placenta and maybe what happens in the brain. So you have inability of these maternal vasculature to form, leading to poor metabolite transfer, poor oxygen transfer, and poor potential immune transfer. So what may be causing this? So we set out to look at this. But before I say I'll show you the findings that we had. It's been known that the impact of preterm delivery or preterm birth is manifold. So you get development of pulmonary malfunction and wheezing, atopic asthma. You have neurodevelopment defects, which is very interesting by itself because of the connection between Hofbayer cells and microglial cells. You get increased blood pressure and reduced insulin sensitivity and gestation and diabetes in the infant. So the first question we wanted to ask is, could we identify a biomarker of preterm birth during the antenatal period? So we looked at a panel of 28 different cytokines in the plasma, and these plasmas was matched to the placentas from this cohort that I showed you, the PIMS cohort. So they're small numbers because we have longitudinal data and matching placentas, which are fairly difficult to come by. So what I'm showing you here is the heat map of the cytokines, and what I've here is these are small for gestational age. These are HIV positive and initiating ARV prior to conception. There appears to be here an increase in that population of certainly pro-inflammatory cytokines and lower anti-inflammatory cytokines, but all linked to small for gestational age. But when we looked more closely and statistically, there were two analytes which appeared to relate to preterm birth, and that was vascular endothelial growth factor, VEGF, and factor 13A1. Now, VEGF is maintained at high levels at 3 time points prior to delivery and drops in the preterm birth group. In terms of factor 13A, it was low, but spikes in the group that was initiating antiretrovirals during gestation, but remains low in those that were on ARVs prior to conception. So we began to look at the two of these, and could any of these become a biomarker potentially? And low numbers, granted, but it's a hint that maybe these could become potential biomarkers. These would need to be validated in a further cohort. But what this did allow us to do was to begin to look at factor 13A1 in the placenta and try and identify what is the relationship between factor 13A1, preterm birth, and antiretrovirals. So and trap the CO2, causing an extra puff in your pastry. So the first analysis that we did was to look and take existing public data and to do an in silico analysis and to identify in terms of the gene expression of where factor 13A1 lies. And what I'm circling here is factor 13A1 appears in this area here of the UMAP, along with CS factor 1 receptor, along with CD163, along with CD68. And this was homing in on the Hofbayer cells. When we looked at actually then looked at some of our placentas and we stained for those 3 or 4 markers, CSF1, factor 13A, 63 and 68, we found that, and these are within the villus tissue, the fetal part of the placenta, and within the middle there you can see these constellation of placental macrophages as stained by these markers and individually and merged. And these are the Hofbayer cells. And in fact, when we quantify where these lie, if you look here on the right, the blanks are the positive expressions. So this is the predominant population that we find, which I've just shown you is CSF1 positive 68 and factor 13, and a minority or a second population of addition of CD163. So there's a kind of a mixture of different types of macrophages that are expressing factor 13A1 in the placenta. Now, it seems to be exclusively found in the villus tissue. And what I'm showing you here is when we looked at the maternal membranes, which encapsulate the placenta, whether it be the parietalus or the basalis, which are the two membranes, the decidual membranes, we find that the factor 13A is present, but at low density. It's mostly found in the villus tissue and mostly found on the expressed on the Hofbayer cells within the trophoblasts. And what we also, and this is comparing the HIV negatives with the HIV positives, but what we also found in the HIV positive women, and these placentas were exposed, we could not detect any HIV in these placentas, there was lower density of factor 13A expressing Hofbayer cells in these placentae. When we stratified by those that were initiating or stable, i.e. during gestation or pre-conception, there was no difference in the factor 13A. But when we began to break down in terms of where factor 13A is expressed in terms of infant birth weight or weeks of delivery, we find, and what you can see here in red is the preterm birth, and this is preterm birth, low birth weight and low numbers of weeks of delivery. So when we looked at this in a different way, what we found in our control group is that this is also found. When we looked at preterm births in our HIV negative group, we found low factor 13A expression. When we looked at for average for gestational age and breaking it down between preconception and during gestation, there was also low, but they were the same. When we looked at small for gestational age, which is below the 10th centile of birth weight, we found also low compared to controls, but the same. However, when we looked at preterm birth, there was a clear significant difference between those that were on stable ARVs, i.e. pre-conception, with the density of factor 13A, and this was significantly different between those giving birth preterm and initiating ARV during gestation, which was very similar to the negative group. So in summary one, preconception ART results in poor placental vascularization, manifesting in maternal vascular malperfusion. And factor 13A is low in antenatal plasma and matching placenta. Is this an antiretroviral effect? So we wanted to identify this, so we turned to a HUVEC, a human umbilical vein cord angiogenesis model, and we wanted to understand some mechanisms. So here I'm showing you the development of the HUVEC and the addition of factor 13A1, exogenous recombinant factor 13A1, because the first question we wanted to ask is, does factor 13A1 actually result in tubular development and angiogenesis? And the answer is yes, it does. So this is the red dots here. This is control, this is with medium, and this is with addition of factor 13A. When we add an inhibitor of factor 13A, we can see some kind of significant reduction. But so this sets the scene, a baseline of factor 13A enhances angiogenesis in this model. What we then wanted to do was to mimic the in vivo situation of treating those macrophages. So what we did is we took adult macrophages and we cultured them with interleukin-10 and macrophage colony stimulating factor for 48 hours, and we developed a decidual-like macrophage. So we could call these placental-like macrophages. So they're not quite Hofbayer cells, but they are placental macrophages. So what we did is we then put those macrophages into culture with the HUVEC cells to assess the macrophages alone without any kind of additional growth media to identify tubular and branching development. And then we treated those macrophages with the same combination of antiretroviral drugs that we were giving to the patients, and we washed off completely and put that back into the culture. So what I'm showing you here is this is with growth media. This is with the macrophages. You have an enhanced level of branching and an enhanced level of tubular width. And this is the addition of the macrophages that had been pre-treated with the antiretroviral treatments or the antiretroviral drugs. And you can see that there's a significant inhibition of branch numbers and branch width. And in fact, if you add in an inhibitor of factor 13A into the assay, you find a titratable inhibition of the ability here of these macrophages to develop branching and width, and very, very similar to what we find in the pre-treated macrophages, which have been treated with antiretroviral drugs. And in fact, if you even add antiretroviral drugs to the assay themselves, you find and these are the drugs, you find, and this was matching the drug regimen at the time of this cohort, you find a stepwise inhibition. And in fact, when you add in exogenous factor 13A, you can partially restore branching and tubular width. So, in summary of this part, exogenously added factor 13A induces formation of branching and branch width in the HUVEC angiogenesis model. Adding cultured macrophages to the HUVEC assay was sufficient to replace the conventional supplements in endothelial cell culture, and addition of combined or single ARVs disrupted the macrophage effects and was partially rescued. So what does this mean? It means that the MVM that we found is possibly, and with preconception ART, is possibly linked with a mechanism of macrophage dysfunction. We found in vivo that it's on the Hofbayer cells. In our cultured placental macrophages, it appeared to play a very important role once we added the ARVs to those macrophages. So could it be working through the expression of factor 13A in the macrophage, and antiretroviral drugs interferes with this, giving rise to this in vivo phenomenon that we found in women starting ARVs prior to conception. And there's partial restoration of exogenous factor 13A, and this in fact lays the foundation for potential adjunctive treatment, especially in women who are already on ARVs and want to start a family or have an additional child, because we certainly know that they are at risk for placental malfunction. So with that, I will finish. And these are my acknowledgements. So I want to acknowledge Dottie Otwich, who's from Kenya and is a postdoc in my lab who did much of the mechanistic work, and our macrophage gurus who helped us interpret some of the macrophage functions, and our scientists in the laboratory and our clinical staff within the Obs and Gynae unit and within my research unit. Thank you.
Thank you, Dr. Gray, for the excellent lecture. And also thank you for keeping the time. And this talk is open for discussion, but I would like younger people to ask questions. Students and postdocs. Yes, yes.
Yeah, you have shown that the FX3A1 is a potential biomarker in the preterm birth. Have you looked into preeclampsia cases also where it can work as a biomarker? Because you have also shown that it is present on villus.
Yeah, excellent question. And the answer is ongoing. We're now turning over to a preeclampsia cohort whether they're infected with HIV or not, to identify this, because I would predict it's probably there as well. I think it's possibly more accentuated with HIV, but yeah.
Sorry here. Excellent presentation. So is ARV only regulating factor? Do you think that it's working only by regulating the levels of factor 13A? Because factor 13A's role in angiogenesis is quite complex. And do you think that there are additional mechanisms at play in terms of how ARV is inhibiting? And should one rethink how early one starts ARV?
Undoubtedly, there are more than one mechanisms. In fact, the fact that we found VEGF as well being significant points to the role of VEGF as well, which we haven't yet explored. But and what we don't know, of course, is what the finer detail of the signaling, or if there's disrupted signaling through factor 13A. I agree. I think we're touching the surface. Yeah.
Questions from back, students? Yes, there.
An excellent talk, Dr. Gray. Thanks for that. So is it the same thing is applicable to all chronic infections like hepatitis B, C, and the anti-DAA treatment? So how can we infer from this?
Yeah, I don't know. I don't know of any data that shows that, and we haven't begun to look at that. We're beginning to look at CMV HIV co-infection, but I don't know about the other. Yeah.
And are there any studies which say like co-infection with TB? Because like we have more of HIV TB. Are these EVA cohort, are they TB negative or some?
Are they what? Sorry.
TB negative.
Oh, TB. They're all TB negative. Yeah, yeah. We haven't. Active TB disease was the exclusion criteria in our code so far.
Okay, my co-chair wants to ask.
Yeah, it's an excellent presentation. I have a question. What about socio-economic status, feeding, or food quality, nutrition? If this study was conducted in an industrial country, would you see the same result?
Yeah, so these are women who live in that poor housing environment, often high unemployment, food status is low, to be honest. So would we find the same in a more affluent or setting? I think is your question. I don't know. Yeah, it's a good point.
Clive,
yes.
I think a wonderful presentation. I'm just wondering if there's anything known about effects of antiretroviral treatment on, let's call it vascular functions. Is it a known side effect? So does it affect only the placenta, or is there anything else reported? Like, for example, in the eye or wherever?
Well, I think clinical manifestations of cardiovascular disease in people on long-term antiretrovirals may point to some kind of vascular disease or vascular maldevelopment. I think the placenta offers a very exaggerated kind of environment where we can look for this because it's so important in terms of the developing fetus. But it would not surprise me that this is a general phenomena. And in fact, even though there's now capsid inhibitors, which are very promising as a pre-exposure prophylaxis, still some of the ARVs are being, classical ARVs are being used in a PrEP. So this opens up a new area to look at what are the direct effects of antiretrovirals, even on HIV negative people. But your point, yeah, point is well taken.
Okay, since we are short of time, we stop discussion here.
Thank you. Thanks very much.
And you can ask a question to him during lunch hours. So thank you, Dr. Gray, for your excellent lecture. And I thank all the audience for keeping patience. Thank
Dr.
you
Gray,
very much.
please stay on
Have a
this
good day.
stage. We need to take a photo.
Thank you, Professor, for a wonderful talk. Next, we have award presentation 3, the Mid-Career Scientist Awards. I will request Dr. Amit Awasthi, the President of Indian Immunology Society, and Dr. Kaustav Nayak, the Secretary of Indian Immunology Society, to kindly come up on the dais for the felicitation of the awardees. The recipient of this award is Professor Mirza S. Beg from IIT Indore.
It is my pleasure to introduce Professor Mirza S. Beg. He has been selected for GP Talwar Mid-Career Scientist Award. The selection was done by previous Executive Council, and as a customary, he will be giving a talk before we felicitate Professor Beg. He's from IIT Indore, and he's going to present his talk on targeting malfunctions in chronic inflammation. So the stage is yours, please.
Thank you so much for introduction, and thankful to Indian Immunology Society for giving me opportunity to share my research. I'm thankful to conference organizers for such a wonderful hospitality. I mean, anything you need is just phone call away. So thank you, everyone. My today's talk is entitled Targeting Malfunction in Chronic Inflammation. It's not the malfunction, it's a function of malprotein. Malprotein, which is an essential protein in chronic inflammation, just downstream of the TLR4 receptor. So it's actually the malfunction of malprotein. I mean, we can consider both way. So because talk is related to chronic inflammation, I just like to give a simple definition of inflammation. It is body's way of signaling the immune system to heal, repair damaged tissue, as well as defend itself against foreign invader, such as viruses and bacteria. The function of inflammation is to eliminate the initial cause of cell injury, clear out necrotic cell and tissue damage from original insult and the inflammatory processes, and initiate the tissue repair. So with this simple definition, it seems inflammation is good for our body. But why we need to study inflammation, why we need to target inflammation? To understand this, we need to understand two concepts, which is acute and chronic inflammation. Acute is self-regulating and turns off when no longer needed. It's rapid, short-lived response to injury infection that heals damaged tissue. And chronic inflammation is persistent and doesn't turn off when no longer needed. Prolonged inflammatory response, a lot of cytokines, chemokines, reactive nitrogen species, oxygen species start damaging our own tissues. So at that stage, that pro and anti-inflammatory tuning is defective. And at that stage, we need to understand, what is the signaling mechanism. And because of this deviation from normal inflammatory response, we could see disease consequences of chronic inflammation that can be seen in pulmonary disease, neurological diseases, autoimmune disease, arthritis, diabetes, and many other diseases. So in this, in last couple of years, we did a lot of signaling, we dissected many signaling, but in this talk, I'm just focusing on two major fundamental signaling. One is the nitric oxide mediated signaling, and one is adapter molecule, importance of the adapter molecule in inflammation. So I will start with the nitric oxide mediated signaling in chronic inflammation. And by the way, NOS1, we identified in macrophages. Initially, I mean, it is basically in brain cell, but we identified in macrophages and establishes important role in inflammation. We all know we have three different types of the nitric oxide synthase. One is NOS1, which is neuronal nitric oxide synthase. Another one is the NOS2, inducible, which is present in macrophages, and once macrophage is active, you know that they start expressing. Another one is the eNOS, which is endothelial specific, and also called NOS3. All three, obviously, nitric oxide synthase, they do the same thing, converting arginine to citrulline and produce nitric oxide. And this nitric oxide is a mediator of inflammation. NOS2 and NOS3 has been appreciated in inflammation. Early, many investigators have shown these research, but NOS1 in inflammation, I mean, we know not much. So we establish the role of nitric oxide synthase 1 in inflammation. So because it's a bit complex, so I thought, I'll just start with the hypothesis over here. So TLR4, when it's getting activated, it's activates NOS1, nitric oxide synthase 1. And obviously it's active, it will produce nitric oxide. And that nitric oxide will bind to SOX1, that is called suppressor of cytokine signaling 1. And once it's nitrosylated, it will get degraded through ubiquitination and proteolysis degradation. And if it is degraded, then it no longer to bind P65. SOX1, normal condition, controlling P65 to active. But when we have any kind of the signal that activate macrophages, it's getting SOX1 getting degraded and NF-kappa B is active. It will transfer to nucleus and start pro-inflammatory cytokine expression. So this is essential to have this control before infection, because otherwise, if we have the simple bacterial infection, you can imagine the consequences. So at the first stage, SOX1 is controlling P65. So this signaling is something we consider breakthrough, and it's kind of the switch for inflammation to turn on. Just quickly, I mean, we have a lot of data to validate this thing, but I kept only very key data over here. So this is the survival curve in mice. We used LPS model. In wild type, you could see there's significant decrease in the survival after a lethal dose of LPS. But in NOS1 knockout mice, we have increased survival. NOS2 and NOS3 showing a significantly decrease in fact, as compared to wild type, it's mice dying very fast. This is because of some other reason. As I mentioned, eNOS is an endothelial NOS and might have role in overall cardiovascular function. NOS2, at some level, though it's a determinant of the severe chronic inflammation. So NOS2 at some level, it's also helping the feedback inhibition. So maybe because of those reasons, these two knockouts showing significant decrease in survival. But this is what we identify NOS1 knockout increasing the survival. Not just the LPS model, we use cecal ligation puncture model. And here also we could see NOS1 knockout significantly increases the survival. We extracted lungs, and you could see in NOS1 knockouts, we maintain the overall alveolar architecture nicely. NOS3 is almost similar to wild type. So this was the knockout study, but we wanted to see whether pharmacological inhibition is doing the same thing. And we used TRIM, which is NOS1 inhibitor, and we could see the same effect. Mice treated with LPS, before LPS we injected with TRIM intraperitoneal, and we could see significant increase in the survival. And then we extracted lung, and we could see we maintained the overall alveolar architecture in NOS1 knockouts. This is about the total cytokine expression that we determined, and we could see all important cytokines like IL-1 beta, IL-1 alpha, IL-6, all are significantly down in NOS1 knockouts as compared to wild type. We also compared it with NOS2 and NOS3 knockouts, but that almost similar to wild type. Further, we determined macrophage cytokine mRNA level. We treated macrophages with LPS and plus minus. In this case, it was in NOS1 knockout, so macrophage are depleted with NOS1 enzyme. So in these cases also we could see significant decrease in the cytokine level. Further, we also determined total at the protein level, because we wanted to see the protein is also significantly down, and we could see TNF alpha and IL-1 beta, they are at the protein level, they are also reduced. So in the hypothesis, in the first slide, I mentioned that it's NOS1 that producing nitric oxide, and that nitric oxide is nitrosylating SOX1. And that's why we have NF kappa B P65 intact. And that slide is exactly telling the same thing. So this is, we treated macrophages with wild type, and we could see intact P65 because SOX1 is degraded in normal condition. P65 is decreased in NOS1 knockout because we don't have nitric oxide there, and we have SOX1. So SOX1 will target P65 and leading to its degradation. When we use MG132 in NOS1 knockout, we can maintain total level of the P65. NOS3 also behaving same way as wild type. And you could see significant drop in total P65 level in macrophages in NOS1 knockout. In NF kappa B, we have two subunits, P65 and P50. So we wanted to know whether P50 is also affected or it's just P65 related mechanism. So we did the plot for P50, and you could see there is no change in total P50. So it's related to P65. And then we separated cytoplasmic and nuclear extract, and we could identify it's actually the nuclear fraction that is decreased in this mechanism. Because it's SOX1 related, and it's a decrease in SOX1 after nitrosylation, and that slide is telling exactly the same thing. Wild type, we have decreased SOX1 expression, but in NOS1 knockout, we have increased SOX1 expression. So this is what inhibiting P65 total, pushing it to ubiquitination and proteolysis degradation. We did the confocal microscopy, and here you could see SOX1 is disappeared in wild type, but it's there in NOS1 knockout. Whether it is nitrosylation, it's because of the nitric oxide that binding to SOX1. We did the biotin switch assay, And here we could see there is significant increase in nitrosylated SOX1, but in NOS1, there is no, because we don't have NOS1 and we have no nitric oxide. You could definitely ask me, maybe it is because of the iNOS, which is present in macrophages, but this is not true because this study we performed at 1 hour time point. And in macrophages, when you activate macrophages with LPS, it will start expressing somewhere 3 to 4 hours. So there's no chance of iNOS expression over there. So this was the first fundamental mechanism that we discovered, kind of the switch for inflammation, which just initiate at the beginning. There's another mechanism that we dissected, and that is related to adapter proteins in macrophages. We know what is adapter molecules, ligand receptors, and then there are many downstream molecules, and adapter molecules serve an important role in bringing inflammatory molecules together. They're bridging important inflammatory molecule together. So adapters are the important intracellular signaling connecting receptor to subsequent pathway. In macrophages, they regulate inflammatory responses by modulating cytokine production and immune activation. Their dysfunction may result in chronic inflammatory diseases. Understanding of this molecule provide novel perspective for developing a specific anti-inflammatory treatment. So because we interested in macrophage biology, adapter molecules present in macrophages, and especially downstream of the TLR signaling pathway. So we started looking what different adapter molecules are there. So here we listed 6 adapter molecules and their role in different signaling cascade, in complex signaling cascade. Among these 6 adapter molecules, we are interested in TYRAP, which is also called MALT protein, just downstream of the TLR, making interaction with MYD88. And you can definitely ask me, why TYRAP? What's so important in TYRAP? So the answer to this question is, other adapter molecule, they have 1-to-1 interaction. But the TYRAP is having 1 to 17 interactions. I mean, it was discovered in 2001, and every other year, investigator identifying its interaction with this protein, interaction with that protein, and having the important signaling cascade to go on. So we started focusing on TYRAP molecule, and this is the major interaction of TYRAP with different protein, and each interaction having some role somewhere in inflammatory pathway. And I'm happy to tell that we discovered 3 interactions. One is c-Jun direct interaction with TYRAP, p38 MAP kinase direct interaction with TYRAP through PKC delta. So these are the interaction that we discovered, and So these are the interaction, TYRAP direct interaction with c-Jun, and we understand this is upstream of AP1 transcription factor. If TYRAP is active, it will interact with c-Jun, transfer its phosphate to c-Jun, and eventually it will activate AP1 transcription factor. Its interaction with p38 is something interesting. It's not just TYRAP and p38 interaction, it's with PKC delta. So it's actually heterotrimeric complex in which TYRAP bring PKC delta and p38 in close proximity, and PKC delta will phosphorylate p38 MAP kinase. And that is all these 3 interaction we published in multiple papers. I mean, I didn't include, it's a lot of data. I didn't didn't include all of that, but just one slide over here, which is how this PKC and P38 is interacting. So this is the heterotrimeric complex that we identified. We did confocal microscopy to confirm that, yes, these three are interacting. We performed the reciprocal immunoprecipitation to confirm that, yes, these three are interacting. And this is the mechanism, just few Western blot here. Right after the macrophage activation with LPS, we could see in activation of PKC delta. At the same time, we do see activation of P38 MAP kinase. And downstream, also we expect that AP1 should be active. But if it exists in heterotrimeric complex and P38 MAP kinase activating PKC delta activating P38 MAP kinase, then we should see something this type of the result. So this is control, PKC delta. This is the silence. We silence TIRAP. So if we silence TIRAP, obviously these two are far apart. So we silenced here, but it's not affecting the activation of PKC delta. Total PKC delta is intact, but significant decrease in activation of P38 MAP kinase. And obviously we know what is downstream of P38 MAP kinase is AP1 transcription factor, and some level it's also activate NF-kappa B transcription factor. We confirm this by the cytokines, IL-12 and 23, and those are significantly down. So there are many interactions with MAL or TIRAP. All are somewhere important in chronic inflammation. But we are focused on two interactions, BTK and PKC delta. If TIRAP interacting those 17 or 18 proteins, it has to be activated. And its activation will depend on two kinases. One is PKC delta and other one is BTK. So we thought if we block its interaction with BTK and PKC delta, we can stop its interaction with the other proteins as well. So we started focusing on that, and we did some computational study and confocal immunoprecipitation study when and how this TIRAP is interacting with these two kinases. And something interesting we found here. BTK interaction is very early, right after macrophage getting activated. It's just in 5 to 10 minutes. So that's what you can see over here. BTK TIRAP interaction is too early. You could see, they're coming very in close proximity. But PKC delta interaction starts very late. It's after 1 hour or 2 hours. So at early stage, it's BTK that taking part in the chronic inflammation through TIRAP. And in chronic condition, it's PKC delta that taking part in chronic inflammation through TIRAP. So these are some computational study. And as I said, we were interested to identify the common interface. We intend to block the site on TIRAP, which interacting both PKC delta and BTK. So we identified the interface, and we identified the common site as well. And based on that common site, we started a screening of a small molecule. We used two approaches, repurposing and screening with the huge library of a small molecule. And after repurposing, we identify one molecule interacting well with TIRAP common site for those two kinases, and that is dorzolamide. That is that molecule it is currently being used for reducing the ocular pressure, but it's interacting well with TARAP and inhibiting the inflammatory responses. So this figure is showing it's fitting well with the common binding site for those kinases, and we performed we used the LPS mouse model. Here you could see a significant increased survival when we used DZD as an inhibitor. Further, we isolated lungs and we did the sectioning, and we could see in DZD treated mice, we could maintain the alveolar architecture. Cytokine data also suggesting that DZD reducing pro-inflammatory cytokines significantly. In the same screening, we identify another interesting molecule, and that was the levofloxacin. And we particularly interested in levofloxacin because if we propose something for sepsis, that would be the dual targeting. I mean, at one end, we targeting bacteria, we killing bacteria because it's broad range antibiotic. On the other hand, we targeting inflammation. So we targeting inflammation dual way, and another way we targeting bacterial infection.
So Dr. Beg, you have a minute more.
Yes, sure. So we named this preparation LEDOS, in which we used both levofloxacin and dorzolamide. And one more interesting thing over here is levofloxacin and dorzolamide fitting together at the same time. I mean, this we came to know later when we just giving the combination, both drugs fitting at the same time on TIRAP. And that can be confirmed by this Western blot. TIRAP activation is significantly increased right after the LPS stimulation. Levofloxacin inhibiting TIRAP phosphorylation. Disease inhibiting, because we identify these two molecules targeting TIRAP and inhibiting its phosphorylation. And in case of when we treated macrophages with both the drugs, we could see much decrease in TIRAP phosphorylation. So it's actually the synergistic effect. Using levofloxacin and dorzolamide is the synergistic effect, and we can use this preparation for various chronic inflammatory conditions. For this method, we received Best Technology Award at IIT Indore. These are some supporting data which telling that having LEDOS in sepsis mouse model significantly, it's maintaining the overall structure of all different organs. Cytokine data suggesting that LEDOS having the synergistic effect and it's inhibiting the cytokine expression. I mentioned that we used two different approaches. One was repurposing, and another was screening with the really big library, 30,000-40,000 compounds. And we performed the computational virtual screening and all. And we came up with a certain molecule top hit. Then we performed in vitro assays, and we finally concluded one molecule, and that is we named anti-FLA. This is absolutely novel molecule, not earlier reported for inflammation.
Could you please conclude? Your time is up.
Wrapping. So here also we could see, significant increase in the survival in mice. Lung alveolar architecture is well maintained. Cytokine is significantly decreased. So just two more slides, and I'm done here. Further, we're hypothesizing that TIRAP is a very dynamic molecule. Its interaction with different protein having important role in chronic inflammation. And it's something like that. When ligand is binding to TLR, it's activating TIRAP. And and that activated TARP is ready to interact with different type of the proteins. It's not just because there are 17, 18 proteins, and TIR domain, which is accepting different proteins, is very small. So it's circumstances, in what circumstances these proteins are interacting. So early stage, maybe one protein is interacting, in later stage, another protein interaction. So this giving us opportunity, what protein complex we need to target. Like in my study, we identify that PKC delta at the later stage. So I don't need to worry about, obviously early stage interaction is also important, but chronic stage, I mean, if I'm targeting TARP with PKC delta, that would be more beneficial. Not only that, in different type of the stimulants, also, this TARP interaction would be different. So one type of the ligand will make different complexes, another ligand mediated signaling will make the different complexes. So we need to understand in particular condition what interactions are prominent. So if we know those prominent interaction, we need to focus on that, and we can inhibit those interactions to inhibit the inflammatory responses. With that said, the takeaway, NOS1 drive nitric oxide plays a role in TLR-mediated inflammatory tissue injury. The specific regulation of NF-kappa B by NOS1 drive NO represent an important advance in understanding of NF-kappa B biology. TARP regulates macrophage-mediated TLR signaling. TARP activation through phosphorylation promotes inflammatory response by activating pathways like NF-kappa B or AP1 transcription factor and disrupting key TARP interaction. Like drug toslamide and tifla has therapeutic potential in chronic inflammation. With that said, I would like to thank my group, my team, students, and my collaborator, Dr. Yusuf Hamid, the founder of Cipla Pharmaceutical. He's supporting my all drug discovery related project. Professor Asrar Malik, he's is distinguished professor at the University of Illinois, Chicago, and Professor Vijay Shah at the Mayo Clinic, Rochester. I'm working with them for various inflammation-related project. Professor Alexander Orikov, I'm working with the inflammation and atherosclerosis concept. And Teresa, she's in Imperial College London. I'm working with her with inflammation and cancer project. With that said, and I would like to acknowledge all the funding I received, Department of Biotechnology, DST, CSIR, and the New York Academy of Science.
Thank you.
Thank you so much.
Thank you. So we have time for just one burning question. Yeah, I think Priyanka.
Hello, it's a wonderful work here. So as you know that the lipid A part of the LPS plays an important role in activation of TLR4, right? Yes. So, and TLR4 also gets activated, I think, by saturated fatty acids. So do you have any clue that whether this mechanism that you have shown is also activated or works with saturated fatty acid activating TLR4? Thank you.
We actually work, macrophage activation is one of our area. And we're looking macrophage activation in different conditions as well. And one of that is oxidized LDL that we have tested, like LDL and oxidized LDL. And there we could see, yes, there is activation, but it's along with the CD34, 36 will also play important role there. But yes, we do see the activation, and it's less impactful. When we have the LDL or oxidized LDL, but we could see NOS1 activation. And there we have important interaction that is not PKC delta, but is interacting well with CJUN, PKC delta and CJUN. So that's project ongoing, and we trying to inhibit PKC delta and CJUN interaction. Yeah, thank you.
Thank you. Yeah, I think rest of the discussion we can continue in the tea time. So now we are felicitating Dr. Beg with a medal, a certificate, and a check from this Indian Immunology Society.
Congratulations, Professor Mirza, for the award. Next up, we have a technical presentation, a 10-minute technical presentation by the team Spinco.
Okay, testing. All right. Okay, I see some people leaving, some people staying. That's all good. So hi everyone. My name is Edna. I am from Singapore and I represent Standard Biotools on behalf of Spinco that is at the booth outside, just outside the hall here today. So for today, I'm here presenting the solutions for your proteomic needs. And at Standard Biotools, what we are interested in is to accelerate breakthroughs in human health. Basically, how we can help you in your research in order to accelerate your breakthroughs. So I only have 10 minutes to present like really amazing platforms. So if you have any questions, feel free to find myself as well as my colleagues outside the hall later today. So first off, I'll start with the SomaScan assay. With the SomaScan assay, it is actually a technology that is developed by SomaLogic, and it is the current only provider that offers detection of over 10,000 proteins at one go. All right, you can detect up to 10,000 and over proteins at one go. And the reason is because of this DNA aptamers. So this DNA aptamers is named in the SomaScan assay is called the SOMAmer, which is short for slow off-rate modified aptamers. When using the SOMAmers, many people have the question, how is it different from antibodies? In fact, if you compare it with antibodies, it is similar or way more specific than your monoclonal antibodies. We do have a leaflet outside at the booth. You can take it from us. It has the explanation as to why it is way more specific as well. So these aptamers, DNA aptamers, they are really specific to the protein target, and it's being screened from a library of up to 10 to the power of 15 different DNA aptamers. What type of samples you can run the SomaScan assay with is your plasma, your serum, urine, even cell protein lysate, as well as cell culture conditioned medium. Okay, with the SomaScan assay, you can detect over 10,000 proteins, including your cytokines as well. All right, so a whole range of it, amazing platform for foundational work as well as for validation work. If you want to know more about publications or studies that have utilized SomaScan assay for their work, a quick look on the website, you will see a whole range of applications for the SomaScan assay, ranging from hip fractures, the second bottom one that has been published this year in Nature Aging, all the way through to cardiovascular health, and subsequently in other pages, we have it in oncology as well. So a whole range of applications for SomaScan assay. So that's a brief on the SomaScan assay, which is the first one on the right, which is ultra-high plex serum proteome analysis. Ultra-high because it's more than 10,000 proteins that you can detect. Next, we have flow cytometry as well as spatial biology. So you might be wondering, what if I don't want to look at 10,000 proteins? What if I just want to look at a specific population of immune cells, right? I want to do deep cell immune profiling. What can I do? What are your solutions for it? Here we have the CyTOF, which is short for cytometry by time of flight. It is just another flow cytometry platform, but better. Why? This is because we use metal ions that are connected to the antibodies. We do not use fluorochrome, we use metal ions. And because we use metal ions, each isotope has a specific time of flight that's being detected at the back end in the instrument. Thereby, you have discrete signals for clean data. You do not need any staining controls, and you have minimal sample requirement. So all this overcomes the concern of fluorescence-based cytometry. If I place the two charts side by side, or one up and one bottom, the top being the CyTOF, which is cytometry by time of flight, and the bottom being for fluorescence, you can see a big stark difference in just the spectral curve at the bottom and the mass isotope intensity at the top. You have very discrete peaks with CyTOF, which is why for CyTOF, you do not need to do compensation. You save your reagents, you save your sample, and you save your time. So when people are asking, when do I need to consider CyTOF? I would say whenever you are considering any panel, 5 or more colors. 10 colors, more than 20 markers. You have more than 20 markers. That's where you can find out the relationships between cells. You don't want different cocktails. You just want all in one cocktail in order to understand the biology that's going on within the system. So some statistics to show you guys here. CyTOF is the technology of choice when people are looking for high plex studies. We're not talking about 5 markers, we're not talking about 3 markers, we're talking about 20 or more. Even 10 start considering CyTOF. So as of last year, 2023, we have more than 200 clinical trials and more than 60 and growing peer-reviewed publications that show CyTOF for their high-plex studies. And I'm sure many of you will know all these amazing institutes and their logo shown on the screen right here. They are using CyTOF. We are in very close working relationship with them for their clinical and pharmaceutical studies. So CyTOF is the simple and powerful platform that is trusted because of the reproducible and publishable data. If you want to know more about the mechanics and the principle behind the instrument and how it is being done, you can always just have a chat with us. We're just located outside, or you can always come in contact with us. We are more than happy to share the technology. But within the next 3 minutes left, I can't explain in depth, right? So always feel free to knock on our doors and chat with us. And just some example to show you why CyTOF is the quality, produces quality data you can trust is that these are data that's not generated by us. So these are not in-house data. These are data that's generated by our collaborators or by independent labs themselves. They do bridging study on their own and we just ask for any feedback. And these are just examples of the data that we have. So with spectral flow, a lot of cytokines and soluble factors, the data is missing. One of the considerations as to why is because of compensation. Once you factor in compensation, certain low abundance markers and cytokines can start to disappear. Whereas in CyTOF, we do not compensate, right? There is a specific method targeted to each antibody for each of your protein target. What you see is what you get. So in two different studies here, one at Boston Uni, one in another team, they have been able to identify rare immune populations that expresses these cytokines, which was difficult for them to get with spectral flow. And in terms of spatial biology, we have the Hyperion XTI. So the Hyperion XTI enables you to look into your tissues in a spatial manner. So you can stain your tissues up to 42 markers, image them at one go. And the reason why it is the platform of choice is that it removes the concern of fluorescence spectral overlap, and the autofluorescence concern. So in this image here, on your right is fluorescence imaging, some autofluorescence background being caused by the antibody, and thereby masking your CD68, which is your red specks, as opposed to the image on your left, which is generated by the imaging mass spec. You have very clear distinct signal, thereby showing more of your CD68, your macrophage marker. Again, there is no manipulation of the data. These are all raw data that we obtain. Another thing with imaging mass cytometry is that because we do not look at fluorescence, we look at metal isotopes, we are not concerned with autofluorescence coming from tissues. Your brain tissue, skin tissue, lung tissue, all this generate autofluorescence naturally. So this is a paper from Frontiers in Immunology. The statement there is on the paper itself, contributed by the author, whereby they selected imaging mass cytometry to overcome the fluorescence limitation of skin tissue, and it is a 39 marker panel. They stain their tissues with 39 antibodies and they image it at one go. There's no cyclic, there's no stain, wash, remove, and restain and re-image. Is all at once. It saves you time, it prevents your tissue from degradation. And if you were to combine our two systems, which is the XT system, which is our flow cytometry system, and our spatial biology system, which is the Hyperion XTI, you can perform studies as such. So this is in clinical cancer research, where they did paired blood and tissue biopsies from triple-negative breast cancer patients. On your left is data generated from CyTOF, which is flow cytometry. On your right is data generated from the PET patient sample on the tissue itself. So here they evaluated the efficacy and safety of combining the treatments, and the data then suggested is the CD8 T cells and myeloid cells as predictive markers of response and non-response. So you have a more holistic, a more wholesome picture at the end. And I'm on time, but if you want to know more data or more information, we are just located outside at booth 19, just outside the doors. And feel free to reach out to SpinCo as well and Genotypic for Soma Scan if you have any further questions. All right, thank you for staying back, and I hope to have more conversations with everyone outside later today. Thank you.
Thank you, Team SpinCo. We'll now be having a short break for tea. Also, we will like to inform that Dr. Taruna Madan will be informing about the recent developments at ICMR to support medtech innovation. I request all the interested audience to kindly stay back. Also, the company stalls needs to be cleared by 5 p.m. from the Jain Tata Auditorium. I repeat, the company stalls, please clear their stalls by 5 p.m. I request the speakers of the next session to kindly submit your presentation.
Shripad Patil from Nimhans to now take over and guide us through the session. I request the professor to ensure that the talks are adhere to the time. Okay. The chairs will do that, right? Yes, yes. I request the chairs to also press the buzzer just before 2 minutes. Okay, fine.
Okay, good morning, everyone who is assembled here post tea. We have 4 speakers. I know most of them, they still get only 12 minutes presentation time. And let us save about 3 minutes for discussion. PhD from Indian Institute of Science, and now and then Harvard, then he's back.
Yeah, thank you, sir, for the kind introduction. I'm Kushagra, and we have a mouse immunology lab in institute named GNCSR, which is like 10 kilometers from here. And my lab focuses on transcription and how transcriptional changes drive immune cell development and function. I'm sure this audience can appreciate that our genome inside the nucleus is not linear, it is folded. Now, if you look into this picture on the left-hand side, you can see that our genome is basically partitioned into compartments. We have A and B compartment, which basically signify your active and repressive chromatin. And within these compartments, we have domains called topologically associated domains. And within these domains, we have transcription going on. Now, when transcription happens, you will have opening of chromatin, and that will cause a lot of superhelical tension in the chromatin. And this local DNA topology can have further repercussions on the overall architecture of the chromatin. Now, there are many proteins which control the architecture of chromatin. 2 of them we study in the lab. One of them is cohesin. Cohesin is a multi-protein complex that basically entraps the chromatin in its ring-like structure and basically controls the TAT formation, as well as enhancer promoter interactions are also controlled by cohesin. And the other group of proteins are topoisomerases. These are DNA metabolic enzymes that control the topology of the DNA during replication as well as transcription. There are 6 different mammalian topoisomerases. We focus on topoisomerase 1, which is eukaryote specific. What we do in the lab is basically we try to understand how these proteins regulate immune cell development and function. And through that, if we can understand the detailed framework of their mechanism, and if we find some new tenets in their mechanism, this will be applicable to other systems as well. And today I'll be focusing on one of the projects in the lab where we are trying to understand role of topoisomerase 1 in T cell development. T cell development, as all of you know, I don't think this audience needs introduction to that, but for the uninitiated, it happens in thymus, and your progenitors travel from bone marrow to thymus and differentiate into CD4 or CD8 positive T cells. Now, there are 2 rounds of selection which happen in thymus, negative selection and positive selection. Positive selection is controlled by cortical thymic epithelial cells or CTECs, while the negative selection is controlled by MTECs, and it is driven by a gene named AIR. Now, a few years back, when we are trying to understand the mechanism of immune tolerance, we had this data in our hand where we injected black 6 mice with a drug which can target topoisomerase 1. And what we found was in periphery, you detect autoreactive T cells. What we have done here is we have stained the TCR repertoire, which can target an I antigen through tetramer staining, and you can see these cells increase. And when we followed up this work in my lab at JNCSR, what we found was these mice had smaller thymus as well. So this was a little surprising for us because we are seeing that thymus size is decreasing, but you have more autoreactive T cells going in the periphery. Now, at that point of time, not a lot of data was there to basically suggest that what role topoisomerase 1 can have in T cell development and immune system in general. So we thought that we'll develop a project on this, and we started working on that. Now, I'm sure all of you will agree that one of the best way to understand gene function in immune system is to create deletion, and we use Kree-lock system here. We crossed top 1 flox mice with some specific Kree drivers and obtained the knockout lines. In thymus, there are many cells, but 2 cells are major. Obviously, you have T cells, but as I mentioned, you have these thymic epithelial cells. So we used 2 drivers, FOXN1 Kree for TEX and CD4 Kree for T cells. And I'll be majorly focusing on the data which is coming from TEX-specific deletion of topoisomerase 1. Now, as we all can relate to the fact that science is fun because of its serendipity. So when we opened these mice, in het mice, we saw the thymus. But surprisingly, in top 1 knockout mice, this is TEX-specific deletion of top 1, we don't observe thymus. And this was a little surprising because we did not expect that. Now, in our model, the Kree expression is driven by FOXN1, and FOXN1 controls the development of TEX, but it also controls the development of hair follicles. And nude mice are actually null for FOXN1, and that's why they don't have hair. So the deletion of top 1 should be happening in these 2 cell types, but what we observe in our model that there was no defect in the skin, and these mice had proper hair. And the size was smaller early in life, but they picked up and the weight was similar in later half of the life. We analyzed the tissue morphology and through histological analysis, and we didn't observe any major changes in skin. So the defect is majorly in thymic epithelial cells, and probably that's the reason why we don't have thymus. Now, in nude mice where thymus is not there, as I mentioned, there are thymic rudiments. Literature suggests that there is thymic rudiment, and these thymic rudiments can be identified by the presence of thymic epithelial progenitors, which basically give rise to your CTEX and MTEX. And when we analyzed these TEPs, bipotent thymic epithelial progenitors in knockout model, we did not observe these TEPs. We are still analyzing the embryonic data to understand if we find TEPs in the embryo, and hopefully when I talk to you next time, we will have that data. Obviously, we didn't observe any TEC population in knockout since there was no thymus, and we think that there is no thymic rudiment as well, so you don't have, there is no major TEC population. Now, the major question we had at this time was, what is the mechanism? Because, we see that thymus is not developing and it has not been reported in the literature. We try to search in Jiva's data also that if there is any role of top 1 in certain diseases where thymus is not there and this gene doesn't light up. So we thought maybe what top 1 is doing is helping in development of these TECs from bipotent progenitors. So ideal situation will be that you take tissue from knockout and analyze these cells, but we don't have tissue actually in the knockout. So we switched to het model, and we are right now developing ways so that we can analyze these bipotent TEC progenitors. In the meantime, what we observed was that these TEPs are very similar to postnatal CTEX. So their gene expression profile overlaps with the CTEX, and we thought maybe we use that as a model to understand, are there major changes in transcription of genes which are required for thymus development. Here I'm showing you a comparison of CTEC versus MTEC, and you can see that your CTEC express bona fide markers for CTEX, and many genes which are involved in thymus development, they're highlighted in red, are actually expressed in CTEX. So CTEX can serve as a good model, and when we do this as and probably that is the reason your het mice have thymus. Most importantly, we checked the target gene for one of the master transcription factor for thymus development, FOXN1, and all the genes which are FOXN1 dependent were downregulated in het mice. So this data suggests that probably TOP1 is driving the expression of transcription factors which control thymus development. Maybe it is doing due to the fact, but ultimate result will be there if we analyze BTPs, and probably we'll do that soon. Now, if we look into the periphery, we should not have T cells in these mice, but surprisingly, what we find is there is obviously strong lymphopenia, there is less number of T cells, but there is a sizable proportion of T cells at 6 weeks of age. And T cells can be divided into gamma delta versus alpha beta based on TCR. And what we observe here is alpha beta T cells go down versus gamma delta T cells go up in this mice. Now, we were wondering that why we are observing these alpha beta T cells. So I'm not showing you the data, but if you analyze 2 weeks old mice, this data is coming from 6 weeks old mice, but in 2 weeks old mice, we don't observe these T cells. So this suggested probably these cells are, some cells are basically there in the 2 weeks old mice, and those are expanding. So how do we understand that? So what we thought was that if, is there any difference between the T cells which are present in knockout versus wild type? So one of the ways to understand that will be that we can analyze T cells coming from wild type and knockout mice through RNA-seq and see if there are major changes. And that's what we did. We analyzed CD4 T cells from wild type and knockout. These are splenic T cells. And as you can appreciate here, that many genes which are expressed in knockout T cells. So these, one thing I'd like to mention, deletion is not happening in T cells. Deletion is happening actually in text. But still, a lot of genes which are signature genes for effector population, they are up. These cells are proliferating, and many chemokine receptors were also up. And if we analyze, the effector signature is up in knockout. And we confirm that by flow cytometry. And indeed, if you can see on right side of the plot, major cells which are present in knockout mice are actually effector. These cells were expanding, so they had oligoclonal expansion, and this we analyzed through checking the CDR3 region, and you can see there is lesser diversity in the knockout, which is understandable, and CDR3 length goes down. And most importantly, in these mice, when we analyze the peripheral tissue, in some of the tissues like lung and rectal gland, there is pathology. And you can see that there is infiltration of T cells. So this is kind of surprising for us as well, because what we are observing here is a lymphopenia kind of situation, but these mice probably has some level of autoimmunity going on. So to summarize the talk, what we observe here is that TOP1 is probably controlling the expression of thymus development pathway genes, and that's how you have normal text, but in knockout, that is not happening. Now, I'll just take 1 more minute and wrap up this presentation. So what happens if you delete TOP1 in T cells, which we can do by using CD4-CRE, as I mentioned earlier. So we did single-cell RNA-seq from the T cells which are present in thymus in wild-type and knockout mice, and you can see that there are no larger changes. But if you carefully look, your DP1 cells are increasing. DP2, numerically, they don't change. But if you see, immature DPs are increasing, but mature DPs are decreasing. DP3s are going down, and this change is further translated to CD4 and CD8 T cells. And if you want to know more what happens, why this happens, you can visit the poster by one of my students, Prerna. And another student is presenting a poster, Harshdeep Kaur, on how TOP1 controls gene expression in macrophages. And with this, I'll end my presentation. I'll acknowledge my lab. We have a wonderful set of people, very driven. And this particular piece of work was led by Amit with the help of Pallavi, Harshdeep, and Prerna. Mark Zilka from UNC donated this top 1 flox mice to me, and that's how I could conduct this study, and all the funding sources for generous resources which we have in the lab. And thank you all for the patience, and I'll be happy to take questions.
Thank you, Kushagra. Excellent timing, good work. Giridhari?
Yeah, Sagra, wonderful talk. I'm wondering that if you have top 1 deficient mice, what about the hematopoietic progenitors? Are they okay, or they have some defect?
So in this particular strain, we have not done that, but in the strain where we are deleting top 1 in CD4 T cells, we have analyzed, and we don't see any major changes there. So,
and then what about B cell compartment?
In the mice,
these mice, yeah.
In these mice, it's perfectly fine. B cell compartment is fine. Total numbers are fine. We are still trying to see if they produce different antibody. Yeah.
All right. So you showed the skin pathology that they were normal, but what about if you give a small trigger, inflammation, and then how they respond?
Yeah, those kind of experiments are next set of experiments. Yes, Shilpa.
Yeah, so I just wanted to know in your knockouts, so when you check the peripheral CD4 T cells, so there is a drop in the alpha beta TCR T cells, but have you checked the FOXP3 positive cell regulatory T cells compartment? Is there any change in the regulatory T cell compartment as well?
So that doesn't change much. So the population is very small. We see hardly any cell which are FOXP3 positive in these mice. So most of the cells are basically your classical Th1 kind of cells which are proliferating. We don't observe so many of FOXP3 positive cells.
And your CD4 specific knockout or T cell specific knockout, so you see the similar pathology?
Yes. So I didn't show you the data, but what we observed, you see cells in thymus, but they don't migrate to the periphery. So pretty similar lymphopenia is there in that mice, which is still surprising for us, and we are trying to understand why that happens. One of the hint we have is probably the maturation of these T cells is affected. So you see T cells in thymus, but if you analyze maturation markers, they are still immature, and that's why probably they are not migrating.
Thank you.
Professor Nandi?
Yeah, good talk, Kushagra. Did you say that early on there are peripheral T cells, but by 8 weeks they go away?
No, other way around. So early we don't see, but later we see. So we think that it's lymphopenia mediated proliferation.
Right.
So early in life, some cells seed in, and since there is a lot of immunological space available, they basically start proliferating, and probably they are seeing some antigens. That's why it's probably TCR mediated proliferation.
Have you looked at activation status of those cells?
So if you see CD44 is very high on those cells, so I'm imagining these cells are basically effector, so they are activated. We have not done specific assays for that, but the data suggests that.
Okay, I think we'll close the discussion. Thanks again, Kushagra, and he's here fighting for during lunchtime. If there are more questions, Let's move on to the next 2 talks. My co-chair will take over.
Yeah, the next speaker is Subir Biswas. Subir Biswas has done his postdoctoral training at Cancer Center, Moffitt, Florida. And then he has contributed to unveiling the importance of spontaneous anti-tumor humoral responses in epithelial malignancies and mechanism of immunosuppressions. He has published in very reputed journals like Nature, Science, and Immunity. So over to Subir Biswas.
Yeah, good morning, everyone. First of all, thanks to the organizers for inviting me and also for keeping the antibody structure in the logo of IMMUNOCON 2024, despite of so many T-cell lectures. So, yeah. Okay. So this study is actually, I have to go back to some of the previous studies that I have done to understand why we were interested to target. So this is one of the first study where we tried to target intracellular antigen by an antibody, and there is no antibody available so far in the clinic where people have tried to target intracellular antigen or any molecule by an antibody. So these are my disclosure. So now the field recently has appreciated, alongside of the T cells or the cellular immunity, the humoral immune response is equally important in controlling the tumor growth in many epithelial malignancies. Now, as all of you probably know, that antibodies in human as well as in mice, we have 5 classes of antibodies, and among these 5, IgG, E, and A are class-switched antibodies. So when there is an infection or also there is a maturation of B-cell mediated immune response against a tumor antigen, this is a class switching event. So in an earlier study that we published in 2021, we were surprised to find that IgA and not IgG is the most dominant class of antibody in high-grade serous ovarian cancer. We analyzed 534 different cohorts of high-grade serous ovarian cancer, and IgA was the most dominant one. And that was supported by TCGA, and it was same in a similar microenvironment, in a similar cancer in human, which is endometrial cancer, having a similar mucosal microenvironment. But this is not the striking thing that we only observed. Rather, the most striking observation was that the epithelial cancer cells characterized by the EpCAM positive CD45 negative, they were coated by non-specific and non-specific antibody, non-antigen specific antibody, and we were very surprised because these antibodies do not have any specificity, but they were still coating the epithelial cancer cells, and it was not happening with other isotypes. So if we go back to the basics of the IgA biology, IgA can be a monomer or a dimer, and when IgA is a dimer, a small chain called as J chain, it holds the 2 Fc region together and makes it a dimer. Now, what happens in the mucosal epithelium, there is a receptor, polymeric immunoglobulin receptor, and this receptor is expressed on the basolateral surface of mucosal epithelium. And dimeric IgA with their J chain binds with this PIGAR and goes inside the cell with a fragment of PIGAR known as secretory component. Then they come out through the other side of the cell known as the apical surface of the cell. And this is how they clear viruses in the gut. Now, we were curious and we ran Western blot, and you can appreciate that quasi-universal expression of PIGAR, almost virtually all lysates we analyzed for high-grade serous ovarian cancer, and then we expanded this also in epithelial endometrial cancer. Now, what we did next, we took a cell line, OVCA3, that expresses PIGAR on their surface. We labeled non-antigen specific IgA with a fluorochrome, and we incubated that with that cell. And as you can see, over time, the antibodies first binding and getting accumulated inside the cell. Now, this doesn't happen when we use a similar thing, but the antibody is in IgG backbone. Remember, all these antibodies do not have any known specificity for any tumor antigen. Okay, and then when we cleaved the Fc region of the antibody, it was not happening with pepsindization. As well as when we ablated PIGAR by CRISPR, it was not happening. So it is a very PIGAR Fc region phenomenon that we observe. We confirmed that this is a transcytosis because we found the secretory component outside of the cell in the conditioned media by mass spec, and where we observed throughout the length of the secretory component, it was found only when the cells were treated with IgA but not with IgG, or when we blocked with the secretion blocker brefildin A and workmannin, we didn't see secretory component outside of the cell. We further confirmed by mass spec. Now, we were very interested to expand this work for other cancers and how we can utilize this phenomenon. So when we looked into the TCGA data for mRNA expression of PIGAR, you can see there is a clear dichotomy between the epithelial malignancies and non-epithelial cancers like lymphoma, sarcoma, melanoma. There's a very less expression of PIGAR in these non-epithelial malignancies, while in all epithelial cancers, there's a high expression of PIGAR, with only exception of diffuse large B-cell lymphoma. So we thought, can we use this system of PIGAR-expressed tumors and we can we generate antibodies that can target inside tumor cell. So for that, we made this system. So we produced recombinant antibodies using published VH and VL pair sequence, and we put that in human IgA as well as human IgG4 backbone. And we also transduced OFCA3 cells with a fusion protein that either has a wild type or the mutated version of the KRAS G12D mutation with mCherry. Now you can see that when we label this antibody and incubated with these transduced cells, the red color is the mCherry and the green is the antibody. You can see over time the antibody is going inside the cell, and there's a dramatic change in the pattern of expression of the mCherry fused with the KRAS. So what we thought, probably this antibody is going inside the cell and hauling the mutated protein outside of the cell. But this is not happening when the cell line was overexpressed, transduced with the wild type version of the same protein. And also when the antibody is in the IgG4 backbone. In the left side, you can still see the antibodies going inside the cell because it is a IgA1 which will go inside the cell because the cell line is PIGR positive. But the IgG4 version antibody is not at all going inside the cell, which we knew from our earlier publication. And then we confirmed that by mass spec that it is truly going outside of the cell, not only by this confocal, but the mutated protein is outside of the cell. We confirmed that there is not a true polarity of the cells, but still there is a polarity like the mucosal epithelium that when we are treating from one side, the mutated protein was found on the bottom chamber. So there could be a polarity of this event. Now we know that mutant KRAS is a protein that goes from the inner membrane, inner plasma membrane side inside the cells through the recycling endosome and comes back to the membrane. But the IgA goes, like the transcytosis event goes through the early and late endosome. So as you can see at the basal level, this protein, the mutant KRAS protein, is in the, if you see the upper blot, it's mainly in the recycling endosome compartment when we IP'd with RAB8A and 11A. But when we treated with the IgA antibody version, you can see there is a dramatic change in the localization from the recycling endosome compartments to the early and late endosome. And that was not happening when we used the same antibody, but in the IgG4 version. Then we used RAG1 knockout mice, and we utilized these transduced OFCA3 cells, and we injected intratumorally these antibodies, and you can see there is a dramatic decrease in the tumor growth in the mice where we injected the IgA1 version, but not the IgG4 version of the antibody. You may be curious that why the irrelevant IgA that do not have any specificities also decreasing the tumor growth. For that, we have to go back to the earlier study where we have shown that treatment with the non-antigen specific IgA in PIGR positive cells, it also upregulates interferon receptors, as well as it dampens RAS pathway, it dephosphorylates ERK1, 2, and thereby it controls tumor growth. So it was not surprising for us, and we confirmed that the antibody is truly specific because you can see there is no difference between the irrelevant IgA and the mutation-specific IgA on the right side. To make the observation clinically more relevant, we use the similar experiment, but we use NSG mice in this case, where there is no functional myeloid cells or NK cells even. And we also utilized the new small molecule inhibitor that is under development is MRTX1133, which is a small molecule inhibitor for G12D mutation of KRAS. And we observed that our antibody is doing way better when we have injected both the compound and the antibody at same temporal points, like every 4 days. But this compound is supposed to be injected daily. So we also did that, and still our antibody is doing better, although very similar. Now, what I have shown is a transduced cell line that already has an intrinsic wild type KRAS. So we also ran a lung tumor microarray, and you can see PIGR is also expressed universally there, and it is also heavily coated by IgA but not by IgG. So we utilized two cell lines, A427 and SKLU1, that has intrinsic G12D mutation of KRAS. And you can see that change in the decrease in the tumor growth is even greater than in the OVCA3 cells that we transduced ectopically. So similar observation was found when we injected daily MRTX1133. Still, our antibody is doing way better. Now, someone can argue that this mutation is very close to the membrane, so that somehow the antibody is accessing a mutated KRAS in just beneath the plasma membrane, or maybe from a dying necrotic cell. So we expanded this to another mutation which is deeper into the cytoplasm, which is IDH1 mutation R132H. So we purchased syngeneic cell lines, isogenic cell lines with IDH mutation on wild type. And we also made recombinant antibody, IgA antibody, against this mutation. Now, you can see, although it is significant, but it was not the dramatic growth reduction when we used. But our surprise was more to observe that this is also showing no reduction for the irrelevant IgA, non-antigen specific IgA that we always observe in other cancers. So we were so surprised, we immediately checked the expression of PIGR, and we observed that this is one of the few cell line that doesn't have a huge PIGR expression. That's why we were not observing the growth reduction by irrelevant IgA. So we overexpressed it. And now you can see we rescued the effect of irrelevant IgA as well as the reduction of the tumor growth by the mutation-specific IgA was huge. So this confirms that it is not only for the mutations that are very close to the membrane, but also it can target something deeper into the cytoplasm. So cutting the long story into short, so virtually we can target anything inside the cells using dimeric IgA that is specific for a mutation. And now we are working on also targeting something inside subcellular organelles, whether we can do that. And also we are trying to make antibodies in other backbones, for instance in IgG, because making dimeric IgA at large scale is difficult, and the half-life of IgA is less than IgG. So those things are going on. So these are the conclusion. We have published this in Immunity already. So thank you so much.
Thank you, Subir, for keeping the time. We can take 2 questions, please. One question.
Yeah, hello for the nice talk.
Thank you.
And I actually had 2 questions. So my first question is regarding the coating of these IgA antibodies. Do you also see that in immunologically cold cancers, like, for example, prostate cancer?
Prostate cancer, very less, but in bladder cancer, huge. So yes, I mean, it differs, and also not only IgA, we also see IgG and sometimes IgE also. So it depends on the location of the tumor and also the subtype of the tumor.
The targeting, is it specific to mutated proteins, that is point mutation induced cryptic protein, or can it also target those proteins that arise due to like structural events? Or
yes, we can. In the Nature paper, we have shown targets that are otherwise self proteins but overexpressed in the tumor microenvironment.
Yeah, thank you.
Yeah.
Yeah, Jyoti has a question.
Madhu. Oh, Jyoti.
So with something different approach, it's actually nice to see than regular standard T methodologies or B cell methodologies. So I would have you given a thought can there be some interference through other antibody fractions while you are focusing more on IgA? Means, of course, in few cancers, as you mentioned, but will there be any, say, IgG or IgD related any interference? Do you anticipate that?
Yes, that's a fantastic question. So the observation that we found in ovarian cancer and endometrial cancer is only true for those 2 cancers. But this mechanism of targeting something intracellular needs the binding of the antibody with PIGR. Now, yes, it is possible with IgM, but you need to make pentamer of IgM. Otherwise, it won't bind. IgA, at least you can make it dimer. Both is challenging. So what we are trying to do now is to make chimeric IgG antibodies with a PIGR binding site. as well as to make other approaches, like some of the, I cannot tell those names, but there are some receptors that also binds with IgG antibodies, and they may also have some, similar utility in this type of immunotherapy.
Thank you, Dr. Subir.
Thank you so much.
From ACTREC, one of the leading cancer institutes of the country. Thank you so much.
Thank you so much.
The next speaker is Santosh Kumar. He is from CCMB. He would be presenting a talk on the initial events of inhibitory signaling by killer cell, Ig-like receptor in human natural killer cells. A brief information about him is he has done his PhD from NCBS and on amyloid formation, and he has done his postdoctoral at NIH on NK cells, and his second postdoc he has done Stanford University on chronic fatigue syndrome. Over to you.
Okay. Hello, everyone. Yeah. So I'm going to talk about the initial events of inhibitory signaling by killer cell Ig-like receptor, KIR, in human NK cells. So NK cells express many receptors that can activate them. Upon binding to the cognate ligands on the target cell, activating receptors accumulate at the resulting NK cell immunological synapse. And signaling by them induces the polarization of lytic granules towards the synapse and the subsequent degranulation. That is the exocytosis of the content of the lytic granules into the synaptic cleft. And this can lead to killing of the target cell. Activated NK cells also secrete many cytokines and chemokines, and thereby they can control the activity of other immune cells. So the activation of NK cell is tightly controlled by class 1 MSC-specific inhibitory receptors. In human NK cells, there are two major classes of MSC1-specific inhibitory receptors. One of them includes the members of killer cell Ig-like receptor, KIR, that bind to the MSC molecule HLA-A, B, or C. And other class involves NKG2A, that is specific to HLA-E. So inhibitory receptors of both these classes have immunoreceptor tyrosine-based inhibitory motif, item, in their cytosolic tail. So we work on inhibitory KIR, particularly KIR2DL1. It has two Ig-like extracellular domains that are oriented at an angle of around 55 degrees, and it has two item motifs in the cytosolic tail. So the unligated KIR is maintained in a state where the item motifs are not phosphorylated. Upon binding to the cognate HLA ligand on the target cell, KIR accumulates at the resulting NK target inhibitory synapses, and it becomes item phosphorylated. Phosphorylated items recruit and activate the protein tyrosine phosphatase SHIP1, which in turn dephosphorylates the guanine nucleotide exchange factor VAV1. And through an unknown mechanism, the protein kinase c-Abl is recruited here. I don't know what's happening. c-Abl is, okay, that's not working. c-Abl is recruited here. This stopped working, looks like. Okay, let's look. I can handle with pointer. But how about this? This is also not working. So through an unknown mechanism, this protein kinase c-Abl is recruited here. Okay. How can I go back? Okay. And this c-Abl phosphorylates the small adapter protein CRAC. So both VAV1 dephosphorylation and CRAC phosphorylation contribute to the inhibition of NK cell activation. So here, the inhibition by KIR is going to block NK cell activation. So the target cell is not going to be killed. It is going to be spared. So in our body, most of the cells, express class 1 MSC molecules, and therefore they all have the potential to block the activation of NK cells. So in this talk, I'm going to discuss about two interlinked events that trigger the inhibitory signaling of KIR. One of them is about electrostatic interaction of the cytosolic tail of KIR with the plasma membrane, and this interaction restrains the phosphorylation of ITIM motifs. And the other is about the potassium-induced signaling of KIR, wherein it appears that an influx of potassium from the cell exterior in the NK cell disrupts this electrostatic interaction and thereby triggers the inhibitory signaling of KIR. And these insights have largely come from the work of Sitanshu Kumar Sarangi, who is a PhD student in the lab who just finished his PhD. So let's get started with the first one. So the cytosolic tail of KIR is an intrinsically disordered region in solution. And it is a neutrally charged polyampholite. That means that it has equal number of acidic and basic residues. And if you look at it little closely, you will find that there is the basic and acidic residues are linearly segregated. The juxtamembrane region is overall basic, and the intervening sequence is acidic in nature. Our experiment with purified cytosolic tail of the KIR and lipid vesicles indicated that the cytosolic tail of KIR, despite lacking a net charge, interacts electrostatically with the plasma membrane. To test this interaction in resting NK cells, we used a FRET-based assay. We expressed full-length KIR tagged with TFP at the cytosolic end, and you can see that KIR is nicely expressed at the plasma membrane. And we membrane labeled these cells with rhodamine di R18. So here TFP acts as the FRET donor and R18 acts as a FRET acceptor. So now you can see that upon R18 photobleaching, that is the acceptor photobleaching, there is a substantial increase in the fluorescence intensity of TFP, that is the FRET donor, indicating a FRET signal. We did not get any appreciable FRET signal with KIR mutants where the selected basic residues in the cytosolic tail were mutated either to alanine or serine. So these results told us that the cytosolic tail of KIR interacts electrostatically with the inner leaflet of the plasma membrane. So let's have a look on NK target inhibitory synapse. So this is an NK cell which has been membrane labeled with R18, and it expresses full-length KIR tagged with TFP at the cytosolic end. And it has made an inhibitory synapse with a target cell that expresses the cognate HLA-C ligand. Now you can see that, as seen before, KIR accumulates at the inhibitory synapse. And this is the place where KIR functions and signals locally to exert local inhibition. And now you can see that the FRET signal remains intact at the extrasynaptic region, indicating that the cytosolic tail of KIR remains bound to the plasma membrane at the extrasynaptic region. However, the FRET signal is lost at the synaptic region, suggesting that KIR dislodges its tail from the plasma membrane locally at inhibitory synapse. So these experiments, along with many other experiments which I have not shown here, suggest the following model for tyrosine phosphorylation and inhibitory signaling of KIR. So in resting NK cell, the cytosolic tail of KIR is electrostatically bound to the plasma membrane. And due to this binding, the tyrosine residues are inserted in the lipid bilayer. They are not accessible to the kinase. And that's how the spontaneous phosphorylation of KIR is restrained. And HLA-C binding induces KIR to dislodge its tail from the plasma membrane, and now the tyrosine residues become accessible for signaling. So next, we tested the role of different physiological ions, and we found that the tail dislodging and inhibitory function of KIR are dependent on potassium ion in the extracellular medium. So here you can see that, as expected, KIR dislodges its tail from the plasma membrane at inhibitory synapse that are formed at physiological level of external potassium. However, it fails to do so when the external potassium concentration is reduced to 0.5 millimolar or less. Similarly, KIR exerts a strong inhibition at physiological level of external potassium. However, it largely fails to function when the external potassium concentration is reduced to 0.5 millimolar or less. So these results told us that the tail dislodging and inhibitory function of KIR requires potassium ions in the external milieu. So the requirement of external potassium for the tail dislodging that occurs in the cytosolic phase of the plasma membrane, hint toward a potassium influx from the cell exterior. And this inward rectifier potassium channel is well suited for doing that job, because under certain circumstances, these channels favor inward potassium current over the outward potassium current. So there are 18 members in this family, and we screened some of them, and we found that this KCNJ12 does this job. So here you can see that, as expected, KIR accumulates at the inhibitory synapse, and now we see that the KCNJ12 channel also accumulates here. And here is an NK cell that has made conjugate with a vesicle that has peptide HLA-C complex attached to it. And now you can see that peptide HLA-C complex on a lipid vesicle is sufficient for the synaptic accumulation of both KIR and the KCNJ12 channel, suggesting that the KCNJ12 channel accumulates at NK target inhibitory synapses along with KIR, probably through association with KIR, but we have not yet checked the direct association between them. So here we have monitored the intracellular potassium level in NK cells at activation synapse, where KIR is unligated, and in inhibitory synapse, where KIR is ligated with a cognate ligand, the HLA-C ligand. And now here you can see that the level of intracellular potassium in NK cell at inhibitory synapse is higher. And we don't see such an increase in the intracellular potassium level with the KCNJ12 knockout NK cells. So this told us that the potassium influx that occurs in the NK cell at inhibitory synapse is mediated by this KCNJ12 channel, that is an inward rectifier potassium channel. So here we have tested the effect of the inhibitory function of KIR in KCNJ12 knockout cells. So as expected, KIR dislodges its tail from the plasma membrane at inhibitory synapse in wild-type NK cell. However, it fails to do so in KCNJ12 knockout NK cells. Similarly, KIR exerts inhibition of NK cell activation in wild-type NK cells, but it largely fails to function in KCNJ12 knockout NK cells. And these experiments are done on a suboptimal concentration of external potassium, that is 0.5 millimolar. And here we know that KIR fails to, at this concentration of external potassium, KIR fails to dislodge its tail from the plasma membrane, and also it largely fails to function. However, the overexpression of KCNJ12 restores both the tail dislodging and inhibitory function of KIR.
Kumar,
So,
if you conclude, please.
Yeah, just one minute. So how does potassium promote KIR tail dislodging? So in our in vitro experiments, treatment with sodium chloride induces dissociation of KIR cytosolic tail from lipid vesicles. And sodium can also do that, but it does at very high concentration, that is 1 molar, that is non-physiological. And our calorimetry experiments suggested that potassium binds to the cytosolic tail of KIR, doesn't bind to the cytosolic tail of KIR, but it does bind to anionic lipid vesicles. So based on these results, it appears that the binding of potassium ion to the membrane anionic lipid disrupts the electrostatic interaction of the cytosolic tail of KIR with the plasma membrane. And to understand which all steps of KIR signaling is dependent on external potassium, we studied a mutant KIR where whose cytosolic tail cannot bind to the plasma membrane. So unlike the wild type KIR, this mutant KIR exert inhibition more or less independently of the external potassium, suggesting that the only function of potassium is to dislodge the tail from the plasma membrane, and the downstream events of the KIR signaling are independent of external potassium.
Kumar, time up.
Yes. So this is the model that we propose. KCNJ12 channel accumulates at inhibitory synapse along with KIR. It provides potassium from outside to inside that triggers inhibitory signaling of KIR that blocks NK cell activation. And finally, this is my final slide. So our study provides a few points of a broader relevance. Number one is the polyimpholytic IDR, that intrinsically disorder protein, despite lacking a net charge, can mediate electrostatic interaction, provided the acidic and basic residues are linearly segregated. The second point is that our study provides a new cellular function of potassium in controlling electrostatic protein lipid interaction. So calcium is known since long time as an ionic signal for lymphocyte activation. So our study suggests that potassium can be an ionic signal for lymphocyte inhibition. And finally, our results suggest that this KCNJ12 channel is a new checkpoint molecule in human NK cell, and this is extremely important because the monoclonal antibody against KIR has not been successful in cancer immunotherapy, and this channel can provide an alternative handle. So I thank people involved here and also the funding agency and my host institution, and thank you all for the patience. And now I'm happy to take questions.
2 questions, please. We can take 2 questions. Ravindra has 1 question. If there are no questions, sir.
Okay, looks like there are no questions. So
Thank you.
on behalf of my co-chair, Professor Patil, and myself, and all of you, thanks to all the speakers for keeping to time, excellent presentations. All the very best for all these young investigators. Thank you.
Yeah. Thank you, sir. I request the chairs and the speakers to assemble on the stage for a group picture.
Yes, got it. Yes, I think so. Yes. I'll just.
Thank you once again. Now we move on to our next session, the third special lecture by N.K. Ganguly, Department of Biotechnology and Research, Sir Gangaram Hospital, New Delhi. As Professor N.K. Ganguly could not be physically present, so this is a recorded lecture, and the chair of the session is Dr. B. Ravindran from ILS Bhubaneswar.
Just do it from here. Yeah, sure. I have to introduce him, that's all.
So first, you can introduce. You can take 15 seconds.
Well, yesterday evening, Dipankar told me the boss is not coming. So that, in a nutshell, introduces Professor N.K. Ganguly to all of you. He probably started working on clinical immunology much before Indian Immunology Society started existing. So He has been everything for everybody. He was the Director General of the Indian Council of Medical Research for many years and had mentored a lot of people. He's officially affiliated to Sir Gangaram Hospital, but we know that he has been all over the place. That's about enough for introducing Professor N.K. Ganguly, and we will have a 15-minute presentation, a recorded one that Dipankar received yesterday from him. Apparently, he's not available for taking questions, so we'll not have a question answer session. And obviously, the photo op at the end of the session with chair and the speaker will also not be there. Thank you.
It's a real pleasure for all of us to have Professor Ganguly with us. Professor Ganguly is well known for his numerous contributions to Indian science. I would request Professor Ganguly to begin his talk. Thank you so much for being with us.
Thank you, Dipankar. It's a great pleasure to be here with this tremendous conference. I apologize that I could not be personally present because of unavoidable reason. So today I will be talking about precision medicine, which has come through the new immunotherapy. But in the immunotherapies, people don't respond. All of them don't respond. Only 30 to 40% of them respond. 60% don't respond. And these are very expensive. So people become poorer and poorer and are not able to complete the treatment. So we were looking for other markers which could have a better result. And we came into conclusion that the desmoporin 3, which is a transmembrane protein, is the molecule which is present on the surface of the some of the cancers, where it takes care of the secondary immune stimulus, which are done by the co-stimulatory molecules. So even if there is PDL or TIM3 or CCL4 or other substances which cause immune blocking, the DSLS3, they provide that particular linkage. And if you see that in this transmembrane protein, there are a lot of sensors and a lot of points where it could contact with the different cell system. So it can directly, without after the T3 signaling has happened, it can directly contact with the specific T cells, and a specific stimulus could come up with the antigens which are now the cells are able to present. So if we look at it, the DSL3 is a transmembrane protein, as I said. It can, it is p53 responsive gene. It is highly specific in the squamous cell, lung cell, than NSLC, but it is not there in the adenocarcinoma. 99% it is over there. So we differentiate between the squamous cells and non-squamous cells. So it could be diagnostic also. So it could be very good diagnostic marker. Predictive biomarker for Mycobacterium W therapy. Mycobacterium W, which was actually discovered by Dr. Talwar for the leprosy. We have done a lot of work on the cancer with a formulation which we have prepared. And this formulation is now in the market known as Mycidac, marketed by Cadila. And this is a unique formulation. Next, please. So this is 3 protein is produced in the endoplasmic reticulum and is transported to plasma membrane in the healthy individuals. But in rapidly dividing cells with cancer or amyloidosis, it is expressed in cytoplasm. So it is very strong in signaling arena. It was first identified in bladder cancer, but it is also expressed in the squamous cell NSLC, head and neck cancer, ovarian cancer, etc. It is normally, the FDA-approved technique is that it is normally detected through immunohistochemistry, as are the HER2 and the PDL1. So this is, as I was telling, that it is both diagnostic, it is prognostic, and it is predictive. It is differentiated with lung and squamous cell cancer. It is a better odds of progression of the disease, and it can also differentiate between good response and poor response. Next, please. So we looked at the different solid cancers, that how it is expressed. So out of 570, we found that 320 were positive, 61.9%. Range was 1 to 90%. If we move further, so if these are the cancers where it is expressed very high, the total predictive score is given later. There was no, it was not known that what it should be the percentage where the people will become more responsive. But we found that we could have a cutoff at 25% or 50%. 50% works better. It has a better prediction. Next, please. So it is given. It is in many cancers, as I had told you before, but it is not there in the pancreatic cancer. We didn't find it in pancreatic cancer, and in the prostate cancer also, it was very low. Next, please. So, let us see that whether those cells which are cancers which are expressing DSS3, they also express the TILs. There are TILs there which have a PDL1 expression. So if we see, we could categorize them into 3 pools. One where the DSC is positive alone, which is the blue bar, and 52% presented alone. There are places where the TIL was also positive, as well DSS3 was positive, tumor-infiltrating lymphocytes, and there were cells where only the tumor-infiltrating lymphocytes were there, but DSS3 was not there. And then there were subjects where didn't express DSS 3 at all. So here the selection happened that you could give the mycedec and people will respond where that TIL is not there. But where TIL and DSC is there, if you give nivolumab or pembrolizumab as well as the MW, the response will be much better and higher. Next, please. So this actually is an animal study, and this is a sarcoma tumor. And if we see the DSC positive cases, we see the survival is much better, and the progression-free is also much better than the DOCC. Where the desmopolin-3 was negative, there was no survival benefit. This is NIH-funded project. This was another animal study where we have a pancreatic cancer, where myeloma. Since pancreatic cancer doesn't express DSS3, the responses were poor, whereas in the myeloma we get very good response. So the next animal study shows the synergistic response with anti-PD-1 and GITR. That is the glucocorticoid induced tumor necrosis factor. And we see here that MW with anti-PD-1 works very, very good. Very good. Next, please. So here we can see that the FOX3P3 is reduced. The PD-1 positive cells are reduced. The DST, the TILs are increased in these subjects. 50% of CD8 positive cells increase. So the if you give MW, it blocks to the desmopolin-3 receptor, and the CD8 positive cells increase. So this is again a predictive marker. We have 221 randomized samples, and we can see then the squamous cell carcinoma, which has positive DSC, it has a huge survival rate. This particular trial was done by us. So and it is this chemotherapy was done in a randomized again controlled clinical trial, and we can see that the actual advantages over here, the grade of grade 4 and grade 3, the toxicity was very, very less, and in the head and neck cancer, the advantages were very high. About 8 to 25, 8 out of 25 survived in the negative ones, and 20 out of 25 survived in the positive ones. This is very significant. Here you could see that whether this was able to stop the reduce the tumor lesions, and we can see here that this has been very specific in the response, and this is actually the metastasis in the lungs, and we can see that metastasis is reduced, and a stable disease and no evidence of the disease was there. So this one again, we can see the specificity in the immunotherapy and monotherapy, and we can see that the active stage specific immunotherapy with the monotherapy was very, very advantageous. So the TLR2 agonist is the TLR2 agonist. When you give this, you find very nice response. So this one shows that the upper panel shows the lesions in the lung, liver, and adrenals, and the lower panel shows that after treatment, and you could see that lesions have disappeared after the treatment. This was a single case study. So this is the hazard ratio, and in the hazard ratio, if the CD8 and these cells are there, as well as the DSS3 expression, the hazard ratio was very, very favorable. And this were different kinds of cancer. The best results were available in the head and neck cancer, ovarian cancer, and the lung cancer. So here you could see that how the lesion, which was head and neck cancer, was almost disappeared after 11 weeks later. We also have another case where it was compagative. We have published this. The entire lesion disappeared there. So I am showing you now another study where I will give you the study design. Next slide, please. In the study design, we had given 3 shots of 2 to 3 weeks interval of the nivolumab into the NSLC3, which is allowed by FDA. We did microbiome, immunometabolome, proteome, genome study, sequencing studies. And we see that the one which, since it inhibits the AKT pathway, we saw a distinct advantage of the ones which were given nivolumab and were also desmoglein-3 positive. Next, please. And this is the survival, which has a huge advantage. So this one shows the TILs, that the ones which were responding, they had CD8 positive, Ki67 positive, that is activation marker positive. They had a CD8 the ganzyme B positive. These were responders about 80% plus positive, and the PDL1 was also positive in them. So this shows in the slides in the flow cytometry the blockade pathway, which includes proliferation effector of PD1 CD8 plus at Ki67 T cells. And in the responders, in the non-responders, the bad macrophages, monocytes were there, the monocyte 2 were there. So this one is a cytokine. These cytokines actually control AKT, the cytokine 23 and cytokine 21. These were raised in the responsive ones, And cytokine 18 was also positive, which comes with the aging. So most of these were aging patients. So that one, and of course, I2 was increased. Next, please. So they in the sequencing, there were two genes which are mutated. We know that desocolin 3 is responded by TP53. So we had a mutation which actually helped in the responder. And FGFR4, there are certain drugs which are being used in FGFR4, which also is a strong immunomodulator. Next, please. So when we looked at the microbiome, we found very interesting study. In the microbiome, we found that those who didn't respond had this was done through 16S sequencing. They had Escherichia coli and they have Klebsiella. And those who responded had Bifidobacter, Doria, and one of the numbered ones, which was and Mycophomonas. So this was very, very interesting and important. Next, please. And we see here that they had very strong link with the metabolomic pathways. Those who were responding with the, I will come to the metabolomic, which was important. And here the red ones are the ones, metabolic pathways, which were linked to the microbiome. And if these were there, cancer patient responded better. There's a slide which I have picked up from Atra. She is from North Carolina. And in this particular slide, she has the glucuronidase, the part, which is about 65 of them. And these are run by the microbiomes. And when we give the cancer drugs, they cleave the cancer drug. So the microbiome not only are beneficial as a immune modulator, they also, if they're bad ones, they can reduce the amount of drug which is available over there. So we have now a study which I'm not showing here because it is unpublished, that we have identified the good donors and these donor microbiomes are very, very specific, and we have given the microbiome to the patients, and we have got some very interesting results out of that. Next, please. So this I had already explained. So these are the ROCs, and if we could look at the ROCs, we can see that the combined ROCs are very, very good in our system. If we had DSST positive and we had given Nivolumab, and we have got the next piece, if we have got the good metabolites, and then these are the results you could see in the Kaplan Meier, that very nice things were done. If we, particularly the urea cycle, if we had urea cycle over there, urea and the combined. So this was the slide which I was showing, where we had a huge lesion, which ultimately disappeared after our treatment. So the work was done by Kriti Jain, my PhD student, and the oncologist was Shyam Agarwal. And we had the help from Dr. Amit Awasthi as well as Dr. Avatosh Das or Michael. Thank you very much.
Thank you so much, Professor Ganguly, for the wonderful talk. We really appreciate your time. What I will suggest is for people who are interested, can maybe write to Professor Ganguly and then get feedback from him. Is that okay, Professor Ganguly? Yeah, and we hope to see you. We hope to see you in person in Bangalore sometime soon. Okay, thank you again so much, sir. Thank you. We really appreciate. It's a real pleasure for all of us to have Professor Ganguly with us. Professor Ganguly is well known for his numerous contributions to Indian science. I would request Professor Ganguly.
I request Professor Nandi to thank Dr. Ravindran.
It's good enough. You did.
Now we move on to our next plenary lecture number 14 by Professor Sergio Lira, Precision Immunology Institute, Icahn School of Medicine at Mount Sinai, New York. I request Professor Geeta Rai BHU to now take over and guide us through the session.
So it gives me immense pleasure to introduce a friend and a remarkable researcher, Professor Sergio Lira. And can you go to the introduction slide, please, someone? So Dr. Sergio, who received his MD from the University Federal de Pernambuco, Brazil in 1982, and PhD in Physiology and Pharmacology from UC San Diego in 1988. And after his postdoctoral research at the Roche Institute of Developmental Biology, he held positions at BMS and Schering-Plough. So since then, he has been at the Icahn School of Medicine at Mount Sinai and currently serving as the Leon M. Harry B. Helmsley Professor of Immunology. So I would like to tell the audience that this is Sergio's first time at the IMMUNOCON platform, and I have enjoyed his talk in the past, and I can tell you he is a remarkable storyteller, which you will see yourself. So goes to you, Sergio.
Hello, thank you very much. Thank you, Geeta, for the nice introduction. Thank you, Dr. Nandi, for inviting me here to the meeting. And today I'm going to be telling you the story about inflammatory bowel disease. Our lab studies inflammatory bowel disease, and the title of this talk is "It Takes More Than Two to Tango." Of course, it takes more than two to tango. It takes music, it takes a maestro, it takes an audience, and this is a theme that I'm going to come back to during my talk. So, as I mentioned, we study inflammatory bowel diseases, which, as the name suggests, are diseases that affect the intestinal tract. There are two main diseases, inflammatory bowel diseases. One is called ulcerative colitis, in which the whole colon is affected, and it's very inflamed. And the other one is called Crohn's disease, which was incidentally discovered at Mount Sinai 100 years ago, almost 100 years ago. And this disease, you can have any part of the intestine being affected, both large intestines and the small intestine can be affected. The etiology, the cause for this is unknown. Until today, we don't know what causes this disease. It falls into the class of inflammatory, autoimmune, diseases, and a disease that affects millions of people worldwide, and it has a very severe morbidity. So a number of therapeutic approaches have been developed to treat this condition. And some of this was due to the fact that genetic studies have suggested that 200 gene loci have been associated with these conditions. So one of them, in fact, was a mutation affecting the IL-23 receptor signaling. A null mutant, patients carrying null mutants of these receptors, they are less susceptible to the development of IBD. So that was the initial trigger that prompted people to start thinking about IL-23 as an element contributing to inflammatory bowel disease. And there is something very interesting, is that despite the fact that we know a lot about this 200 genes and the impact on disease, what's happening to IBD is very similar to what happens to many other autoimmune inflammatory diseases in that the incidence of these conditions is increasing dramatically. And you can see this is the incidence for ulcerative colitis from the first time it was described in 1859 to now. And you can see that a very similar curve is occurring also in the so-called newly industrialized countries like India and Brazil and so on. So this suggests that in addition to these well-known genes and inflammatory processes, there is something out there that's contributing to the increased incidence of this condition, but we don't know what it is. It has been speculated that environmental factors are causing this, but we have absolutely no clue what are these environmental factors. And this is also true for MS, it's also true for type 1 diabetes, and so on and so on and so on. So I can tell you that we didn't set up to study environmental influences on IBD. What we were after were understanding essential mechanisms associated with IL-23 signaling, because as I mentioned to you, there was all this data linking IL-23 signaling with less susceptibility to IBD. IL-23, as many of you know, is a molecule that's produced by myeloid cells as consequence of inflammatory stimuli. And it's a dimeric protein, cytokine. There's two subunits, P19 and P40. And the expression of this molecule leads to activation of several gene programs in T cells, NK cells, innate cells of the ALC3s, and so on. And this is associated with production of a variety of cytokines such as IL-17, IL-22, interferon gamma, and so on. And this molecule has been associated not only with IBD, but also with psoriasis. And not surprisingly, therapies directed at blocking IL-23 function have been quite successful. In fact, IL-23 and IL-17 have been extraordinarily successful in treating patients with psoriasis. And IL-23 is now showing to be equally effective in treating patients with IBD. But despite the fact that this is happening, and we are super excited with this because this molecule was discovered at the end of the 90s, and a few years later now we have the molecule in the clinic treating people, we still don't really know how IL-23 is doing what it's doing, okay? And so that's what we were interested in understanding. We were interested in understanding how IL-23 causes, or if it causes and sustains disease. So there was something very intriguing about the biology of IL-23, and that's what I discussed with my postdoc, Lily Chen, when she came to the lab. I said, Lily, I know this from the time I was working at Schering-Plough, where IL-23 was discovered in one of the laboratories at DNX Research Institutes in California. What we know is that if we inject IL-23 into a mouse, okay, and I knew that from old time, or if you inject in the tail vein a DNA vector containing the P19 and P40 subunits of IL-23, what's going to happen is that in the course of 4 days, between days -4 and day 0 here, you can see that injection of a vector that does not have IL-23 doesn't do anything in terms of IL-23 levels, but within 4 days, only 4 days, the levels of IL-23, which are now transfected into hepatocytes, are massive in the bloodstream. You have very high levels of IL-23, and these levels are sustained up to day 35 for sure. So yes, we have IL-23 being produced in these animals, and what happens to these mice? So what happens is the following. Animals injected with vectors that contain no IL-23, the skin of the ear is absolutely normal. You can see it very well here, and very nice normal skin. However, the skin of a mouse that has been injected with this IL-23 vector is very inflamed. You can see very clear that the dermis and the epidermis are affected. There is increased proliferation of the epithelial cell layer here. You see the presence of the so-called Monroe abscesses, which are collections of neutrophils. You can see increased vascularity. This is a very different skin than this one here. And this resembles very much what one sees in psoriasis in humans. So we had disease in the skin. But what was the paradoxical finding? The paradoxical finding is that there was absolutely no disease in the gut. And I've been telling you that IL-23 is important in the gut because IL-23 receptor mutations are associated with less susceptibility, because IL-23 expression is found within these inflammatory lesions in the gut of humans, and because a number of other experimental studies have supported the role of IL-23 in this process. Yet, high levels of IL-23 in the gut don't do anything. So I said, Lily, perhaps we need to do this slightly different and try to localize the expression of IL-23 to the gut. And she went on to produce mice, engineer mice, in which IL-23 was placed in the rosal locus here, and this was flanked by, there's a cassette, a stop cassette in front of the P19, P40 subunits. And these mice were crossed with mice that expressed CX3CR1. This was placed in the CX3CR1 locus. Why? Because the cells that express IL-23 express this chemokine receptor here for a molecule called fractalkine. It used to be called fractalkine. So this is what we decided to engineer, a mouse in which we could prompt the expression of IL-23 whenever we wished. And evidently we got mice. These were the cells in which they had YFP. We could see that there was expression, but not dramatically high. Low level expression, but still there. So Lily got very excited, I got very excited. We went on to do the experiments. And she comes in and she injects the mice with different amounts, with a given amount of tamoxifen at different time points. And then she opens the mice and she comes to my office and she's ready to commit harakiri like Vijay was telling you about. Absolutely nothing here. Nothing. Nothing. And, this is very disappointing, as many of you know, because you put a lot of work in trying to make something work and nothing. So she said, but I think I want to try something. I said, okay, what is that? She said, well, I want to put it in the food, because I think that maybe, we're not really covering it and so on. I said, I don't believe that, because you injected so much tamoxifen in these mice that the screen recombinase has fluxed everything. So what she did then was she came up with this concept of giving the mice food for different lengths of time. And, what I told her to do, and this is very important, I said, Lily, go to the vendor and get the vendor to put tamoxifen on the food, okay? She said, fine. So she went ahead and she did that. First treatment, 7 days, one cohort of mice, nothing, nothing. She said, well, I want to insist, I want to persevere. And she gave her mice the rest of 1 week. And what she did was, she came back to me and she was super excited. She said, look, the guts are inflamed. The large intestine of these mice are inflamed. And this was the first time that they had seen that. You can see all these mononuclear cells here. The epithelium is intact. There is no transmural infiltration, but there is inflammation there. There's no doubt about that. There is an inflammatory process happening there. So we go out, have Chinese dinner. The lab is very excited, and all that jazz. And you say, okay, now let's open a mouse at day 42, because by this point, the mice probably progressed. Now we have really serious inflammation in the gut. And she goes and she opens the mice at day 42 and, well, not the same mice, evidently, another cohort, because these mice were dead. So nothing. So something had happened here. These mice that had received this tamoxifen for this time point here, 7 days, they had resolved the inflammation. And of course, we were excited and not excited. Excited because we said, well, this is what we see in humans. We see that in humans we have flares and then it resolves. And what happens then if you look at another round of treatment? And then that's what we saw. And that was another Chinese dinner, because here we had now a situation where we had the whole epithelium had sloughed off. The cells were dead. This is like ulcerative colitis in humans. In the ulcerative colitis, the inflammation is restricted to the mucosa and submucosa. There is no transmural, no inflammation through the muscle layer. There are no granulomas. This is a typical example of ulcerative, because it's ulcerating colitis. So very excited. And again, the prediction was that you stop the treatment, the disease is going to disappear, and that's exactly what we got. So this was super exciting. Now we had a model where we could, trigger inflammation. We could trigger very intense flares of inflammation. And then we stopped the treatment and the mice would resolve. So dream come true. We had a system that we could study this molecule in detail. And what happened was that essentially the question became, all right, but if IL-23 is being turned on, how can we have these flares? And how can we have resolution? So we start to measure IL-23 to see if there are variations in the level of expression of IL-23. And essentially, there were no differences in IL-23 expression. So this could not be variation in IL-23. This was essentially due to the fact that something else was contributing. We didn't know what it was. So we went through this exercise of trying to figure out what could be it. And evidently, the most logical and the simplest explanation to what we had was what? What we were giving the mice tamoxifen, right? So we figured, well, tamoxifen is an estrogen receptor antagonist. We are giving the mice, this is a molecule that may not have neutral activities. It may have activities of its own. So could it be that this is tamoxifen? Little problem here. What's the little problem? I told Lily to put tamoxifen in our diet. And I said, how are we going to test this now? So I went to her and said, Lily, how are we going to test this now? And then she started to smile. And I said, why are you smiling? She said, because I didn't put tamoxifen in our diet. And I said, what did you do? She said, I bought another diet with tamoxifen. And I wanted that commit harakiri. And I said, this is another variable that we introduced in the system. But hey, by chance, by pure chance, we have not very good condition to try to test the role of tamoxifen. So you go back to these people from whom you bought this new diet and tell them to sell you the same diet, but without tamoxifen. Because if it is tamoxifen, we're going to figure it out. So the diet that she bought is called diet 2019. And some of you that have mice here know that this is something that you give to mothers to help them to lactate. Has more fat than normal. And she put this tamoxifen. This is the diet that we use in our facility. So 1 cycle, 2 cycles, 3 cycles, boom. Inflammation, edema, the whole bit. So the experiment is to give the mice now, to prime them, 2019, and then she did that, and the mice were sick. And we were like, holy cow, to use an expression in the West that's not so popular here. But then 2019 was causing the switch between one diet and the other, was causing disease. And I said, hallelujah, we have something so hot here. Another Chinese dinner, almost got bankrupt in my lab because of all these dinners. But very happy to have that. So the unhappiness happens when you start to write grants for the NIH, because you say, okay, now I found this. I want to identify what in the diet is cholecystogenic. So I got reviews from our colleagues at the NIH, the study section, and they said, you are never going to figure this out. They gave me a horrible score, and I went into severe depression because I said, I have something so interesting here. Please, So I wrote a little bit of a rebuttal, and I tried to convince them that this was important and that we will try. So how do you go about that? I don't use anything very sophisticated in my analysis of my experiments. So what we did was we took a page from the people that do food allergy. And we got this diet, we macerated it, we put ethanol, and we heated it up. So we're trying to extract the lipids, which we thought were the most important component of this. And we extract the lipids, and we got the rest of the stuff. So when we did that, we got, that's the protocol here. So when we did that, the ethanol extract didn't cause anything. So we primed the mice, gave this ethanol extract with this stuff, the goodies, the fat and so on, nothing. But the aqueous extract caused disease. So we said, okay, this reduces the complexity a little bit. So it could be a sugar, it could be a protein. And evidently, we started thinking about, giving the mice different diets with different composition of sugars and different proteins and so on. And it just happened that Lily had already ordered some other diets. And she had a diet that was called diet 2018. 2019 refers, 19 refers to the percentage of protein in the diet. So it's 19% of that is protein. And she had a diet that had 18%. Okay, so I said, Lily, this is not going to work. Why are we doing that? Let's give 5%, 2%, 1%, 0% protein. And she said, I want to do the experiment. And I said, okay, go ahead and do the experiment. Always prove that, let the people do what they want to do. So this is 2019, okay? So that's the whole story. We got 1, 2, 3 cycles with 2019 and tamoxifen here without tamoxifen, disease, boom. And then the same thing, tamoxifen, and 2018. And then she comes to me and she says, this is what happened. We had no disease. I said, no, this is not possible. This is not possible. You're joking. This is not possible. It cannot be that 1% protein accounts for this. So I said, Lily, I'm going to take a vacation. When I come back, you tell me how this works, okay? So I went on a vacation, had a great time. I think I came here to India, in fact. Good point. People should come to India more often. So I got back and she had this biggest smile on her face. And I was like, oh my God, I knew that there was something good there. And this is a great moment for many of us who train people because, you know, when you see this kind of happiness in the faces of your trainees, you know that you're doing your job. You know, you're giving the people a chance to discover things. And that is so fantastic. So she came to me and she said, Sergio, this is how 218 look. And you know, many of you look at mouse pellets. This is how a mouse pellet looks like. And I said, okay, Lily, and what is the difference? And she said, this is how 2019 looks like. I said, what? She said, yes, you know, when I ordered this 2019, I asked them to mark the pellet so that I would know which is the diet that contains tamoxifen. Make my life easier. I said, Lily, you are telling me that the difference between these two diets, besides that 1%, is the color? She said, yes. And the color is the difference, right? So what is in the color? In the color, there is this thing called red 40 or allura red, okay? So all of you here probably never heard of this thing, okay? However, I would like to tell you that this is more present in your life than you ever imagined, okay? Why? Because this small molecule here that is a nasodiide, has a double bond between these two nitrogens. This is simply, my friends, ladies and gentlemen, the most abundant food colorant in the planet Earth, okay? So this is a molecule that's used in food, it's used in medicine, it's used in cosmetics. In fact, many of you have used a lipstick this morning that has red on it. So this is very abundant, super abundant. Just an example, okay? 30 to 40% of all food sold in the United States of America to young kids have this compound, okay? This is no joke. This, you know, Fanta has this stuff. You know, Kool-Aid has this stuff. You know, M&M's has this stuff. Everything is. Everything. Okay? So, however, it's approved by the FDA for use. You know, there have not been reports that this thing is too bad, except that in some Scandinavian countries, people have started to notice that this may be associated with attention deficit hyperactivity disorder. But you know, so far, so good. And you know, in 1980, the amount of this stuff that was produced was gigantic. Today, imagine that's even higher, much higher. But this is just a hypothesis, right? So, I mean, let's try to prove that this is the real thing. You treat animals with tamoxifen, induce IL-23 in the gut, nothing happens. Nothing happens. However, if you do tamoxifen, if you can even inject it, you don't need to put in the food. But you give red 40 3 times in the water, this is what happens, my friend. The gut becomes completely ulcerated. This is red 40 alone, okay? Red 40 and IL-23, of course. And, you know, just to prove the point that, you know, this stuff is everywhere, we got Kool-Aid, we got Pedialyte, and the concentrations of red 40 in these two things here is actually lower than what we use in our mice. And you do red 40, you get disease. You do Kool-Aid, you get disease. You do Pedialyte, you get disease. So it's very, very striking, okay? So, and the other thing that's quite interesting is that for the disease to happen whenever you express IL-23 and you give the mice red 40, you need to have something, okay? That's the part of, it takes more than 2 to tango. which is this. You take cells from animals in remission, our transgenic mice that we keep giving them tamoxifen. At some point, I showed you that the disease stops. There's resolution. So we got the cells from the mesenteric lymph nodes from these mice, CD4 T cells, and we transferred the cells to rag mice, and we gave these mice RED40 2 cycles. And predictably enough, the mice develop disease. So this is showing that disease is caused by the CD4 T cells. And in fact, we have done a bunch of other studies showing that T cells, the CD4 T cells are the effector cells, very important. And we know the CD4 cells are the pathogenic cells here. But if you do the same experiment in rag germ-free mice, you see that there is no disease. So this suggests that the microbiota is important in the process. How important? How could the microbiota be contributing here? So what we think happens is this. We tested a bunch of other compounds, right? We tested a bunch of other things, yellow 6, red 4, yellow 5. All of this stuff is in your food chain too. Believe me, all of us are taking this stuff all the time. There are other food colorants that they're non-azo dyes and they don't cause disease. So the colitis is induced by this. This is not capable of inducing colitis. And what we think, and in fact, I think we have actually shown that very clearly, is that this azo bond that's present in these azo dyes here is very, very important. If you make a compound that is partially reduced and that cannot be acted upon by the regular azoreductases that exist in our commensal bacteria, this compound cannot be broken down. As a result, the mice don't get sick. So what this suggests is that what the bacteria are doing are breaking down this compound, and that breaking down this compound, they generate potentially highly nitrogen reactive sort of compounds that can bind either to bacterial antigens or to self-antigens. We don't know at this point what it is. But we think that the binding, that the processing of the dye by the bacteria is very important. Oh, again, something very interesting. Al23, bacteria, and a compound that's present abundantly in the environment. So that's what their story was, and this is the experiments that prove that if you get E. fecalis, for instance, or you can get an anaerobe too. This is an anaerobe, and we got Bacteroides ovatus as an anaerobe. If you cultivate that, we put RED4 into these things, they break down, the color disappear. That means that the bacteria getting rid of the double bond here. And if you get this material here and you now feed animals that have been primed, they develop disease too. So the processing yields biologically active, reactive material that can cause disease. Okay, so that's the bottom line here. And the story was all developing this way until, we go to meetings, you talk to your friends, and, the question becomes, if you all take this stuff, how come we're not all sick? Well, the first thing I pointed to them is that mice never take M&Ms. That's one thing. That's very obvious. They don't take M&Ms. We just introduced M&M in their lives. And we thought that perhaps the easiest answer for that is that once we are first exposed to RED40 or any of the other food colorants, if we are normal, if we don't get any changes in Al23 signaling or any other immune disturbances, you're going to be fine because we're going to learn to tolerize it. So we did an experiment to show that. So what we did was we treated mice first with this, and then we did the whole L23 gig here. And what we saw was something very exciting, is that, in fact, if you did that, you would tolerize. If you don't tolerize, everything goes according to the plan. But if you give the mice this thing here, you pretty much prevent development of disease induced by L23.
Sergio, you have 1 more minute.
Oh, okay. So I will try to rush. So we generate Tregs. Yes, we do. We did experiments that prove that, in fact, we generate Tregs that's specific for RED40. And essentially, this is to show you, I'm sorry, I'm going over time. And then the point is this. If you combine effector cells with regulatory cells taken from a mouse treated with water or with RED40, the ones that are treated with regulatory T cells that are exposed to RED40 are able to prevent disease. The ones treated with water don't. Okay, the other thing that's important is that you deplete regulatory cells from a mouse and give the mice RED40. You don't need L23. You get disease. And very, very, very clearly you get disease. So this suggests that one of the things ALTO-23 may be doing is it may be affecting regulatory T cell function. We don't know how exactly it does. That's our hypothesis at this point, but that's a possibility. So to make things, to summarize it, we have tested these 2 food colorants here. They're broken down by the bacteria in the gut, and they produce these metabolites that can potentially, we imagine, we don't know that for sure, under normal circumstances lead to development of tolerance. In the presence of ALTO-23, this does not happen very effectively, and we end up with an effector function in which we generate a very specific class of CD4 T cells that have cytotoxic properties. We have published this already. And these cells also produce interferon gamma. And you can block disease by blocking interferon gamma. So this is actually quite exciting. And this is a summary slide to say that perhaps what's happening in real life is in terms of these mice, and I would like to stress mice, is that the changes in ALTO-23 expression shift the immune balance in favor of development of responses to antigen modified by Red40, and that this may be caused by a disruption of ALTO-23 to the tolerance mechanism. We're testing this hypothesis now by deleting ALTO-23 receptor in FOXP3 positive cells, provided to us by our colleague, Dr. Vijay Kuchu, who is not here right now, but I would like to thank him and to show that to him that we also have our lions. And these are our lions. He has his lions. We have our lions too, just to compete with them. If you want to know more about it, you just go to this and you figure out who they are. But evidently, the work was done by this absolutely amazing postdoc, Lily Chen, and that's her husband, who's also an enormously gifted scientist. This is my wife and my greatest collaborator, and this is Mr. Red40. And actually, I told him, that I was going to come here and I'm going to present his story to the audience, and I told him the title of the talk, and at the end he got a little bit confused, and he looked at me and said, "But what is tango?" So that's it. Thank you so much.
So we can take a couple of questions. Okay, Dr. Sanjeev.
Wonderful lecture with your one-liners, making everyone laugh. That's the beauty of this lecture, enhancing the acceptance and imbibing, the information. I am a psoriasis patient. Okay, I just want to understand. You showed your data about IBS. Is this food-related issues also observed in your psoriasis data?
No, we have actually tried our best to see if we could get something like psoriasis in the skin. ALTO-23 by itself can do it. We don't need any modifier like this Red40 to induce that.
Yeah, but most of the people feel that psoriasis is a skin disease. Well, it's just a manifestation on the skin. Everything else is inside, right?
Well, yeah, actually, we have done other studies where we express ALTO-23 specifically in the skin. And what's really interesting, since you studied this, is that we get disease in the skin, and we get also over time development of psoriatic arthritis without circulating levels of ALTO-23 using the methods that we have at hand to detect ALTO-23. Right. So the diseases happen in the skin. Something also is going to be contributing to development of the joint disease.
Yeah, so I'm just giving an idea. If I take a red wine glass along with fish with this color, it gives pains to my knees. When I just take red wine, it doesn't.
But, this is what I'm saying. Look, this is mice, okay? First of all, mice. Second of all, I would love to study this in humans. And in fact, I proposed this to the NIH, and they said, there's no evidence that this stuff is important in humans. Of course, there is no evidence. Nobody studied it. So how can it be? But then that's the story. Yes.
Professor Lira, there's an excellent way of presentation of your experiment. Have you experimented with calcium where there is a downregulation of the IL-23? Because the previous question was the skin surface where vitamin D get expressed. So any causative thing of vitamin D with IBD?
We have not studied the role of vitamin D in this setting. We have not. As you can imagine, there are many different things that one can explore in this. And this is evidently with a lab with 3 people, we cannot really go after that, unfortunately. Thank you.
So, okay, I think there's no more questions. And you've clearly gained more fans now here. So with that, I close the discussion. And I think we can go for the lunch. Oh, there's a next session. Okay.
We are apparently supposed to take a picture together. But I don't know.
Okay.
Yeah. Thank you, ma'am. Thank you, sir. Now we move on to our next plenary lecture 15 by Professor Hamsa Putalakat, Department of Biochemistry and Chemistry, La Trobe University, Melbourne. I request Dr. Jyoti Kore, ACTRAC, and Dr. Jagdish Mittur, iBiome Bengaluru, to now take over and guide us through the session.
Good afternoon. While Hamsa is getting ready with his talk, I just want to say a few words about this talk. Thanks to the previous speaker, put everybody in good humor. Thank you so much. 49 million cases around the world, and 11 million people approximately dead, plus or minus. And the single causative agent is sepsis. And today we're going to hear something about sepsis from Dr. Hamsa Putalakat. Hamsa originally is from this part of India. He was born in Kerala and then moved to Bangalore to do his MSc in agriculture, and then moved to Baroda, and then moved abroad to Australia. He worked as a postdoc in VHI, Walter Eliza Institute for Medical Research, Melbourne, Australia. And he's been in Australia for about 30 years, and nice to have him back here in Bangalore. I met him for the very first time in Melbourne about 30 years ago, and he's really progressed from strength to strength. Currently, Dr. Hamsa Putalakat is the head of the Translational Research Lab of the Department of Biochemistry and Chemistry, La Trobe University, Melbourne. His research focus has been, for quite some time, understanding the molecular mechanisms of apoptosis for nearly more than 25 years, where he made several seminal discoveries. His research now is focused on developing a therapy for polymicrobial sepsis and in developing a hemodynamically neutral drug for specifically for systolic heart failure. As you all know, it is the case of a highly overzealous immune system, a bit more like overzealous firefighters. Just for a small fire, the fire brigade blasted the whole place with excess water chemicals, damaging the whole place. That's pretty much what sepsis is about within your body. His lab has, excitingly, his lab has identified a master regulator of inflammation and immune cell death in polymicrobial sepsis in mice and its homologs in humans. And one exciting news is that via the concept of open philanthropy, Dr. Putalakat Lab at La Trobe in Melbourne has received a substantial donation of 1.4 million Australian dollars. With this funding, he's really hoping he and his team will put more efforts towards developing an antibody-based treatment for polymicrobial sepsis, a very complex and life-threatening condition affecting many, many people worldwide. I must also mention, referring to the panel discussion last evening, Hamsa has been very much involved in grassroots level dissemination to the public as secretary of the Victorian branch of ANZAS, which is Australian New Zealand Association for Advancement of Science, and also through the contributing to the university's outreach program. And as I know him personally, he's a big fan of soccer, motorbikes, and does a great barbecue when you go to his house. Thank you, Hamsa.
Thank you, Jag. That was a lot of introduction. Sorry to keep you waiting. It's lunchtime almost. You must be hungry. And Sergio, you did a wonderful job, and it's going to be a hard act to follow. Okay, I'll try my best. Thanks to Dipankar for organizing this. So my lab, like Jag alluded to, we work on two important research areas. One is developing a hemodynamically neutral therapy for systolic heart failure, and the rest of the time we're trying to save the world. That is, trying to save the world from the scourge of sepsis. So sepsis is an intractable and insidious disease. It kills about, that is a conservative estimate of 11 million people worldwide every year. That means a equivalent of Spanish flu every 5 years. And if you look at the research funding, there's nothing much in terms of research funding. So if you compare with, this is an old data from in the United States from 2008. So every 100,000 population, you get about 338 cases, 31 cases of cancer, and you get funding of $2.3 million. Whereas sepsis, it's even more prevalent, but you get only $91 million. And this gap has widened further. And the reason for that is, reasons are manifold. One of them is awareness. It is, we don't know much about sepsis. Awareness is a big part of the problem. It is out of sight, out of mind kind of disease. For example, this is what I keep asking people in, a few slides are missing there. It doesn't matter. So especially in the context, in Indian context, with the emergence of antibiotic-resistant bacteria, if you end up in ICU, there is a 50% chance of getting sepsis. That is 1 in 2. That is quite scary. So the reason for lack of awareness, that's one reason why we don't get funding. The other reason is actually sepsis researchers themselves, which I'll come to in a bit more in detail later. For many years, we have been using wrong animal models, which seldom translate into human context, and people are sick and tired of this. So nobody wants to fund, nobody wants to touch this with a 10-foot pole. So that is another reason why we don't get much funding. So what is sepsis? Sepsis is actually a body's overreaction to a systemic infection. So you can get a cut while working in the garden, playing in the playground, or shaving yourself, or even urinary tract infection, particularly in women, sore throat, or even teenagers squeezing a pimple. So bacteria from this site can enter your bloodstream. When that happens, our body overreacts. So it is a body's overreaction to an infectious systemic infection. So first stage, what will happen is your body will produce copious amounts of cytokine. So that's where we end up being, it's a cytokine storm. So at this stage, during this stage, you have vasodilation, your blood pressure drops. So that leads to hypoperfusion of your vital organs, including your liver, kidney, brain, etc. So you can get multiple organ failure. But this can be mitigated in ICUs by various means, such as fluid resuscitation, adrenaline injection, steroid therapy, antibiotics. All that can, to a certain extent, mitigate this risk. And you get only up to 20% mortality. After this, what happens is you enter a protracted immune suppressive phase where your immune system crashes. You don't have a functional immune system to defend yourself against a pathogen or any opportunistic pathogen. So once you're out of ICU, you enter the community and get community-acquired pneumonia, you get nosocomial infections, and 80% of deaths happen during this phase. But the problem is all the clinical trials and attempts have been made to control inflammation. But we know, immunologists, you know, that controlling inflammation is a risky prospect because you need inflammation to control infection. So if you block inflammation, there's nothing to inform your immune system that there is an infection. So that's where all the clinical trials failed. So this is the last one that was taken out of market. This is Exigris by Eli Lilly. This was withdrawn from market because it was ineffective. In fact, there have been more than 100 clinical trials in the last 30 years. Each one of them failed. All of them were targeting the inflammatory phase. In fact, people say that sepsis killed more pharma companies than people. So the standard treatment for sepsis these days is steroids, corticosteroids. It started in the 70s. As of now, even now, that's the first line of treatment. And this was a study published by the Georgia Institute of Global Health saying that steroid is the final answer for sepsis. Its science is settled. However, it doesn't reduce mortality, but it reduces the time that a patient spends in ICUs. So what they're saying is, I have underlined it here, that it reduces the time spent in the intensive care unit, that not only frees up space for other patients, it saves health system a lot of money. So we are measuring the health outcome in terms of how much money saved, not in terms of how many lives are saved. So that is the stage of sepsis research. So we decided to take up this challenge, not because we wanted to study inflammation. Like Jag said, I did my postdoctoral study at the Walter and the Eliza Hall Institute, where all the seminal and pioneering work on apoptosis was done. So I thought I had a better understanding of apoptosis. I'll try to understand the apoptosis mechanism of immune cells, how it happens. So we had few things working in our favor. We developed and optimized this method of cecal slurry-induced sepsis, peritonitis. The standard method for gold standard is cecal ligation puncture, which is very time-involving and invasive. And if you're doing a high-throughput screening, whole genome screening, you can't subject mice to multiple rounds of sepsis using CLP. So whereas this one, you can titrate the dose such that you can make it mild so that mice survive multiple rounds of infection. So that's one thing. We also developed this two-hit model where you induce sepsis. When they recover, you can challenge them with secondary infection agents such as Pseudomonas intranasally or Candida intravenously. So we developed these two models for testing our hypothesis. Then we had access to CRISPR technology, and also we had CRISPR barcoded library that had about 100,000 different guides covering all 25,000 protein coding messenger RNA in the mouse genome. That is 4 guides per gene. So what we did was we took hematopoietic stem cells, mouse hematopoietic stem cells, we reconstituted mice, and we subjected these mice to peritonitis 3 times. And whatever remaining cells we found, we fact sorted and did next-gen sequencing. So I'll explain this in a bit more detail. You take mice, you radiate them, myeloablate them, then you take fetal liver cells, hematopoietic stem cells, introduce lentiviral particles containing the guides, and these lentiviral particles have GF or BFP, that is blue fluorescent protein, as a surrogate marker. So you can introduce that into these mice. That is reconstituted mice. You induce peritonitis, sublethal sepsis in these mice. The idea being, when induced sepsis, immune cells die by apoptosis, except for those cells where a particular one gene may be modified in some way by CRISPR. Those immune cells will survive. And when you subject these mice to multiple rounds of sepsis, then you get a clonal selection, and these cells, the cells that have undergone CRISPR editing will be the predominant population. So we can fact sort this based on the surrogate marker blue fluorescent protein, and we take those cells and do a next-gen sequencing. What we found was a gene called TREML4, that is trigger receptor expressed in myeloid-like. That is coming up about 10 to the power of 5-fold over the background. And that's a receptor with the extracellular immunoglobulin domain and a cytoplasmic domain. So the next obvious thing to do was to make a knockout, which we did. And first thing we noticed was thymic apoptosis. That is, you can see in wild type during sepsis, thymic apoptosis happen, whereas this is totally reduced in the knockout. In fact, if you look at the thymic, that is wild type undergoing sepsis and knockout. So there is no thymic involution in the knockout. So this is reminiscent of what happens in neonatal sepsis. When newborn children get sepsis, thymic atrophy happens and they live with the consequence for the rest of their lives. So we knew that we were on the right track. We subject these mice to acute lethal sepsis. They survive compared to wild type. And we looked at, it's a busy slide, just look here, these two things. This is granulocytes. In the bone marrow, you can see more granulocytes in the knockout compared to wild type. And then we looked at neutrophils, because neutrophils account for about 70% of our white blood cells. And what we found was in the knockout, you can see mature neutrophils compared to wild type. There is hardly any in the bone marrow. And in the blood, you can see a long trail of immature neutrophils in the wild type, whereas what you find in the knockout is all mature neutrophils. That is during acute phase of sepsis. And then we did same experiment in a 2-hit model where you induce sepsis in these mice, sublethal sepsis. When they recover, you challenge them with intranasal Pseudomonas and look for survival. As you can see here, wild-type mice that undergone sepsis, they died during the secondary infection challenge, whereas knockout survived. And if you remove neutrophils by Ly60, anti-Ly60 antibody, you can see the protection in TML4 is gone. So we know that this is all dependent on neutrophil. It's all to do with the neutrophils. We tested other models as well, including influenza. We couldn't find any difference. These mice are equally susceptible to influenza PR8 compared to the wild type. And during chronic sepsis, again, you can see mature neutrophils in the bone marrow, much more predominant or quite increased amount of functional neutrophils existed in knockout compared to wild type. And that is true in the blood and even bronchial alveolar lavage bile neutrophils from the lung. You can see there is more immature neutrophils in the wild type compared to knockout. And these neutrophils from the lungs can phagocytose Pseudomonas 3 times more efficiently than wild type. And these neutrophils produce less myeloperoxidase compared to wild type. And that leads to less inflammatory damage to the lungs. You can see what you can see here is tunnel staining of wild type, which is absent, and there is less inflammation, and all the infections clear in the knockout compared to wild type. So the conclusion from this part is TML4 is responsible for neutrophil homeostasis during acute and chronic polymicrobial sepsis and Pseudomonas infection. So the next part rest of the time, I want to tell you about is inflammation. So quite often when they study inflammation in the context of sepsis, the agent of choice is LPS. Many mouse models have been, people have published a lot of work using LPS. And this was the basis of ISI developing this drug called eritaran, which is an LPS analog. It binds to TLR4, doesn't do anything. So when they induced sepsis in mice using LPS and administered eritaran, it protected. When they took it to the clinic, it was an absolute disaster. And use of LPS is the reason why people think that what you find in mice, you can't translate into humans, sepsis research. And this was a paper published in 2013 from a group in Stanford saying that what you find in mice cannot be reproduced in humans, which was countered by another group saying that they cherry-picked this data. And Richard Hotchkiss, a guru in the field, favors these people. And this data, this result, emboldened this animal rights group called PETA, that is People for Ethical Treatment of Animals, to lobby NIH. And they have a policy now not to fund. They prioritize human-based research for sepsis, not mouse-based research. Also, recently, there was a paper in Nature, this was in July, saying that you should not use mouse model for sepsis research. That's putting us in a very difficult situation based on flawed mouse models. Another thing to note is, if you look at the JAXLab data of website, they have this mouse strain called C3HEJ, which has TLR4 mutation, meaning they don't respond to endotoxin. You can inject them with the endotoxin, they are fine. But these mice are more susceptible to gram-negative bacterium. They die. So there is absolutely no protection. So the bottom line is LPS-induced sepsis model is totally, totally flawed. It has done more disservice to sepsis research than anything else. Not only in mice, even humans, there is a report saying that TLR4 mutation doesn't have anything to do with sepsis onset in humans. So in any case, we looked at TREML4, involvement of TREML4 in inflammation. As you can see here, during polymicrobial sepsis, inflammatory markers are downregulated. It's not completely gone, but there is some inflammation, but substantially less in the serum as well as at cellular level in macrophages and neutrophils. And we did a RNA sequencing study of bone marrow-derived macrophages in cell culture, and we couldn't find any difference between wild type and the knockout in response to LPS activation. And that is the same result by ELISA. However, if you inject LPS in these knockout mice, you can see there is downregulation of some cytokines, not all of them, suggesting that TREML4 acts as an amplification loop for some of cytokines, not all of them. So the conclusion from this part is that LPS endotoxin model is a poor model for studying polymicrobial sepsis, and bone marrow-derived macrophages in vitro suggest that TREML4 and TLR4 do not cooperate in eliciting inflammatory response. And in vivo LPS studies in TREML4 mice suggest that TREML4 is an amplifying mechanism for TLR4. We also worked out the signal transduction mechanism in these mice during sepsis. As you can see here, there is reduced ER stress, and as a result, apoptotic genes downregulated in TREML4 knockout mice during sepsis. And we looked at calcium flux, and this is by Indo-1AM staining in cells. You can see TREML4 doesn't induce much calcium. That is corroborated by mass spectrometric analysis of neutrophils, where all the calcium-regulated proteins are subdued or way downregulated compared to wild type. And based on this, we looked at all the pathways downstream of DAP12, which is an adapter for TREML4, and they are all subdued in TREML4 compared to wild type. So based on that, this is a pathway, this is the model we suggest that DAP12 interaction with TREML4 leads to PI3, IP3 production, which leads to calcium flux, which induces inflammatory cytokines. At the same time, it induces apoptosis of immune cells. So the next model we studied was a Candida model, where we did the same thing. Candida infection model, a secondary infection. As you can see here, mice survive TREML4, nice survival, only if it has seen cecal story before or sepsis. Only in the context of sepsis, TREML4 knockout mice are protected, not naive TREML4 knockout. And the reason for this is we are working it out. It is to do with epigenetics. It regulates certain genes involved in inflammatory pathway, which we are about to work it out. We have done ChIP-seq and ATAC-seq analysis, and it is waiting to be published. And this is the candida in the kidney, as you can see here, TREM4 knockout, which has seen sickle or sepsis. They hardly have any candida load in the kidney, or that is corroborated by immunohistochemical staining of candida in the kidney. And this is a survival. This memory lasts up to 30 days. I'll just skip through that. Again, this depends on neutrophils. You can remove this protection if you remove neutrophils by Ly60 antibody treatment. So far, so good. That's all about mice. What about humans? So this is a family of genes present in chromosome 17 in mice, and there are homologs in humans as well. So we identified through genome mining, database mining, we identified 2 homologs, TREM4 and TREM2. And we made knock-in mouse strains for both of them where TREM4, human TREM4 or TREM2 expressed from a mouse TREM4 locus, so-called humanized mice. And these mice behave like wild type. They can both, TREM4, human TREM4 and TREM2 can recapitulate what mouse TREM4 does. So here is TREM4 knockout, they survive, but if you introduce either TREM4 or TREM2, they behave like wild type. In terms of inflammatory cytokines, same story. So TREM4, there is subdued cytokine, but it can be restored by introduction of TREM4 or TREM2, human TREM4 or TREM2. So now what we're doing is, so we identified a molecule that can regulate both phases of sepsis, that is inflammation, as well as immune cell death. So the next thing is to identify monoclonal antibody, a blocking antibody for which we are trying now using the Beacon platform. We already we'll get all the results by the end of the month. So we are hoping that we will have a blocking antibody in the not so distant future. With that, I'll stop, though I bagged all these funding agencies. I have been fortunate enough to get enough money for my work from the National Health and Research Council, as well as from Open Philanthropy. Particular thanks to Professor Chris Somerville, who was instrumental in getting me this money. And this is my group. And I think in time, so there's enough time for you to ask questions.
Thank you, Dr. Hamza, for this real, I think, deep insights into this polymicrobial sepsis. I think a little bit different. So I can see some we can ask some questions.
Yeah, very nice talk. Couple of questions. You said initially that sepsis is more like a hyperactivation of the immune response. I always thought that sepsis is more like a dysregulation of immune response. So somehow you can't control it. And that's why, so are there molecules known which kind of work as a brake, like people talk about T cells? Here also the brakes are gone?
It's both. It's hyperinflammation and hypoinflammation. That graph I showed is too simplistic. It can, a hypoinflammation can start early on, right? In terms of T cells, I don't think, I didn't present the data. You can see a phenotype in the thymus, but not in the periphery. And Richard Hotchkiss, the guru in the sepsis field, he used to be a good friend of mine. He spent his lifetime trying to identify T cell role of T cells in sepsis, and he couldn't find any. That's because sepsis is a fast progressing disease. Within few hours, the host has bolted. And you don't have time for that active immune system to kick in. It is to do with the innate immune system. So I don't think T cells have any role in
actually, I didn't mean that. I was saying that somehow your brakes in the innate immune system are gone, and that's why you are producing a lot more than what you should be doing. Like
initially, it is a lot of cytokines produced by the innate immune system. That's right. Then during sepsis, they die. So it goes the other way around, totally flips around. So there is hardly any cells there to produce cytokines or to defend yourself.
Yeah, just one last question from me. That what neutrophil data you have, you think that is a migration or you are generating more neutrophils?
Both.
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 Okay.
Yeah, if we consider the hospital setup where sepsis is there, is really a very dangerous condition, but it's managed by antibiotics and it's phenomenally by gram-negative organisms. So, immune system probably comes very late in picture because, as you said, and it's being a polymicrobial in nature, it's very difficult for immune system to get and help somewhere. That's my feeling.
Yeah, so antibiotics helps, but quite often it's the antibiotic resistant bacteria that can cause much more harm. Yeah, was it a question or was it a statement? What you made?
Polymicrobial condition, there are hundreds of antigens.
Oh, definitely.
But many of them, are controlled. Means those bacteria are controlled by, treatment.
Controlled by treatment, but there can be bacterial antigens present in the system. Controlling bacteria means they may be dead, but there can be a fair bit of bacteria. I mean, that is what we see in the clinic. Antibiotics really does not do much.
But all these antigens are very different. How immune system would sort it out?
I don't know answer to that. Sorry, I don't know. I know what you're saying. There are different.
Okay, we have more questions here. There's one here.
Okay.
Please, and then one there. There are two more there. Yeah.
This was a great talk and burning issue that you are addressing in sepsis. So I have two things to ask. So what's your view on the anti-TNF therapy that few people have tried for treatment of sepsis?
Yeah, anti-TNF Humira was tried to treat sepsis. Anti-TNF is just one of the many cytokines. So controlling TNF wouldn't help in sepsis. That is quite simple.
And I would like just to connect with that on the mid-career talk that Professor Baik gave. She just gave a talk on the TLR4, blocking of the TLR4 with this novel thing. So how would you think that would work on this?
TLR4 blocking. Yeah. So I think I made it abundantly clear. TLR4 blockage wouldn't help in bacteria. So when you use TLR4, you're injecting endotoxin in the absence of any bacterium, right? In real life, when you get an infection, it's not just LPS, there are bacteria present in the system. So in mice with TLR4 mutation, non-functional TLR4, if you inject them with endotoxin, they survive, but you infect them with forget about gram-positive, gram-negative bacteria, they die. It doesn't offer any protection at all. So my opinion is that TLR4-based, LPS-based, TLR4-based drugs will not work. You can mark my words on that.
All right, three more questions. One there, and one behind, and one last one here.
Yeah, over here. So over here.
Yeah.
So very interesting talk. I just had two questions. One is, what about the TREM expression in the other innate immune cells? You did show convincing data for the neutrophils, but what about the other innate immune cells, the expression of this receptor?
Yeah, it is expressed in macrophages and dendritic cells.
Okay.
Definitely.
And in terms of the contribution to sepsis, like how important is also in the disease and macrophages?
Disease and macrophages. Well, I can tell you that if you take out neutrophils by Ly6T mediated, complementary mediated depletion, you can take away the protection. So it is totally dependent. I mean, maybe they're playing a minor role, but it is mostly neutrophils, which accounts for about 70% of your white blood cells.
Okay. All right, just one last question, please, before we close it. It's getting late. The young man up there. That's the last question.
Hi, Professor Hamza. It's a nice, this way.
Oh, okay.
So it's a nice presentation, informative talk. So I have one question in fact, I just want one statement from your side. You mentioned the humanized animals. Why you call them humanized?
It's not, I mean, okay. That's
They are
too
not actually humanized.
No, it's quite too simplistic. Yes, I agree with you.
Because as far as my understanding goes, that you need to replace the mouse system with a human one.
No. Listen, there are different kinds of humanized mice. So I take it back. It's just for people to understand. Okay, we replaced mouse TREML4 with human TREML4.
Okay.
Doesn't mean they're totally humanized.
Exactly.
Yeah.
Yeah.
Agree that. Yeah.
Thanks.
So, okay, I think it actually ignited a lot of questions here and because of the good talk. So I would like to thank my co-chair and I would also like to thank organizers to having us here. And thank you, Dr. Hamza, for a wonderful talk.
Thank you.
Thank you, Professor. I request the chairs and the speaker to assemble for a group photo. Yeah. Before we break for lunch, we have 10-minute technical presentation by Abcam.
How do I connect this?
Senior scientist, Dr. Shen.
Hello. Yeah, thank you. So good afternoon, everyone. I'm the only one who is stopping your lunch time in between, but I'll quickly move forward. So I'm Abhishek Chauhan. I'm the commercial manager for Central Asia, and I'm based in Delhi. And we are representing Abcam here, which is a UK-based company based out of Cambridge. We were established in 1989. We have about 1,800 employees and probably serving more than 150 countries. Almost 50% of the global research papers using antibodies will have Abcam citation in that, and that is where our products are very important. More than 25 years of expertise. We are into a lot of research and diagnostic and therapeutic purposes. Moving forward, we are known as an antibody company, but we are a company which have multiple range of products. We do have about 70,000-80,000 of antibodies. Plus, we have a lot of ELISA kits, and we have modified our ELISA kits into a 90-minute ELISA as well, which you'll come to know in the later slides. We have got specialized products for conjugation, which will help you to conjugate your products. We have specialized bioreactive proteins. We have knockout cell lines, and even for CRISPR. We are into cellular and biochemical assay. We bought over a company called BioVision, and they are specialized in that. We have conjugation-ready antibody formats, and that is why we say we are not only a provider of antibodies. You see here, we have CRISPR knockout portfolio, which have got about 5,000 cell lines ready in-house, ready for suppliers. We have iPSC-derived cell models. We have research-grade proteins. We have bioactive proteins, and we have very premium bioactive grade proteins, which are 95% pure and could be used for cell culture, stem cell differentiation, and anything into cell programming. We have conjugation gates which require only 30 seconds of hands-on time. It is just a stopover into a particular vial, and that takes your conjugation done. It works with 10 micrograms to 100 mg of antibody, and it gives you a very consistent and very precise conjugation. I was mentioning about 90-minute ELISA, and this is how it works. We get pre-coated plate we reduce the time from 4 hours to just 90 minutes. And you can see the process. You are much well aware of it. And this helps you to do more of ELISA in a much more faster speed. There are a lot of requests about smaller pack sizes and sample pack sizes. We do have antibody panels, and these panels will help you. These are pathway-specific panels. They help you to identify the panels of antibodies into a particular set. We give you that. You can try all of them, and probably anything which works better for you could be a use for you to procure in a larger quantity, and that is why we have antibody panels. The whole range of products from our side, we do basic research and study. We help you with target idea and identification. We help you with preclinical development and clinical trials and all the ranges. But that is all about products. I don't want to get too much of detail about the products, but the important thing which belongs here is that why do you need to choose AppChem? And that is where we are here for today's day. So this is one of the paper from Nature, and which says actually about 70% of the scientists, which you refer to papers and you do a little bit of research by looking around. So while you look around, about 70% of the scientists have failed to reproduce the experiments, the results which other people have done. About 50% have failed to reproduce the experiment of the same design. And of course, there was US data, very specific, about 28 billion each year on biomedical research, and it cannot be repeated successfully. That means there were a lot of money which is getting wasted in research, and we are not able to even reproduce the results again, time and again. And that is why we need to choose a right reagent supplier, because we need the results on the first time and every time whenever we use those particular products. And that is why validation of those products are very, very important. And how do we do that? So there's a company called FightAB, which do the analysis of the companies who are into antibody structure. And that is where the specialization, the validation, the quality of the products come in. From the last 4 or 5 years, AppChem have been a consistent performer out there, and we have been getting all their awards. Of course, because we have a validated antibody, we have suitable application of species-related antibodies, and we have polyclonals, we have monoclonals and recombinant antibodies, which are highly specified, highly reproducible, highly specific about the products. And if you use a particular product, you will save a lot of time on your experiments. Our products are already validated, so if you need any data, if you need any scientific information, that is already available on the website. And with us also, if you can always reach out to us. We have done knockout validation for multiple cell lines. We have a lot of technical team at the back end which helps you to identify the products which you need to buy. We have everything on the resource for your publications are also on our website. So when anybody uses a particular product, they can go into the details about who have used what products and how the results are. Those things also help us to build a relationship with our scientific community, which is you. And that is where we grow ourselves as together. And of course, we have consistent products. I think AppChem is known for consistency and reproducibility. So whenever you do your experiments, you keep on working on the experiments and probably getting similar kind of consistent results year over year. And that is where AppChem's specialization comes in. We also offer 12 months warranty for every product which you buy, which is called a Web Promise Guarantee. So any product which you are buying, you don't even need to sample it out. It's just you buy it. It should work for you, first of all, 99%. If it doesn't by any chance, then you can always come to us and we'll try to help you with all the solutions in technicalities, as well as even if it doesn't work after that, we can always get it replaced. This is a 12-month warranty. We also run a app trial program, which is actually a program which helps you to identify and probably try untested applications. It could be species specific, it could be any other application as well. So this is something which we need to get in touch with the AppChem representative all around. If you're using a particular antibody against, for example, anti-rabbit, and you are probably doing any experiments for anti-mouse, probably you can do an app trial, test it out. Even if it works or if it doesn't work, both the situations, AppChem is ready to help you with the particular fund which we have. So this is very, very special. None of the other companies do it, and we do it just because we want to support the science which you are doing. And of course, as I mentioned, that we keep on getting a lot of awards from the Fight AB company. We have been consistent performers. More than 50% of the papers already published on AppChem antibody only. More than 1.5 lakh products, more than 50 countries we are available. And here we are. I'm keeping it crisp and sweet. And for anything, please feel free to ask me anything. Thank you.
Thank you. Yeah. Thank you, AbCam. We will now proceed for lunch and poster session. We request everyone to attend the posters and also visit the company stalls. Participants who have been assigned today's slots for poster presentation are requested to put up their posters according to the serial number provided. We'll all reassemble at 3 o'clock for the next sessions. Thank you.
minutes, so please settle down.
Yeah, I did. It worked. I just checked. I checked everything. So
The way to upstairs is locked. How do you access the?
it's gone. It's there. So this will move the slides ahead. Yeah. And then this is the pointer. Okay. Thank you. You can have time.
Just change the buttons.
I brought it now.
Oh, it's
Yeah, I
okay,
brought
okay.
it now. It goes away on its own. Sleep over. Oh, sleep over.
Good afternoon. Welcome back, everyone. Hope you had a hearty lunch. We are honored to commence with a plenary talk by Professor Sheila Ramanathan from the University of Sherbrooke, Canada. I request Professor Shripat Patil Nimhans and Professor Uma Kanga Ames to now take over and guide us through the session. Since we are running a little behind the schedule, I would request the chairs and the speakers to adhere to time. Thank you.
Dr. Sheila Ramanathan, she has done her PhD in in Madurai Kamaraj University, and then she has moved to Canada, and she's a vice president of immunology over there in Canada, and she has done a lot of work on COVID. She's presently at Sherbrooke, Canada, and over to you, Sheila.
Thank you, Shripad and Deepankar for the invitation. And Deepankar had told me I have to cut down the time. Already I lost 15 minutes. So I'll give you a talk in 5 minutes if I can. So thank you very much for inviting me. So today I'm going to present some data on the functions of IL-15, and it will not involve NK cells. So before I start, there is something that I should absolutely mention. Dr. Professor V.R. Muthukarpan, he's my mentor and the mentor of very many people. So I'm proud and honored at the same time to say that I'm a product of MKU under the mentorship of Professor V.R. Muthukarpan, who was a great immunologist. So the obituary for him will be, of course, loaded in the Indian Immunology Society's website. So I'm not going to read anything about it. But some of the highlights are the fact that the first department of immunology was established in India in MKU, and we are some of the products. And Professor VRM is one of the persons who have trained very many immunologists who are there everywhere, and they are holding his mentorship to their heart. So with this, I will continue with my talk. So we all know that cytokines play a major role in various stages, but in the peripheral homeostasis, IL-7 is required for the maintenance of T lymphocytes. And in addition, IL-15 is required for the maintenance of the CD8 memory T cells. We know that 3 signals are required for the activation of naive T cells for them to develop into fully effector T cells and then memory T cells and whatever they are supposed to do. And we also heard recently that not all naive T cells are born equal. And I would like to add to it that the environment also does not deal them the same cards in an equal manner. We know that these naive T cells are constantly in an inflammatory milieu, either due to chronic inflammations or due to infections. And so they are exposed to these cytokines in a very, very regular basis. So cytokines like IL-6, 15, 21. I'm just mentioning the ones that are involved in T cell homeostasis. So we asked, in collaboration with Ilangumaran, we asked a simple question: what happens to the naive T cells if they see these cytokines first before they see their cognate antigen? So we used a simple model of P14 cells. We exposed them to the cytokine combinations, followed by the stimulation with either the agonistic peptide or weak agonists. And as you can see here, the effector activity of the cytokine prime cells was increased. This is the cytolytic assays. So then we wanted to know, how does it translate in terms of autoimmunity? So for that, we used the same model. We used the RIP-GP mice, which expresses the LCMV GP protein in the islets of pancreas, and the P14 mice, which is specific to the peptide from GP. So these mice do not develop spontaneous T1D. However, when they get infected with LCMV, they develop T1D because the activated T cells can now see the antigen expressed in the pancreas and they delete it. So we asked whether the cytokine priming can induce type 1 diabetes in this model. So the cells were either primed with cytokine alone or with the altered peptide ligands or combined. So cytokine priming followed by APL and then injected into RIP-GP mice. And what happens for type 1 diabetes? So as you can see here, cytokine priming followed by altered peptide ligands resulted in the induction of type 1 diabetes in these mice. So to cut the long story short, what these data show is that naive T cells can be, exposed to these cytokines and their antigen sensitivity increases, and on the positive side, it can help in anti-tumor immunity, and on the negative side, it can cause autoimmunity. So to address this in a better, to address the role of IL-15 specifically in a better model, we use the model of type 1 diabetes, spontaneous T1D in NOD mice. I don't have to introduce the model to all of you. We used IL-15 because the role of IL-21 had been already shown at the level of CD8 T cells. So we wanted to know about IL-15. So we generated mice that were deficient for IL-15, and we observed that the incidence of diabetes was reduced in the IL-15 deficient NOD mice. Similarly, when you inhibit the IL-15 signaling with an antibody to IL-15 receptor beta, which is shared with IL-2, the incidence of diabetes is also reduced. So IL-15 is a very special cytokine. I'm sorry, I'm just going a bit fast so that you have your evening free. So IL-15 is a special cytokine. It is produced in the producing cell. It associates with its private receptor, which is the IL-15 receptor beta, here, as it's shown here. And then it's trans-presented to cells that express the beta-gamma receptor. So this is called trans-presentation, and this is called cis-presentation when it acts on its cells. And over the past 2 decades, it has been shown that both IL-15 deficient mice and IL-15 receptor alpha deficient mice, they essentially phenocopy each other, which means that they all lack NK cells, memory CD8s, ILC, gamma delta, or whatever that you can think of that are dependent on IL-15. So, to our surprise, when we generated IL-15 receptor alpha deficient NOD mice, they had no phenotype, meaning they all developed diabetes as usual. Whereas it's the IL-15 deficient mice that was protected. And we were in a question because both these mice lack the same lymphoid subsets, but they behave differently, and of course, couldn't send it to the grand panels nor to the journals. So we asked what is happening. So to do that, we wanted to know whether the pathogenic role of IL-15 can extend beyond that of these lymphocyte, IL-15 lymphocytes dependent subsets. So to that, we generated tons of mice, different genotypes. I'm not even going to go into the details of it, but I'll just show you the important ones that provide some idea. So in NOD mice, you can take the splenocytes from the diabetic NOD mice and transfer it to a immunodeficient mice like NOD SCID, and they will develop type 1 diabetes. So next we transferred it to IL-15 deficient NOD SCID mice, and here we see that there is no type 1 diabetes. So even though the splenocytes transfer T1D here, they don't do it here. Again, surprisingly, these mice do develop T1D. So this already suggests that the IL-15 receptor alpha from the trans-presentation is really not required in this context. However, IL-15 signaling was required in the recipient because you see here NOD SCID gamma, which lacks the common gamma chain, so it cannot signal through IL-15 compared to and also other cytokines. So these mice do not develop diabetes in the same experiment. So these results suggested to us that IL-15 for polyclonal T cells in this model, IL-15 and not IL-15 receptor alpha mediated trans-presentation is required for the induction of diabetes. So we have shown these results in various other models. And just to summarize this whole part, so IL-15 deficiency protects NOD mice from T1D. Transient blockade of IL-15 signaling during the pre-insulitic stage can attenuate T1D. And activated diabetogenic T cells may or may not require trans-presented IL-15 to cause disease. And IL-15 in non-T cells is required for the pathogenic potential of these diabetogenic T cells. So how do we distinguish the functions of IL-15 transpresentation versus that's not dependent on transpresentation? So we resorted to multiple models so that we can address the question appropriately. So you're all familiar with the RIP-GP model developed by Pam Oshie, where I mentioned previously that the GP antigen is expressed in the pancreas. So when these mice are infected with LCMV, nothing happens in wild type mice. However, the mice that express the GP antigen, they of course clear the virus, but they also develop type 1 diabetes as a collateral damage due to the visibility of the T cells destroying the islets because of the RIP-GP antigen that's expressed there. So we asked what happens in the same thing in 15 deficient or receptor alpha deficient mice. So in IL-15 deficient mice, we saw that 30% of the mice develop diabetes, whereas in the receptor alpha deficient mice, again, LCMV infection induced type 1 diabetes in 100% of the mice. So by multiple models, we could show that. So the next question is, what happens to the viral clearance? Surprisingly, we found that in the IL-15 knockout mice, the viral clearance was not efficient, whereas in the receptor alpha knockout mice, the viral clearance was clean, meaning there were no virus because it's acute infection, not the chronic strain. So this created a question because IL-15 we know is required not for clearing just by LCMV, but various other bacterial, fungal, parasitic infections. Multiple papers are there out to show that. But we were the first one to show that the trans-presented IL-15 is not required for clearing LCMV infection, at least in this case. So we were wondering about what about other infections. So, and also whether the protective role again is through the dependent lymphocyte subset, because these mice lack NK cells, gamma, delta, name it, whatever you say is IL-15 dependent. And so we resorted to the low-dose Listeria infection model. So low dose is usually cleared within 3 to 5 days by the innate immune system, whereas high dose requires antibiotics. So in this, you have the classical activation of all the innate molecules that are involved in the bactericidal activity. So here also we found the same thing. As you see here, 15 knockout mice succumbed to low dose infection, whereas 15 receptor alpha knockout mice, they were fine. Gamma knockout is shown for a control. And again, bone marrow transfer showed that the IL-15 can come from the bone, from either the hematopoietic or the non-hematopoietic system. Sorry, I'm unable to explain the details of this for lack of time. So all these results showed, and one of the major cytokines that were absent was IFN gamma in the IL-15, infected IL-15 knockout mice. And as a consequence, of course, CXCL9, which is IFN gamma dependent chemokine. NK depletion had no effect in the wild type or receptor alpha knockout mice. And this was also repeated in another attenuated strain with Salmonella strain. So this means that IL-15 knockout and receptor alpha knockouts behave completely differently. So just the phenotype, the point is that the inflammatory monocytes were reduced in the spleen. The rest of them are just controls to show this point. And we carried out proteomic analysis of the infected spleen, and we found that the 3 genotypes, they segregate completely differently, indicating that whatever is happening in them are completely different. So to summarize this part, IL-15 deficiency compromises early innate immune response to infection. And this is important because this protection was usually conferred upon or attributed to NK cells or gamma delta cells or ILC1s, but they are absent in these mice. And we found that early interferon gamma was affected, and these were the results I showed you. So this raises the question, are IL-15 knockout mice, are they really immunocompromised, and what happens to cancer immunosurveillance? So we used a classical model of MCA-induced fibrosarcoma, where you paint the mice skin in a part with MCA, and it's a carcinogen, and over 8 months, some of the mice will develop fibrosarcoma. We were surprised to find that wild type and IL-15 knockout mice, they had comparable incidence of sarcoma, whereas the 15 receptor alpha knockout mice had decreased incidence of sarcoma. And so we carried out proteomics on the tumor tissue to understand the pathways that are differentially expressed, and we have observed about 34 proteins that are, this is at the protein level, 34 proteins that are differentially expressed, and these belong to the innate pathway. These are all not published results. So to summarize, in autoimmune diseases like T1D and RA, where IL-15 is implicated, probably these diseases, it's interferon gamma independent, and IL-15 acts on non-T cells. For example, synoviocytes, or in T1D can be the antigen presenting cells or pancreas themselves. In another model, others have shown that in psoriasis, receptor alpha dampens the immune responses. They also have observed similar results to what I showed for T1D. And then in cancer, we are studying what is the role of these differential signaling. And in infection, it seems to be interferon gamma dependent, of course, because infections need interferon gamma. And what are the other cells that are really IL-15 dependent but not dependent on transpresentation? So these are the questions that we are left with today. So I'd like to thank the students and postdocs who did the work over the years, and the funding agencies and our collaborators. Thank you for your time.
The paper is open for discussion. Dr. Sheila has been working for quite some time on IL-15 in Canada. So we can take 2 questions, please. Yeah, in the front.
Probably I went too fast for you to assimilate.
Nice presentation. So IL-15 receptor alpha, can it be a marker for immunotherapy in cancer? Like expression of it can be looked at, and
It is used, it's not really a marker for that, because many cells express IL-15 receptor alpha. It's not regulated by activation or the functional state of the immune cells. So it's expressed by most of the cells.
okay, like an overexpression in the tumor cells or something like that?
It has to be really subtle, so I doubt if it would translate to that.
Okay, thank you.
Any other question? Thank you, Sheila. If there are no more questions, thanks once again.
Thank you. Oh, sorry.
I would request the speakers and the chair to come up for a group picture. लगा दो. तो मैं नाम सुन. Wait. Thank you. Next, we welcome Professor Christian Ingwerda for our next plenary lecture. I request Dr. Rajiv Kumar from BHU to now take over and guide us through this session.
A very good afternoon to all of you. It's my pleasure to introduce Chris to all of you. I and Chris started working very long back. So we are working together for last 14 years. Chris is group leader in Queensland Institute of Medical Research, where he is group leader of Immunology and Infection Laboratory. He is also head of Infection and Inflammation Program at QIMR. Chris works on experimental and human malaria and leishmaniasis, and his works are mostly focused on understanding the CD4 T cell response. So with this, Chris, over to you. Floor is yours.
Thank you, Rajiv. And I just wanted to also thank Dipankar and his team for putting on a really great conference. It's been a real pleasure being here and hearing some of the great science that's going on. Today, what I want to do is tell you, first of all, about some of the work that my lab does, and then some of the key questions that we're interested in addressing, how we go about that, and then give you some rationale for the project that I'll tell you about today. So my group works on two protozoan parasitic diseases, malaria and leishmaniasis, both of which are transmitted by mosquito and sandfly vectors respectively. And we're really focused on understanding the host immune response to these parasitic infections. We have preclinical models for both malaria and leishmaniasis, but we also use clinical samples as well. And I guess the model we've been working by in the past few years is really trying to use the clinical samples for really discovery projects, then go back to the preclinical models to test specific pathways or test hypotheses we generate from that discovery research. Our principal focus in the lab is CD4 T cells, and I won't go through this slide because we've had plenty of this during the meeting, except to point out a few of the features that are salient to the talk today. With malaria and leishmaniasis, the main CD4 T cell subsets that we're interested in are these Th1 cells, which produce pro-inflammatory cytokines, which in turn activate phagocytes. The leishmania parasites we work on, they reside within parasites. Malaria red blood cells, they need to be removed by macrophages and then killed, and that requires their activation by these pro-inflammatory cytokines. So in the context of malaria too, we also study these T follicular helper cells because, of course, they're essential for helping these B cells produce antibody, which is necessary to remove, or recognize first and then remove parasitized red blood cells from the system. We did, these other cell populations are more relevant for other infections. We did a work for a number of years on natural Tregs, and you heard about these from Benedict yesterday. But we really found a limited role for these particular cell populations, these regulatory cell populations, in the infections we work on. And I'll tell you about a population that seems to emerge instead of these cells to really regulate some of these inflammatory responses. So this is a more simplified version of the CD4 T cell pathway we work on. Macrophages pick up parasite antigens, present it to CD4 T cells. They get the primary signal, of course, through TCR, co-stimulatory signal, and then this third signal through the cytokine IL-12, which is generated following their recognition by certain pattern recognition receptors in the cell surface. And this really drives this pro-inflammatory response to activate these phagocytic cells. However, these cytokines, of course, are really quite potent cytokines. They can induce tissue damage, and so they do need to be regulated. And what we've discovered over a number of years is, rather than those natural Tregs I talked about before, these type 1 regulatory cells emerge. And these seem to emerge in a number of different infectious diseases. Vijay Kuchuru talked about them a little bit yesterday. He mentioned IL-27 drives the production of these cells. We also know from some of the work that we've performed and others have performed as well over the past few years that type 1 interferons are another major driver of the development of these regulatory cells from these Th1 cells.
They produce the potent anti-inflammatory cytokine IL-10, but they also upregulate a number of co-inhibitory receptors on their cell surface, which really regulates the inflammation caused by these cells. So it appears to be a cell autologous regulatory mechanism that's turned on in many infectious diseases. And in fact, if we take samples from the leishmaniasis patients, or we take samples from children in malaria endemic areas in Africa, stimulate them with parasite antigens, the most dominant response that we do see is this type 1 regulatory response in these individuals. So that really begs us the question about, if these people have these pre-existing regulatory responses, can this go some way to explaining why vaccines don't work so well in these individuals? Because the antigen-specific cells that are there are regulatory cells rather than these potent antiparasitic cells. But I'll talk more about that in just a minute. So the goals of our group really are, the problems we address are to work on why this vaccine hyper-responsiveness occurs in disease endemic areas. This is mainly in the context of malaria infection. We know that when we test a lot, these vaccines in Western countries, in malaria naive individuals, we can get 70% protection in those individuals. We take it to the field and it drops down to about 30%. So what's the immunological basis for that? And I just alluded to in the slide before one of our key hypotheses there. Secondly, the duration of the immune responses following vaccination is really quite poor. It lasts somewhere between 12 and 18 months before it drops away. It's really not good enough for these malaria vaccines. That really needs to be improved. And the real danger there in the context of malaria, of course, is because it's looking like that with these vaccinated children, they're not developing the anti-disease response as well. So all we're doing is delaying that susceptibility to severe malaria from a very young age to a later age, which of course creates its own problems. So that's something we really need to seriously think about. And again, we're thinking about it from the immunological point of view, about how we might change this up to actually improve these type of responses. And of course, the way we want to do that is to develop host-directed strategies to improve these antiparasitic vaccines. But we want to do it in a way that we're not being unrealistic. We're not developing antibodies with this process or cellular therapies. We're looking for, I guess, repurposing drugs that are already licensed, have extensive safety profiles, so they can actually be implemented into these areas where they're relevant. The other thing I didn't mention was drug treatment as well. And I think drug treatment is a really underutilized strategy for actually improving host immune responses. We know from malaria studies that if you mass drug treat a community, that the protection in that community lasts a lot longer than the half-lives of those drugs. So we're actually immune boosting that as well. So can we take advantage of that process? We know, for example, in leishmaniasis, visceral leishmaniasis, a disease we work on, that once a person develops disease and then they're drug cured, they're effectively immune for life. So what is the basis for that immunity that comes after that drug treatment? I think there's a really key questions, and I'll get to some of those just now. I guess, so this is a study we just finished. I'm not going to talk about this study today, and I'm happy to talk to people about it afterwards. But I put this up as really the rationale for why we've gone back and done the work that I'm about to tell you about. So we did this, it's a host-directed study in my institute at Queensland Institute of Medical Research. We do controlled human malaria infection studies where we get healthy volunteers, we infect them with malaria parasites, and it's generally to test drug treatments, and we were able to study immune responses. But in this particular study, what we did is we infected people with blood stage parasites. When they reached a threshold of parasitemia, we treated with an anti-parasitic drug, artemether and lumefantrine, plus or minus this JAK1, JAK2 inhibitor ruxolitinib, or a placebo. And the rationale for this was that we wanted to target the type 1 interferon signaling pathway to see if we could at least transiently reduce the number of those type 1 regulatory cells. And we did this study, and it ended last year. We're still going through the data. We generated so much data. But the bottom line was that we achieved what we wanted to do. We transiently reduced the number of type 1 regulatory cells, boosted the number of T follicular helper cells and Th1 cells, but it didn't make a squat of difference when we reinfected those individuals to their control of parasite growth. But what was happening was a whole lot of other things. And what that told me is we really we did not know enough. We don't know enough about basic CD4 T cell biology to really start targeting them in a really strategic and effective way. And so we went back to the drawing board to some extent. And what we wanted to do is really understand more about CD4 T cell subsets. And we chose to do this in the context of our preclinical visceral leishmaniasis model. The reason we chose this is because this model relies on the infection with a human parasite, Leishmania donovani, which is the same parasite that's endemic to northeastern India here. And it gives us a very, very striking, but I think useful response, whereby the liver is a site of acute but resolving infection. If you reinfect the animal, it's effectively that tissue is immune, whereas in the spleen, we get this slowly growing but chronic infection becoming established in the spleen. And this is somewhat akin to what we see in visceral leishmaniasis patients, where asymptomatic patients, which account for about 90% of people infected with this parasite, are able to control parasites very effectively and remain immune from reinfection, whereas those that go on to develop visceral leishmaniasis have this chronic infection, which if it's not treated, the people ultimately succumb to disease and die. So what we did is we set up this particular experiment. It's a single cell experiment, and it really relied on the availability of these cells, which were produced by Judazona. And I should point out all this work I'm going to talk about was performed by my postdoc, Jess Engel, who did a fantastic job. Now, these are CD4 T cell TCR transgenic cells. They recognise this PepsiK peptide, which is an immunodominant peptide, part of an immunodominant protein found in all Leishmania species. So what we did is we had these CD45.2 PepsiKs. We adoptively transferred them into a congenic mouse, different isoforms, so we could tell the difference between the recipient and the donor. Then we infected the mice a day later with Leishmania donovani. This is just the strain name. And then we sampled over all these time points here. So we isolated the spleens and the liver, and then we isolated these PepsiK cells back out of these animals, and then performed single cell RNA sequencing. Now, in addition, with this experiment, what we did is we treated half the mice with ambizone, which is the drug used to treat visceral leishmaniasis patients. Very effective drug. The other half received treatment control, so we could try and understand what the drug effects were as well. So this is all the single cell data concatenated into a UMAP, and you can see it clustered together really nicely well. Jess, my postdoc, said I pulled out about 10 slides, which is all the sort of quality control slides. But because my colleague and friend Jeff Hill, who is at the Fred Hutch in Seattle, said to me, Chris, with CD4 T cell single cell data, generally what we find, you can tell it's going to be really good data if it looks like, if the UMAP looks like a fish. So I looked at this and I thought, oh, that's no good. Then I thought, that looks like a fish. So that was my quality control, and I thought, this is good to go. Anyway, so we looked at these, we looked what we find within this concatenated data. And remember, this is all the data collected in the different tissues over the course of infection. We find these different subsets of cells. These are our old type 1 regulatory cells. We find a number of different types of effector cells. We find these tissue resident-like cells, and we find these stem-like cells that Rafi was talking about in the context of CD8 T cells in the morning in the morning talk this morning. And so one of the really striking things, well, not striking really when we know the disease, is what we found is that all these cells were dominated by a Th1 cell response. They all express TBET. These are the Th1 signatures, not really the others. We do find some GATA3 co-expressed as well, but really a very Th1 dominated response. And this kind of made us, so the implications for this is that we get different types of Th1 subsets here. That's no surprise. A lot of people have been talking about this for a number of years now, but it really made me a little bit concerned. Is all this data simply because we're using this monoclonal T cell receptor transgenic? And so really to address that point and check that, we're fortunate that we also have available a MHC class 2 tetramer, which has the same peptide this TCR transgenic recognised as well. So we did a second experiment where we infected mice, and then we pulled out the antigens, the PepsiK specific cells, using the tetramer, and that's just shown over here. And just for those of you who want to do those experiments, the key point in doing these experiments is to use the control tetramer at the same time, because otherwise you get a lot of non-specific binding. But so we did that experiment, and then just to skip over this really quite quickly, we did the same concatenation of data. overlaid it with the data we had from our PepsiK transgenic cells, and you can see that it overlays pretty well with this data. So it looks like we're getting fair representation of the tetramer positives with the TCR receptor transgenics. We sequence the T cell receptors, alpha beta chains in those cells as well, and you can see we get a distribution throughout the different clusters with those tetramer positives as well. And then we've looked at the spleen and the liver. We get no sort of obvious accumulation of particular clonotypes in one or either of the tissues as well. So we're pretty confident that the transgenic data we generated was representative of a bona fide T cell response. And so we started looking at the tissue-specific differences throughout the experiment here. And I guess the first thing that we came across, and I'll go through this quite quickly in the interest of time, because I'll only talk about it again at the end, is we found these tissue resident-like memory cells, which really popped up in the liver, and they were really amplified following drug treatment, these tissue resident cells. And so we had a bit closer look at the other markers in those cells, and you can see these really markers associated with those cells are really upregulated in those cells from the livers treated with the drug down below. And that's just further shown here, where we're using CXCR6, classical marker, and CD69 for these cells. And you can see in the ambisome treated liver down here, we get a real accumulation of those cells. This is day 56. In the untreated one, we see some accumulation, but not as great as that, and very few in the spleens of those mice. So we see those cells. So just moving on now, another factor we found that was quite interesting was this transcription factor, BHL-HE40, which seemed to be present more in the spleen than the liver in several of these different clusters that we found, the TR1 cluster and these two effector cells clusters. This transcription factor piqued our interest because it's been previously described to play a role as a repressor of IL-10 production. I mentioned before, IL-10 plays a key role in regulating immune responses during visceral leishmaniasis. So we had available a BHL-HE40 knockout mouse. We infected those mice, and you can see they indeed get an increase in parasite burden, so it's playing some role in the host immune response to this infection. When we look at the CD4 T cells in those mice, what you can see is we get a reduction in gamma production in the liver. We see that increase in parasite burden and an increase in IL-10 production. So it's doing what had been explained in the literature, so big deal. But the thing that did pique our interest is the mice lacking this transcription back BHL-HE40 also had increased levels of TCF7 positive CD4 T cells, suggesting that BHL-HE40 not only was repressing IL-10, but was also repressing the generation of these stem-like cells. So the data I just showed you previously was a complete knockout animal, and we can't be sure that BHL-HE40 expression was related to CD4 T cells. So what we did is we went back to our TCR transgenic cells, the PepsiKs, and we had two different lines, one which was CD45.2, one had expressed both CD45 congenic markers, so we could distinguish them when we put them into a recipient CD45.1 mouse. And what we did is we CRISPRed out the BHL-HE40 in one of the lines. We had a control line as well. We put both cells into the same animal, infected that animal, and then look what happened 14 days later. And what you can see is indeed that if we knock the BHL-HE40 out of the PepsiK, we got a significant reduction in the number of these TCF1 positive stem-like cells in those animals. So it looks like BHL-HE40 is a repressor of those cells. So we looked a bit more closely at these stem-like cells to see what sort of role they might play. And what we found quite interestingly is they start to accumulate in the spleen, but then as the chronic infection becomes established, you lose those cell populations. However, if you do treat with drug, this stem-like cell population is maintained. And we're just showing over here the enumerating the cells there as well. They express all the classical genes associated with the stem-like cells shown here. So in order to ask the question about what sort of role they might play, we set up a similar experiment to the one I just described before, where we CRISPRed out the TCF7 in the PepsiKs. We had a control group. We put the same cells into a recipient mouse, then look 14 days later. You can see the TCF7 was knocked out quite well. We get a reduction in the TH1 cells, no change in the type 1 regulatory cells. So this was interesting. So it looked like this TCF7 was playing some role in the generation of these Th1s. It's not a great role, but the problem with this experiment, of course, is we've started with a TCF7 knockout right from the start of infection. As we heard from Rafi this morning, TCF7 is induced in these cells to generate these properties. So what we did is we performed this different experiment. Now, TCF7 is a transcription factor, of course, and so you can't really sort live cells based on TCF7 staining. So what we did instead is we used these surrogate markers, Li108 and CXCR6, which are known to distinguish TCF1 positives and negative cells. And so we sorted those cells and we looked at them. You can see here that these cells here, which express the Li108 high, CXCR negative, TCF1 positive, and the other cells are TCF1 negative. So what we did is we took those cells, we transferred them into a recipient mouse, and a day later we infected those mice. Sorry, no, we didn't do that. What we did is we put them into a mouse, we infected that mouse a day later, and we let it go for 14 days. We then sorted those cells out of those animals as indicated above, and we took the TCF1 positive cells and the TCF1 negative cells, and we adoptively transferred them into a second group of recipient mice that had been infected at the same time, so we're putting them back at the same time of infection, but because they're congenitally marked, we're able to track what happened to those cells during infection. And what we found, interestingly enough, was that we can only pick up cells 14 days later from this population here, the stem-like population. Now, the TCF1 negative cells, we just couldn't pick them up at all. And we really don't know whether this is because they have an inability to expand or we simply didn't put enough cells in there, and with those sort of experiments are ongoing. But nevertheless, these cells here produced an interesting result, and we picked up the cells in everything except the amber zone treated liver here. And this is just my final data slide to show the results from these experiments. And what we found is that when we looked at the cells, they expressed a number of different markers. Obviously, they weren't just stem-like cells. But I'll just point you to the punchline over here. These are two different types of stem-like CD4 T cells that we found. One that was Ki67 high, so proliferating cell, and one that was Ki67 low. And you can see in the untreated spleen, the spleen that wasn't treated with drug, we're getting predominantly those proliferating stem-like cells. In the spleen that was treated with drugs 14 days prior, we get both types of cells. We don't see those cells at all in the liver. So what we're thinking here is that these stem-like cells are really quite important. And I forgot to point out as well that, of course, they generate all these other different types of cells, central memory cells, TR1 cells, different types of effector cells as well. So what we're thinking here is that these stem-like cells might be really quite important for generating these new effector cells either upon reinfection or upon continued infection. And actually treating these animals with drug was a really nice way of actually generating those cell populations. And I think they might be really quite an important population to try and target in the context of either vaccination or drug treatment. So just to summarize this, so we started off with this very simple model. We now know that these Th1 cells come in many different flavors. Our results really didn't come down to the TCR transgenic that we used. We found similar results with the tetramer positive cells. Drug treatment resulted in a really dramatic change in the CD4 T cell subset landscape, and it really resulted in the development of specialized tissue-specific memory T cells. I rushed over at the start, but those tissue-resident memory cells in the liver, and also those stem-like cells in the spleen. And those cell populations were, their presence was greatly enhanced by drug treatment, removing that chronic infection in those tissue sites. So I think they'll be important cell populations to follow up on. So just to acknowledge the people in my group up here, particularly Jess, who did most of the work, assisted by others. I should point out that the next step here is to really try and look at some of these markers in some of the visceral leishmaniasis patients. And of course, that will be done with Rajiv and Shyam Sundar at Banaras Hindu University, who we've done many other studies with as well. And we also get help from others, our core support and our funders, of course. And I'm happy to take some questions. Thank you.
So session is open for question. So,
wonderful talk. I want to ask you for visceral leishmaniasis, and particularly out of 4 species of malaria, particularly falciform malaria, if there's a reinfection happens in the mice, then what is the state of the CD4 T cells and the dendritic cell that is being expressed in the primary infection?
You're talking about malaria. You're talking about malaria specifically. I'm not.
Yeah,
not leishmaniasis.
Plasmodium falciparum.
So malaria's, and I didn't say this, but we work on all the mouse malaria models, and I think they're all terrible, to be honest. And that's why we do our work predominantly with clinical samples and more. Because the problem is that if you infect a mouse once with that species of malaria, you go back and reinfect it, it's solidly immune. And in fact, it's solidly immune to most of the other rodent species of malaria as well. So it's a really difficult model to address that question that you just asked.
Hi, great talk. Just a small technical question. So you use C57 mouse for your experimental leishmaniasis model, right?
Yeah.
So since they have a type 1 immune response bias by default, do you think that could also be exaggerating some of the Th1 subpopulations you're seeing here?
Because of their type 1 dominance, you mean?
Yes, yes.
Yes, yeah, definitely. Yeah, that's right. I mean, the reason we use the C57 black 6 mice because it is a model. It has a lot of the knockout animals in the background, but it's not ideal, and it does have all those sort of caveats you just mentioned there. That it is a pro-inflammatory model in that respect. It does have a dominant type 1 interferon pathway, which we think drives a lot of these regulatory cells that are set up there. So every model has its limitations. I think the biggest thing is to recognise those and test your hypothesis in as many different systems as you can. So as I said, our rationale here now is to take this now into the clinical samples to see if we can find these cell subsets in the clinical samples. We now have the ability to CRISPR out some of these genes in human samples as well. So that'll be the next step as well. So it's really trying to do different.
So one more question there.
Thank you.
Thank you. Great talk. I wonder if I could get you to speculate on what you might expect to see with the human, in the human infections, given the some of the diversity between the mouse and the human CD4 differentiation and activation, not just markers, but also functionality. What do you expect to see in the human system?
So I don't have to speculate. I can tell you what we do see. So
Okay.
from that trial that I put up at the start there, one of the things we did is we did some single cell sequencing there as well. And one of the most fascinating things that I think we found in that trial is that when we looked across at these CD4 T cell subsets we're focusing on, in the different individuals, they were completely different at baseline. So obviously that's their genetics, their previous vaccination, infections, their microbiome, everything completely different. We infected them, and they changed. The subsets changed. Then we went cured them. And we looked 90 days later, and they'd all reset back to where they were at the start. The same subset differentiation. So to me, that says that what we need to do, that's another barrier that we have to overcome when it comes to vaccination or introducing these type of therapies, because everyone's different. So we need to try and find these pathways that are common, and find the really important subsets that we want to try and expand at least transiently, and see if we can embed them with this sort of some sort of stem-like property that then allows them to survive and be able to respond to the next infection.
Yeah.
So thank you, Chris, for your nice talk and completing it well within the time.
I wanted to hear the band.
Thank you, Professor Christian Engovarda and Professor Rajiv Kumar, for this engaging session. We are privileged to host Professor Vishwadip Dikshit, Director of the Yale Center for Research on Aging, USA. I request Dr. Girdhari Lal, NCCS, and Professor P. Kondaiya, IISc, to now take over and guide us through the session.
All right. Perfect. So we finish in time and we'll again finish in time. That's organizer has reminded. So it's a great pleasure to invite Dr. Vishwadip Dikshit, and he has finished his DVM and MS from Haryana Agriculture University, and then PhD started from Haryana and finished in Hanover. He's a fantastic work on immunometabolism and effects on hormone on thymopoiesis, especially looking at how thymic regeneration and T cell senescence. And when we say senescence, aging comes automatically. So he's going to talk about the immunometabolism, how diet influence it, or his work mostly on those areas, and how the immune system and immunoaging is going to affect. So Dr. Dikshit, floor is yours.
Thank you. All right. Thanks again. It's really a great privilege to be here. It's my first time in IIC Bangalore. I was lucky enough to be at the IMMUNOCON meeting last year, and it was great fun, and it's very nice to be invited again. So thanks to the organizers for doing that. So we're gonna, I think, switch gears and talk about something different, which is aging. And so I guess this slide I see there are several students here. And so for those of you who don't really think or work too much about aging, this slide kind of crystallizes why we think this is one of the most fundamental biological problems that affects almost every species, not almost every species, in fact. And the reason why aging is especially important and interesting to a biologist or to a biomedical scientist is shown in this slide, which is very straightforward. So if you notice that something happens as we get older, which is that by the 5th decade of life, the risk of almost every chronic disease increase with age, right? You can count those diseases here. You name it: heart disease, type 1 diabetes, of course, chronic kidney disease, and Alzheimer's disease is one of the major ones. And the question in the field has been whether this is simply an association or whether there is some sort of a biological mechanism underlying the process of aging itself that predisposes one to multiple diseases, right? And this is what the field is trying to understand. Because the idea is, which is a rather lofty goal in my view, the idea is that, let's say, if we are extremely successful and let's say we cure heart disease and we have a great drug to cure heart disease, the problem is that as we get older, that you may just trade heart disease at that point of time with chronic kidney disease or Alzheimer's disease or another form of cancer. In other words, if one understands the basic mechanism that drive the process of all these diseases, and if we identify that and if we target that mechanism, can we potentially delay the onset of all these age-related diseases at a time? And in this slide, you again can notice why aging is such a major risk factor. And if you just compare cancer, okay, we all know that smoking is a very major risk factor for cancer. But if you just compare smoking to just aging, you would notice that it's like 30, 40-fold higher. Same thing is with heart disease and with Alzheimer's disease. And why this is important is because we are all aging, right? This is the demographics of the world. And you would notice that within the next 20 years or so, the number of individuals that are going to be above the age of 65 are going to be higher, close to 2 billion just in the next few years, all over the world, including India, right? And I still recall when I was a postdoctoral associate at the NIH, and somebody was giving a talk, and there was some young student who stood up and asked this question to this professor, Richard Miller. He says, "What is aging?" The guy used to work on CD4 cells, nothing against CD4 cells. and I remember Rich Miller's answer. He was kind of perturbed by the question because of the dismissiveness of it. So he says, aging is what converts a graduate student into a professor and a professor into a dead person. Which means it's probably relevant to all of us because aging affects everyone, right? And this is not only relevant from the disease point of view, but also it has major economic issues in the society, as one can imagine. So, if you put economists from London School of Economics and a scientist from Harvard, you get huge numbers. So what these people so they did some modeling and they came up with this concept that if we extend the lifespan, healthy lifespan by 1 year, it is going to, see, that's aging. I need to wear glasses to actually read it. How much is that? $38 trillion in 1 year if one increases healthy lifespan by 1 year. Of course, I'm not neither an economist, and I don't really care too much about the money part of it. And the reason why we are interested in this for simple reason that aging is the biggest risk factor for diseases, and there are mechanisms that underlie this process that has a huge impact on the organism. So some of them are listed here. These could be considered as hallmarks of aging itself. And I'm going to talk a lot about inflammation in this process. But what happens with age is that there is increase in inflammation, there is elevated molecular damage, of course, cellular stress, changes in reactive oxygen species, there's metabolic dysfunction, impaired proteostasis because of alterations in autophagy, stem cell exhaustion, and of course, transcriptional dysregulation due to methylation. And interaction between these various mechanisms is thought to lead to this kind of a vague term that we use, which is called loss of resilience, right? But the loss of resilience by itself does not cause disease, right? So how does disease manifest with age? So in this background of loss of resilience, if you have an associative trigger, that trigger in the background of loss of resilience because of interaction of these above mechanisms leads to a particular disease. So I think the most binary example of this interaction and the manifestation of age-related disease is this disease called as gout. Okay, gout's a disease of elderly, but gout's not going to happen unless or until you have urate crystals, right? Same thing one can say with infections and increased risk of infections and mortality of infections in elderly. That's not going to happen unless you have a pathogen. Same thing with heart disease, you have to have dyslipidemia as an associative trigger. Okay, so these are the major mechanisms that kind of regulate the process of aging itself. And I don't want to go into too many details, but we're beginning to understand that, of course, immune system is central, absolutely central to almost every mechanism that is at play in biology, in tissue function, and so on and so forth. And these are four very main features of immunological aging. First, I think you're all aware of, which is involution of thymus. Thymus involutes with age. We have less naive T cells. There is restriction of T cell diversity. I'm going to talk about these other three hallmarks as we go along. I'll also discuss a little bit about origin of inflammation and how inflammation is manifested. But this concept of increase in inflammation with age is really not a new concept, right? This was actually put forth by Ilya Mechnikov those days, to almost 150 years ago, when inflammation was really not well understood. So since there are students here, you all know that Mechnikov got a Nobel Prize for discovering phagocytes. But for students, I think it's also important to remember that he was a guy who coined the term gerontology. Okay? So he was one of the few immunologists, actually the starting immunologists, who actually studied aging in great detail. And he wrote this rather controversial book when he was at the Pasteur Institute called Prolongation of Life Optimistic Studies. I suggest you read it if you get a chance. It's a very interesting read. And Meshnikov kind of predicted in his book that, and he said that the phagocytes resist the influence of invading poisons better than any other cell of the body, and sometimes are stimulated by them, producing disease. And of course, this is 150-year-old science, so we did not know, we didn't know all the fancy words. Now we have a little bit fancier words for poisons and what stimulation means. But basically, Meshnikov was predicting that macrophages will have something seminal to do with the diseases of aging, and now we have a little bit of evidence for it. So this is a macrophage that has been isolated from a thymus of a mouse that is about 18 months, which is fairly old for a mouse. And you would notice that this macrophage is accumulating these poisons. Okay, we call these poisons now damage-associated molecular patterns. So here is presence of lipid within this macrophage. We don't quite know where this lipid is coming from, whether this is de novo lipogenesis or whether this lipid is accumulated because macrophages are phagocytosing cell membranes from the dying T cells. There are protein aggregates right here. This is nucleus, and these are lysosomes, right? And lysosomes are full of electron-dense material, see? And you can also notice these structures that are present within these macrophages. These are crystals, right? And these are all the poisons that Mechnikov was talking about. And because these cells are so resilient in a way that they're able to clear these things, but with age, they keep on accumulating. And now we know that these things are sensed by very specific pattern recognition receptors. And NLRP3 inflammasome is one of the major regulators or the major sensor of that. This is a cytosolic pattern recognition receptor, which was first discovered by Jörg Chopp in Lausanne in Switzerland. And it is especially important for aging because NLRP3 senses molecularly diverse damage-associated molecular patterns or so-called poisons. Okay, so with age, we know that all these things increase. This includes extracellular ATP from dead or dying cells, lipotoxic molecules. And what happens is that these things are sensed by NLRP3 inflammasome in the macrophages. It causes the activation of assembly of this, this complex, which activates this cysteine protease called caspase-1, and caspase-1 cleaves to pro-inflammatory cytokines IL-1 beta and IL-18, leading to this process called as inflammation of aging or inflammaging. And we and others were able to show that if you go upstream, at least genetically, and you downregulate the NLRP3 inflammasome, not only can you reduce inflammation, but actually you can protect an old animal from multiple degenerative diseases, including bone loss. These animals have improved glucose tolerance. Their thymi were less involuted. They were less frail. They have improved cognitive function. And then you would wonder, wow, that sounds pretty fantastic. And the answer is actually yes, because it is really what Mechnikov was predicting is what now the data we are finding after 120 years. And the answer truly lies in tissue-resident macrophages, right? Because tissue-resident macrophages are present in every organ, ranging from microglia to osteoclasts. And now there is more and more evidence that if you actually lower NLRP3 inflammasome, at least in mice, the animals live about 15 to 25% longer. In other words, aging is not just a stochastic process. There are mechanisms that control inflammation of aging, which when downregulated, would not only improve health span, but also actually extend lifespan of an experimental animal, shown here. And this is, for example, in a model of progeria. If you knock out NLRP3 from animals that have massive DNA damage, these animals also live longer. So the question really is, how is inflammation originating in aging? Why does inflammation happen with aging? And this leads us to thinking about some old papers. This is a paper from Dr. Charlotte Preston Mangum, who did her PhD in Department of Biology at Yale in 1960s. Unfortunately, her work is forgotten in a way. And she was brilliant and came up with this concept of enantiostasis because she studied physiological adaptations in unstable environments. And she describes enantiostasis as an adaptive state where, despite an unstable internal cellular milieu, the net effect of regulatory flux is stability or tendency towards it. Okay? It's not homeostasis because because here your cellular milieu is still unstable. I don't know how many of you are very stable at this point of time. I'm not, but we are still functional, right? Because every organism has these mechanisms where they try to adapt and have the regulatory flux towards that, right? So an aging, I think, is especially a state of anangiostasis, because internal milieu in most of individuals that are aging is unstable. There are systems that are out of balance, but at the same time, the overall process is towards stability, or at least maintenance of stability. And we think that the idea is that if we to conserve the suboptimal function, or to stall the further precipitous decline during aging, the endogenous and anangiostatic molecules have to be engaged so that they can oppose this dysfunction, right? Because if there would be no control of these mechanisms as we get older, we simply would not be able to survive. And of course, we know not all of us age in the same way. Many of us age in a very, very healthy way, right? So why does inflammation happen with aging? We actually do not have any idea at this point of time, only some speculations. And what we particularly think is that the breakdown of this adaptive anangiostatic process leads to some sort of an abnormal equilibrium, right? So that there is like a chronic inflammation that happens. It's not like an acute inflammation that you heard today in the talks with various infections. It's a chronic low-grade inflammation that perturbs the system. And where is this inflammation coming from? One of the tissues that we are extremely interested in, and especially relevant in case of aging and perhaps in several other diseases, is the adipose tissue. So you're looking at a CT scan of a 31-year-old person on the left and a 74-year-old person on the right. You don't have to be an MD or radiologist to discern this image. And actually, my sister is here in the audience, and I promised her that I will not delve into any jargon, because we immunologists typically get told, oh, you guys use too much jargon. I said, okay, fine, no jargon. And what you would notice is that with age, even if you maintain your body mass index, okay? So these two individuals have same body mass index, only difference is age. But what you notice is that there is increase in visceral adiposity, there is increase in the fat in the viscera. Why this is important? This is important because adipose tissue is an immunological organ. So you're looking at whole mount confocal image of a mouse where everything that is green is indelibly marked with GFP. Okay, and these are myeloid cells that are indelibly marked with GFP. And you look at all this green spread all over this adipose tissue, and you would think, wow, there's something wrong here. There is inflammation going on. There's something wrong. Actually, there's nothing wrong. This is a healthy 2-month-old adipose tissue, which is chock full of myeloid cells or resident macrophages. And these cells are integral component of this metabolic organ, perhaps organs, multiple organs. In other words, if there are students here who are interested in immunology, there's nothing better to do than to study immune system, because immune system affects everything in the body, right? And we think that the interaction between these macrophages and the fat cells in individuals with age, as we increase visceral adiposity, has something to do with the origin of inflammation. I'm going to run out of time if I go into too many details, so I'm going to skip through a lot of these slides. As I promised, I'm not going to go into the jargon. But if you look at the single cell analysis of exclusively tissue resident cells in the visceral fat of a mouse, you would notice that almost every hematopoietic lineage is present in this organ, right? And if you compare that to an old animal, you would notice major differences. Some things are totally binary. For example, the loss of ILC2s. There's also reduction in homeostatic macrophages. The alpha beta T cells, effector T cells, expand with age, shown here. And together, this is leading to increase in inflammation. And other thing which is very interesting with age that happens is actually expansion of B cells. Okay, and these B cells are very unique. We call them age-associated B cells, or aged B cells that were first described by Mike Cancro in the spleen. And these B cells actually reside in tertiary lymphoid structures, which form in animals, in old animals, without any infection. They're in a specific pathogen-free environment. And what we were able to show that NLRP3 inflammasome through IL-1 beta and IL-18 control the activation of these cells, which expand with age. And if you prevent that expansion, one can lower not only inflammation, but also metabolic disease. And just to add to this complexity, the adipose tissue is highly innervated. There are lots of nerves in there. And you're looking at a 2-photon image of a healthy mouse. Everything that is in red is non-recombined tomato, and in the green are cells that are macrophages that are indelibly marked with GFP. The squiggly structures that you see here is a sympathetic nerve. And you can notice that there are these macrophages that are just sitting on these sympathetic nerves, right? And they're doing something. We don't quite know what they're actually doing, but we think they're doing something really, really interesting. I don't have time to talk too much about it, but maybe if you call me next year, I will give you some more details. But the point being that there are unique subsets of macrophages that are not only important for protection against inflammation, for causing inflammation, priming immune responses, protection against pathogens, but also regulation of organ function through regulation of neuronal function. So we call these cells as nerve-associated macrophages, and this is sympathetic nerves, and you can see these beautiful green macrophages that are hugging these nerves, right? So these are, in my view, like the peripheral glia. They are the peripheral glia of the nervous system. Just like we have microglia in the brain, these are macrophages that are present on the nerves. And we had shown several years ago that with aging, these macrophages have a very unique function. Okay, what they do is because sympathetic nerves control your metabolism, all right? Your metabolism is not in your conscious control. It's controlled by your autonomic nervous system. And what happens is that sympathetic nerves release this neurotransmitter called as norepinephrine. This norepinephrine is responsible for actually causing the hydrolysis of your fatty acids. And those fatty acids will enter the mitochondria, into the TCA cycle, and so on and so forth. This would happen through this signaling on the adipocytes. But these macrophages that reside on these nerves with age increase the expression of this enzyme called as monoamine oxidase A. Previously, this enzyme, by the way, was only studied in brain in context of depression, because monoamine oxidase A inhibitors were used as antidepressants because it basically elevates the amount of dopamine. But what monoamine oxidase A does, if it is very high, it will break down the norepinephrine. So what happens with age is through inflammasome regulation, monoamine oxidase goes up, these macrophages take up catecholamines, break it down so that adipocytes are no longer able to see it. Okay, so like my title of the talk suggested, metabolic checkpoints of aging, and you would wonder, are there actually metabolic checkpoints of aging? And I'm going to convince you, yes, there are, in a few slides without showing you a lot of data and not going into the jargon, otherwise I'm going to get in trouble. So here is just a very simple slide. I'm not going to go into the details. Like I mentioned, we and others have shown several years ago that one of the metabolite called beta-hydroxybutyrate, which is a ketone body, which is produced when you're fasting or when you're starving, is a very potent molecule that would inhibit inflammation and actually expand gamma delta T cells and regulate inflammation. But we are interested to actually identify new metabolic targets that perhaps not only regulate inflammation, but maybe even impact lifespan and health span. And one of the interventions that have intrigued me for quite some time is this intervention called as caloric restriction. Almost 120 years ago, Clive McKay found that he forgot his rats in cages and forgot to feed them, and it turned out that they lived close to 50% longer. And this has now been shown over and over again. I think there was a paper in Nature last week in genetically diverse animals that the caloric restriction is extremely efficient in extending lifespan. There is also now data that the monkeys in the National Institute on Aging also tend to be living longer. So what happens during caloric restriction when you're eating less, right? What's happening is that you are in a state of negative energy balance. Energy. That's what metabolism does, among many things. So when you're eating less and expending more energy, you're in this negative energy balance state. And this is a state when your body has to now switch fuels, right? Because in this state, your in a state where your glucose is limiting. So you have to now mobilize your fats. That's a process through where ketone bodies would be elevated. And then there is integration of immune metabolic axis. And then when this happens, there's actually maintenance of homeostasis and longevity. And the question really is, why does this happen? Okay, it's very intriguing. Why would reduction of calories actually maintain homeostasis and increase lifespan? But there's a very dirty secret in the field. Okay, and the dirty secret is that if you keep animals and you restrict their calories, they have trouble maintaining their core body temperature. Okay, they don't grow very well. They have a somatotropic, their somatotropic axis is retarded or inhibited. They do not breed. Okay, and as soon as you take them out from a specific pathogen-free facility, challenge them with any infection, they fall dead, right? So in other words, is this longevity coming with a trade-off? Is it coming with a systems trade-off? And if it is coming with a systems trade-off, and we make our mice live 50% longer, that's great for that mouse, but what does it mean for human biology? So, so the idea was to actually do a randomized control study in people. Okay, and the reason for this is because your and my mice do not sign consent forms. Mine don't, definitely, right? So if you're a mouse and if you're in a cage and you give that mouse 40% less food, the animal doesn't know if the food's going to come or not, right? But a human in a randomized control trial, if we convince that person to do it, I'm never going to do it. I have 4 minutes, I know. I took 15-hour flight, so I can probably go 4 minutes over, right? So the point being that one has to be able to do these studies in people. And because of all the issues that we have in the animals, first of all, of course, inbred strains have resolved most of their issues. Our mice are in specific pathogen-free facilities. And that was the idea. That was how the trial was done. The trial is called as Calorie. It was done at 3 sites. This trial was initiated in 2006. That's when I started my lab. Okay, you can see I've gotten older. And the idea by the NIH, National Institutes of Health, was that we take people, healthy people, restrict their calories by 25%, okay, and keep them for 2 years, and then see whether it has done something or not. And that's what we wanted to see, whether it's actually relevant to human physiology. And if it is relevant to human physiology, can we identify anything that are core mechanisms that impact the immune system? And this was the general idea, okay? And now this work has started to come out. It took a long time. But here's just a very, very quick overview of what we had found, which is, if you take the adipose tissue biopsies of these individuals, I told you how important adipose tissue resident immune system is. So these are people that came in at the baseline after 1 year and after 2 years of caloric restriction. And you can see there's a major remodeling of the transcriptome. And I recall that when we sent our data to our colleague, Max Ardemov at WashU, and he says, Deep, your knockout mice, we all obviously sent him data in a blinded fashion. He says, oh, your knockout mice have a great phenotype. I said, Max, as far as I know, they're genetically diverse, outbred human beings. But you can imagine just a simple metabolic intervention has a major, major impact. And what we were able to do in this study was to basically now identify and harness what are those enantiostatic adaptive endogenous mechanisms that are engaged, that confer, that act on immune system and regulate inflammation. And I'm going to skip over that and basically just summarize that we have found 2 of those molecules. There are more coming on the way. One of them is called PLA2G7. It's also called as platelet activating factor acetyl hydrolase. It's a secreted enzyme that's made primarily by the macrophages. It is inhibited in humans that undergo caloric restriction. We were able to create animals that specifically lack PLA2G7 in their myeloid cells, and we're able to show that reducing this protein prevents inflammation and diseases of aging. The other molecule that we identify is a matricellular protein called a SPARC. It is produced from the fat cells, or adipocytes. It acts on macrophages to cause inflammation, and this is also a protein that is inhibited by CR. And we created animals that we specifically lowered its activity to show that it is actually a causal factor. So new targets, new metabolic targets that impact immune system. So a few takeaway messages here is, first of all, if you're in a negative energy balance state, which is induced by caloric restriction, which does not cause trade-offs. In humans, these people do not have any trade-offs. They do just fine. They have reduction in inflammation. There is enhanced thymic function, in fact, also in these people. Okay, and we are harnessing those molecules. Another thing which was very interesting, I think for those of you who are heavily into diets and stuff, which is that all one has to do is reduce their calories by 15%. Doesn't matter which time of the day you eat it. Believe me, I am not one of the proponents of caloric restriction, but this is what the trial found. And I'll end by thanking my colleagues and my lab, which is here. And of course, this is a kind of work, especially the human work, is very difficult to do until you have a big team. And we are blessed with those and the funding from the National Institutes of Health. And sorry if I went a couple minutes over, but more than happy to answer any questions. Thank you for your attention.
Thank you, Professor Dikshit. Very nice talk. Any questions? Yes.
Thank you for a wonderful seminar. Here, over here.
There you are.
It's just a curved question. Is it possible that people starve, then they feel that life is never ending?
That's a good question. People make a lot of jokes about it that, if you're doing caloric restriction, it would seem that you're living really long because every minute takes, you feel it. Therefore, I don't do caloric restriction.
Yeah, sir, nice talk firstly. And my question is on, there was no mention in the entire presentation about anything related to autoimmune, because I mean, autoimmune disorders are regulated by immune system. And since, for example, if you take psoriasis in a case, so the inflammation is all due to autoimmune responses. So how is that correlation with aging? And if any study has been done on that, could you please elaborate on that?
If the chairperson would allow me to give another talk, I will give you a talk about autoimmunity, but unfortunately I don't have time for that. One of the things that happened with age is increase in autoantibodies. Okay, it's very well known. What is not known is that because the exoproteome of those autoantibodies is not well known. We don't know against which proteins those autoantibodies are in aging. A specific autoimmune disease like psoriasis that you're talking about has a very different mechanism than inflammation or autoimmunity that emerges with age. Most of the time, it is autoimmune diseases are manifested early in life, some later. So the question about the inflammation that is coming with the autoimmune part with aging is not a settled question at all. We know almost nothing about it. But what is known is that the age-associated B cells are increased. These are CD19, CD21 negative T-bet positive cells that make a ton of autoantibodies also. We don't quite know against which protein those autoantibodies are. Some of them probably are protective, some of them are not. And I think time will tell if we can target those autoantibodies. But in terms of psoriasis and aging, there's not that much overlap of the mechanisms that I was talking about.
Last question.
Professor Dikshit, a wonderful talk. I want to ask you, the senescence and the aging,
yes,
there is a telomerase shortening for all these factors. Any cascade of event that have shown is directly or indirectly related with the telomerase shortening?
Yeah, that's a good point. That's why Liz Blackburn got her Nobel Prize with the telomerase, and it has been studied quite a bit in the context of senescence. And the evidence so far with the telomere shortening and the diseases of aging or aging itself is still evolving. Okay. The diseases where you have the maximum effect of telomerase and the telomere shortening are the diseases where you have the maximum DNA damage. Okay, so if you take a model where there is massive DNA damage, like ERCC knockouts, those animals have progeria-like disease, and telomere shortening is very, very obvious. And there's a reason why the telomere shortening in context of all the aging pathways do not always show up. The second thing is the senescence itself. So senescence, as defined, is a cell that has undergone irreversible cell cycle arrest, right?
Thank you.
And if you look at those cells in vivo, there are very, very few cells that are actually senescent, less than 0.1%. So we know very little about what those cells are and whether those cells are due to increase in telomere shortening. And if it is the case, whether those cells are the ones that is causing aging. And so far, there is evidence for that is still evolving.
Thank you, sir.
Thank you very much, Professor Dikshit. I'm sure a lot of questions will arise because of the topic, very interesting, and this can be discussed in during the tea time. Thank you very much for a very interesting.
Thanks for your attention.
Thank you, sir. I would request for a group picture with the chairs.
Group picture, why? You want to document my age when I come next time? I can age a lot in a year.
Thank you, everyone. We now transition to an interactive session with Dr. Uma Kanga from AIIMS, New Delhi. I request Dr. Ambak Rai from MNNIT to now take over and guide us through the session.
How do we move the slides? Just this? You can use this. Either you can use this or you can use the pointer. It will go like this. इसको आगे करने के लिए इस पीछे. I will use this. Okay, ये pointer है. Okay. Pointer कौन सा है? ये वाला. Okay. May I start? Or in the interest of time. Maybe Gildhari is here. We can
yes, otherwise और late हो जाएगा. चल.
A very good evening. I think I'll fill in because I think Ambak is not reachable right now, but I'm sure he'll come. But anyway, most of us are familiar with Professor Uma Kanga. She is in AIIMS and she has been working a lot on MHC. She has also contributed a lot to the Indian Immunology Society and she was the convener of the Golden Jubilee Indian Immunology Society meeting in AIIMS last year. Uma, please take over.
Thank you, Dipankar, for the kind words and also for the kind invitation for me to be here and talk to you all, our society members. As Dr. Dipankar said, this is my institute where I got trained, and this is where I'm working at the moment. And I thank the institute for all the opportunities I have got for learning and for sharing the knowledge. So as majority of you, or maybe some of you who know Lord Ganesha, would believe that that is the long time back history of transplantation. That was something that we have looked at, and we as transplant immunologists always look it up, as that's the best model of transplantation that we saw. But for the others who don't believe in Hindu culture and all, these are the two people who actually brought this transplantation field to the scientific fraternity, and Sir Joseph Murray and Don Thomas, who done the first stem organ transplant and the first bone marrow transplant, and they were awarded with the Nobel Prize in 1990 for this contribution. Majority of you may know many organs are transplanted, and each one of us can also give this gift of life as live donors, and also we can pledge our organs when we pass away, or we are in a situation where we are brain dead and the organs can be harvested. So I urge all of you to look at the NATO website of the Indian government and also pledge our organs if we are in that age range that we can donate organs. It's a big teamwork and lots of specialties involved. And unless all the specialties work together, it cannot be a successful, vibrant program. And I'm told by my nephrology colleagues that there are about 2.5 lakh individuals who would need a transplant every year, and only about 8 to 10,000 of them can undergo a transplant. And there are several reasons for that. And those who cannot lose their lives. And this is the best therapeutic modality, the replacement of the organ. However, because of the shortage, and the country program is live related, we have only about, a majority of our transplants are 85% of them are live related, and family donors have to be available. It's a very less deceased donor program, so we have very big shortage of organs. And then the cost is prohibitive. A lot of immunosuppression has to be given pre and post transplant, and this immunosuppression leads to a lot of complications like nephrotoxicity, infections, malignancy, and we need to look at how do we predict, that is there going to be a rejection, and how do we stop that rejection. So what are the factors that affect the transplant outcome? One of the major factors is the genetic matching between the recipient and the donor, and that's what we call it at the HLA gene matching. The sensitization of the recipient, the donor-specific antibodies, if present, can also harm the graft. And also the variability in the drug absorption. The immunosuppressive drugs don't stay longer uniformly in every individual, so we have a different metabolism. And infections, as you know, in this part of the world are a major reason. And that's where among this whole team of specialists that are involved, there comes the transplant immunologists. And we have to do a lot of investigations, the immunological monitoring of transplants, by looking at their gene matching, their cross-reactivity, the antibodies, and what kind of antibodies there are. So the immunologist's role in a program is very key. Why? Because when a transplant happens, in the initial stages, the dendritic cells from the allograft will move to the recipient organ, and that is where the direct recognition of the antigen happens. And gradually, over a period of time, the cells from the recipient move to the organ, harvest the antigen, and then they are presented. So there is a dual antigen recognition, both direct and indirect. And this alloimmune response needs to be controlled. Unless this whole immune system is kept under check, these alloreactive cells will migrate, they'll adhere, migrate to the graft organ, and will damage the graft. So a lot of checkpoints to be taken care of. So first of all, the genetic matching between the recipient donors, and this is the gene organization of the HLA-MHC system, which is located on human chromosome 6, short arm, and these are multiple genes that are in this region. Many of them functionally immunologically relevant, but for the transplant purposes, we look at the classical class 1, ABC, and the class 2, DR, DQ, and DP antigens. Highly polymorphic genes. Most of the polymorphism lies in the peptide binding region. So if we look at the cell surface of any individual, there would be 2 HLA-A, B, C, DR, DQ because of the 2 chromosomes. And then if they are homozygous, like in the lower picture, if they are homozygous, you would have limited number of HLA alleles on the cell surface. Whereas if one has heterozygous on all the loci, there'd be about 18 different HLA types. So matching a recipient and a donor is a huge task where you have so much of variability between one individual to the other. And also this polymorphic system, at least about 38,000 genes are known at the moment, and we need to have robust technologies to be able to identify non-ambiguous results for the HLA typing, and to a higher level where we can also look at the coding and the non-coding regions and the expression of these molecules, which vary from individual to individual. So these multiple technologies are available. All molecular-based methods are used at the moment, and we've also had established all of these technologies in our lab, and we keep on looking forward to the technology developments, and so that we have accurate typing. So at the moment, the next-gen sequencing is the most commonly practiced protocol for HLA typing, because it's a long region, it's a 4,000 kilobase region, and then we have to do the continuously look at the the alleles which are we have to be phasing, and the complete phase of the sequence would identify the exact HLA alleles. And we have in our laboratory established this and studied the population. And the one more major important relevance of this technology is that we are able to identify novel sequences which we are not knowing when we do it. Previous to this technology is the Sanger sequencing or the reverse blot technologies. So we have also in our lab studied several populations, published these novel HLA alleles, and contributed to the International HLA database. And from all, just about 1,000 individuals from the Indian population, we were able to identify about 40 odd novel genes. So you can imagine, if we don't use this technology, we are not able to identify exactly the gene types that we have in this individual. I'm sorry, the figure is missing, but just wanted to tell you that how the HLA matches and mismatches are being taken into account. So there's a possibility that you could have a recipient and a donor where they would match exactly for all the HLA alleles, but then sometimes you would have, like example 2, a donor which is homozygous for HLA-A. So there is a match, but there is no mismatch here. So it's not exactly a 6-on-6 match, but there's no mismatch. Similarly, in the example 3, there is a 0 mismatch. So we consider matching as more important, but we also have to keep in mind whether there are any mismatches or not, because mismatches are the ones that lead to development of antibodies, and that are poor, real detrimental to the graft. So you can see that the overall survival of the grafts, more the number of mismatches, higher, poor the graft survival over a number of years. This is a 5-year graft survival, which is indicating. And also, if we use better technologies, the curve on the right will show you that if you're using better technology, the mismatches at the DR, if they are not there, then the graft survival is much better. Also, the episodes of rejection, the HLA identical siblings, less percentage of reaction. The middle graft is also a half match siblings or parents, and then the unrelated individuals, deceased donors, where the rejection episodes would be high. If you look at the proportion of time for the graft function, that also decreases with the increase in HLA mismatch. You would see the green bars where the mismatch is less, the blue bars where mismatch goes to 2 to 3 mismatches, and then further down. So this also reduces because of the mismatches, and that leads to development of antibodies. About 25% of grafts will develop antibodies, and these antibodies can be very harmful. So how do these antibodies come in? They can be due to pregnancy, they can be due to transfusions, they can be due to transplants. So these antibodies, if they are against the donor graft, the donor kidney or donor or organ, they can be very detrimental. And sometimes these antibodies can also be cross-reactive. So if you look at this figure, there are these antigens which have public epitopes, and there are certain antigens which have private epitopes also. So if you are identifying an antibody, we need to exactly identify which private epitope of the antigen the antibody is binding. And sometimes if they are sharing the public epitopes with other HLAs, there could be cross-reactivity, and there could be more alloimmune response in such cases. And these cross-reactive groups are identified and published, and more the number of alleles for which if we see cross-reactivity, when one needs to avoid such kind of donors for these patients who have developed the antibodies. These antibodies can activate the complement, they can act to the ADCC phenomena, do the opsonization or activation of the endothelial cells, and all of this will affect the graft, and the survival would be really poor. I'm just giving you one example of a recipient donor where we performed this HLA typing and we looked at the antibody. We found there were 2 antibodies, HLA-C and HLA-DRB1. However, we didn't have the HLA types of the donor, and we were not able to say whether it's a donor-specific antibody, the transplant should go ahead or not. However, when we performed further assays, we did the exact HLA typing, and we could see that these antigens are observed in the donor, and we could say that these are donor-specific antibodies, and we could tell the clinicians, yes, we would prefer not to do such a graft because there would be antibodies that will attack the graft. And then we also need to understand what kind of antibodies are these. Are they transient antibodies? Are they persisting? Are they the preformed antibodies prior to the transplant, or de novo antibodies? What class against which HLA they are? Whether they are the IgG or the IgM, whether they can fix the C1q, whether they are CH3 binding, and what intensity it is. So there's a huge task involved in identifying these antibodies, and nature of these antibodies, and characterizing. And they do impact, if you look at this graph, wherein you can see that if there is an antibody to HLA-DQ, the graft survival is, rejection-free survival is lower. And if these are donor-specific, DQ antibodies, they are much, much impactful in also the AAMR. And if these antibodies bind to the complement, this complement fixing the complement system gets activated, the C4D will get deposited with the membrane attack complex and the graft will be damaged. And you can see in this graph that if there are donor-specific antibodies to HLA-DQ, the graft survival is lower. However, if these antibodies are C1q fixing, there is a huge difference between the survival, and also if the strength of the antibody is much higher, the graft survival goes lower and lower. So very important to characterize. Herein, if you look at these antibodies which are C1q fixing or the IgG3, the mean predicted probability of a graft loss is much higher, whether the assessment is done prior to the transplant or during the post-transplant scenario. So one needs to characterize really these antibodies. Here the data indicates that in the antibody-mediated rejection episodes, the C3d binding antibodies are much higher, and the C3d negative DSAs, the donor-specific antibodies, have a very low MFIs, the strength, as compared to the C3d positive antibodies. And majority of these C3d positive antibodies are able to also fix the lead to the C4d positivity, whereas if the C3d negative, less percentage of patients experience. So that's where this publication indicates that the presence of these C3d binding DSAs is an independent predictor of the graft loss, and there could be future clinical trials aimed at testing the efficiency of complement inhibitor agents, which can then block this complement fixing activity. Also, when you look at the nature of these antibodies, sometimes these C1q positive, C3d positive antibodies will persist and they will not wash off from the graft, so the transplant cannot be held if that is the nature of the antibody. Again, here data showing that if there are C3d positive donor-specific antibodies, the probability of severe cardiac allograft vasculopathy in the cardiac transplant is also really, really higher. So these antibodies actually are very detrimental. So HLA matching helps us in prolonging the graft survival, minimizing the rejection episodes, preventing the sensitization because lesser amount of antibodies are developed, and provides a better chance if the patient needs a retransplant, because if there aren't any antibodies, a retransplant is much easier to handle, and then reduces the percentage of death due to graft dysfunction. So in this expensive therapy, it's really important that we manage the allograft well. And coming to a few slides on the hematopoietic stem cell transplant, from the solid organ transplant, if we move to stem cell transplant, we need to have a very well-matched donor. So what happens when we do the matching? The recipient undergoes conditioning therapy to kill the defective marrow. And once this conditioning has happened, there would be like a lot of space created in the bone marrow. And the cells that are harvested from the recipient donor are then transfused to the patient, and then they get engrafted, and then the patient is improved and now on the GVHD prophylaxis discharged from the hospital. However, despite this good HLA matching, the milder conditioning regimens that we use and better immunosuppression, the major cause of morbidity and mortality is the graft versus host disease. Here again, because of the conditioning, the donor immune system is activated. Because of the conditioning, the host tissue is damaged, lots of cytokine released, and all of these direct and indirect allorecognition milieu, if it leads to an inflammatory pathway, there could be GVHD, which can be very, very harmful and lead to death if it's a grade 3, grade 4. So again here, matching between the recipient donor is one of the major factors that can influence the graft, although the disease, whether malignant or not, and the conditioning regimens, the stem cell dose, or we deplete the donor T cells also affect, but that is one of the important factors. So in this case, we really need to have a perfectly matched HLA sibling from the family who has exactly inherited the same chromosomes from both parents and can serve as a donor. But if we don't have such a donor, in that case, nowadays the transplants can be done with a half-match sibling, or every individual has a parent as a half-match, because one chromosome is definitely matched. So such transplants are also now started because such donors are always available. Patients don't have to be told that, no, you don't have a donor. At least the therapy is a little different, little difficult transplants to handle. But then it's at least the patient can undergo a transplant. But when there is a mismatch, although the overall survival and disease-free survival is comparable, but when there is a mismatch, there can be leading to antibody, and there could be early, graft failure. And when there is early graft failure, the overall survival really goes low. So one needs to also control over here the antibody development. So unacceptable or highly HLA mismatches need to be looked into. So not now I must say that it's not just the HLA matching. Some of the HLA mismatches can also be acceptable. So if there is this extensive polymorphism leading to low probability of finding a donor, but then there could be some acceptable mismatches. And how do we find? Does each of these mismatches have the same outcome? No, it is not necessary. Some of the times you can have a recipient donor combination where there could be, many antigenic differences, and these are called taboo HLA mismatches. They would lead to an antibody formation. However, if there are few antigenic mismatches within the HLA genomic sequence, those are called as acceptable HLA mismatches, and these grafts can still work better. So some regions of the HLA are important, not necessarily just the HLA antigen. There are certain regions, each HLA antigen would have a small configuration of polymorphic residues called eplets. There could be antigenicity due to this, or there could be immunogenicity, and here we are concerned on the immunogenicity, and that would tell us whether that's an acceptable mismatch or an unacceptable mismatch. And this figure very clearly says that if there are self-epitopes, so if we do a cross-reactive cross-match testing between these individuals, if there are matching for the self-epitopes and then there are no unacceptable epitopes, so there would be no positive cross-match. But when there are unacceptable epitopes, for example, this case and this case, the cross-match would be positive. So we in the laboratories are able to predict virtually without actually performing a cross-match in the lab, depending on these acceptable mismatches, whether this transplant should go on. In the middle of the night, if we have the information, we know what is the sensitization status, whether there are antibodies or not, whether there are acceptable or unacceptable mismatches, and we can let the transplant go ahead in this scenario. And there are softwares to tell us that certain mismatches would have no eplet load and they are safer to go for transplant, whereas some other HLA mismatches between two can lead to a huge eplet load, and this can be detrimental for the graft. Again, there are certain HLA alleles for which there is a permissive, I mean that they are acceptable mismatches. There is one SNP in the DPB molecule in the 3' UTR. So presence of one SNP, the GG, leads to higher expression of this DP molecule. And if these DP molecules, if we have a recipient donor combination where these you see in red, the high expressing DP molecules, and if these recipient donor combinations undergo a transplant, there is a very high GVHD response. If there are low expressing molecules matching or mismatching, the GVHD probability is lower. If there is a high expressing recipient, but there are no mismatches, then also the little bit of GVHD chances are higher. However, when these is the recipient has very high and there's a mismatch with the donor, these are not permissive. So there are certain permissive DPB mismatches, and there are algorithms wherein we can feed the data and get the information that whether they, how much they will impact the graft. And here is data indicating that such mismatches, which are not permissive, higher acute GVHD probability is observed. Again, when there are malignant transplants, we need to look into the NK cell alloactivity, and there, I'll finish in a couple of slides. The NK cell activity, as we heard in the morning, the NK cells have inhibitory and activatory receptors, and the ligands for the NK cells are HLA. So in malignant transplants, we want a good NK cell alloreactivity because then the malignant cells can be killed. We want a graft versus leukemia effect, and this graft versus leukemia effect also balances the graft versus host effect. So these NK-KIR, the KIR-HLA interactions can also help lead to tolerance, and also they have a role to play in responsiveness to pathogens. But in a transplant setting, we do not want a scenario where the NK-KIR combination between the recipient and donor leads to a milieu that is promoting the graft versus host disease. We would prefer the kind of combination in HLA and K of the recipient and donor so that we have a protective effect from graft versus host disease. So, and these combinations do affect the acute graft versus host disease, the chronic graft versus host disease, and sometimes also the relapse of the disease. So these immune phenomena need to be taken care. And that's my last slide, that we also, when we select the donor, we look at what are the acceptable mismatches. The non-inherited maternal antigens, if there is a mismatch towards the non-inherited maternal HLA genes, that is an acceptable donor when we do not have an HLA identical donor in the family, because the non-inherited maternal antigens lead to tolerance, and the patient and the sibling would still have tolerance to those antigens which are although mismatched and not inherited from the mother. So in this modern era, of immunosuppression, we would say that is the HLA typing important or not? Yes, it is. We get a better graft survival, lower frequency of antibodies, require lesser doses of immunosuppression, and then side effects are lower. And then there are low grades of sensitization, which means that a retransplant if needed, and also the overall survival is good. At the end, I would like to thank all of you and the funding agencies who let us develop all of these technologies and perform better monitoring of the transplant. These are my lab colleagues and my PhD students who help us develop all these methodologies and set up for the patient care, and also my family who permits me to travel all the way and share my experiences. Without them, I wouldn't have been in the lab for a long time and also joining the meetings and interacting. It gives me an opportunity to learn a lot also when I come to the scientific conferences. Thank you so much.
Thank you, ma'am. As always, it was informative. So we can take one or two questions.
Dr. Uma, a very nice and interesting presentation. I want to have a clarification. See, in some centers, they are going for swapping organs, donor swapping. Are you also practicing donor swapping in your centers? In that case?
Not in AIMS. There are plans to develop that.
Okay.
That we have a lot of patient and donors where recipient donor pairs where their respective family donor is not suitable. So there is a planning that people are trying to swap the organ from one recipient donor pair to another. So they exchange the donor, and then the graft is expected to be better.
Definitely, chance of getting HLA matching is not there, right?
Yeah.
And also the organ needs to be maintained based on the immunosuppression.
No. So in this case, when there is a swap donor transplant to be done, the major thing that they look into is the antibodies. If the recipient doesn't have a pre-formed antibodies to the donor that they are going to get because of the swap, then the transplant can go ahead with the immunosuppression.
DSA need to be done.
Yes, so the single antigen, so we need to identify antibody to every specific HLA allele. So if once we do that and we know that this is not against the donor that is going to be given to this recipient, then the transplant can. And such a transplant can be not only between two pairs of recipient donor, it can be a multiple chain, 4, 5, 6 pairs who can undergo such an exchange program.
Simultaneously.
So Gujarat IKDRC is doing, and they do in a week, sometimes 10 swap transplants so that 10 patients can benefit, although their own donor is not beneficial from them.
Okay, thank you.
Yes.
Yeah, thank you for the talk. I was just wondering, as you explained very well, that the probability of having a good match is relatively low. So in that case, I was wondering, are the alternative approach has it been tried? For instance, that if assuming that you already know who the donor is going to be, if you take its HLA, antigens, and you sensitize the Tregs of the recipient, let's say in ex vivo or something, and infuse back those Tregs, so you would preemptively have a large amount of Tregs specific to the donor antigens while the transplantation is done, and then with immunosuppression, perhaps the organ acceptance will be higher despite the HLA not being matched, for example, or some such approach.
Absolutely. Very few centers around the globe are practicing this. It's already has been tried, and there are some centers who do the harvesting of the cells. Expanding Treg also takes time, but then when they do the graft, they give a prior dose of the Tregs, and then once the tolerance is developed, then they do the graft. And in bone marrow, they add the Tregs to the inoculum of the harvested stem cells, or they deplete the donor T cells and then take the graft. So these kind of modalities are being practiced, but it's not a routine practice in most centers. Very limited centers practice that. India or globally. Stanford is the one that developed this protocol.
I think we can discuss afterwards. So, ma'am, again, many congratulations. And
thank you.
Thank you, Dr. Uma Kanga, for such an amazing talk. Next, we have another recorded lecture, a historical talk by Dr. Pragya Yadav, Scientist F in IV Pune, entitled as The Journey of Indigenous Vaccine in COVID-19 Pandemic. I would request our chair, Professor Upendra Nongthamba from IASC, to now take over and guide us through the session.
Hello. Yeah, thank you very much. It's a pleasure to introduce today's speaker at this session. Pragya Yadav, Dr. Pragya Yadav is got a PhD from NIV in Pune itself and Pune University, and she is working now as a scientist F there. And she was one of the first who identified the COVID-19 patients in India. And she was one of the first who made the indigenous COVID vaccine, Covaxin, from India itself. Without telling much, I would like her talk to be played here. She is not keeping well, so the talk will be in recorded form.
And today we are here in the historic session of this IMMUNOCON 2024 meeting. And our COVID warrior forefront, Pragya D. Yadav, will be delivering her talk, The Journey of Indigenous Vaccine in COVID-19 Pandemic. To introduce Dr. Pragya is an honor, and I feel privileged because I belong to the same organization as she is, Indian Council of Medical Research. She's currently working as Director in Charge, National Institute of One Health, Nagpur, and Scientist F, and leading Asia's first biosafety level 4 laboratory at ICMR, NIV, Pune, India. She has obtained PhD in virology from Savitribai Phule Pune University, Pune, in 2004, and joined as a scientist in ICMR NIV Pune. She had made significant contributions in the field of virology and biocontainment science and translational research. She is well known for her work during the COVID-19 pandemic. She made sincere efforts in quick isolation of SARS-CoV-2 virus from India, which was utilized by Bharat Biotech International Limited to generate our indigenous vaccine, Covaxin, that actually saved not only Indian lives, but also Indian economy. Over to you, Dr. Pragya. We are all very eager to listen to the story. Thank you.
Thank you, Dr. Taruna, for kind introduction. I would like to thank Dr. Dipankar Nandi and all the organizing team members of IMMUNOCON for inviting me. I know that this IMMUNOCON has been a great success, and I congratulate for their continuous efforts for bringing so many lectures in these 4 days program. So, as we all know, the pandemics has been known in the past, and there were many pandemics which caused a large number of morbidity and mortality. And it was thought that every century there will be pandemic which can lead to major disaster. If we talk about 21st century, we could see emergence of many new viral diseases and re-emergence of many diseases. The re-emergence of diseases had significant decline in the past, but incidence of diseases has reappeared, and they are called as re-emerging infectious disease. With the history of many influenza pandemic in the past, everybody thought that the next pandemic would be influenza virus. But for a surprising, coronavirus emerged as a next pandemic in 21st century. This SARS coronavirus was called initially novel coronavirus, and decoded by China. Very quickly, they sequenced and then they came up with the genome of the virus. The world was waiting what happening to the rest of the part, and this virus very fast arrived to India in the form of 3 travelers who traveled from Wuhan, China to India. And at the end of the first month of January, in the 4th week of January, we could detect 3 cases, and quickly it was a story that novel coronavirus is spreading in many parts of the world. In the March, it was declared as a international health emergency, and very quickly we were in a situation of international lockdown. Large number of Indian people got stranded in Italy, in Iran, and there was many challenges which emerged with COVID pandemic, which nobody realized when the virus initially emerged from China. Then there was another concern which happened, that there was the virus started mutating very fast, and it caused a lot of mutation because of the passage from one human to another, and it was called as variant of concern. Many variant of concern emerged, and some of the very important was alpha, beta, gamma, delta, omicron, and they challenge not only to the different plannings of worldwide that how to deal with this pandemic, but also all the efforts to halt this progress of the disease. In India, we have 3 main waves. The first wave in 2020, second in 21, and then third wave in 22. The second wave, which was caused by Delta, caused the maximum loss of human lives and derail our entire system. Not only India, but there were many countries who got badly affected, and there was a lot of fear with the COVID pandemic as well. Initially, when a pandemic started, there were many challenges. The first was the diagnosis, but people quickly realized that we required some vaccine just so that we can control not only the spread but also the deaths which is happening with the pandemic. During the lockdown, there was no vaccine availability, and we were lacking infrastructure, high risk of infections, and the people who are working not only in the hospital but laboratory, they have constant risk of getting infected. We were not having reagents and PPE which was required to work in the hospital or in the laboratories. There were major challenges of clinical trials protocols, animal models, lack of diagnostic reagent. And once we went through and we started working on vaccine, preclinical models, then pathogenicity of VOCs, the virus isolation, and then once the vaccine got ready and given to human being, then impact on vaccine effectiveness was another challenge. So this COVID journey was full of challenges, and it gave everyday challenges to scientists working on this, and that was an emerging variant of concern. Still, virus is mutating, and recently we have seen emergence of KP3 and other recombinant variant of SARS coronavirus. As such, the Apex Laboratory of Indian Council of Medical Research, National Institute of Virology, which always in forefront of virological diseases, started planning not only the testing but also quickly training VRDL facilities, shipping RT-PCRs, and then also work with the Mission Udan for bringing the people from abroad to India who got standard, making indigenous antibody screening and validation of real-time PCR, ELISA, and many such reagents made in India and also which was imported from other country. Isolation of virus was a major initially concern. Then we started antiviral testing, genomic surveillance. We started working on any animal models so we can establish the preclinical trial methods, and then providing live and inactivated virus to industry for a different kind of uses which was required during the pandemic. There was another initiative which was taken very quickly, sewage water testing. With this, when we started thinking about the vaccine, we need to develop a quick zero diagnostic, and then different methods which was required to develop for vaccine, plaque reduction neutralization method, MNT, animal models, and we also need to develop polyclonal antibodies, and collaborating with industry on different products with the vaccine, We also work on equine sera and many other therapeutics measures which could be used for treatment during this pandemic. We worked with Zydus cadila as well for the DNA virus. But this was not all. The virus mutated further and caused complication to the people. And it also created the new aspect of mucormycosis, reinfections, breakthrough. Then after the 2 doses of vaccine given to everybody, then there was a challenge of breakthrough due to Omicron variant. And there was a discussion started, should we do to booster and which variant vaccine should be given to booster. If there was a questions raised on effectiveness of Covaxin, Covishield, and all the vaccine which was given that time, genomic surveillance, and then how these variants were impacting the assessment of the vaccine. In March 22nd, when PM Modi announced the Janta curfew followed by lockdown, it affected the life of everyone in country. There were many steps which was taken very quickly by government of India to control this disease to spread, and that was the first. All the people were coming to the country, do the self quarantine. Then there was implementation of Epidemic Disease Act, and so on. There were many online apps like Arun Setu was launched very quickly to learn about the infectious patients. These exercise we could learn that we have to not only develop but apply innovative technology to improve efficiency, focus on detection and diagnosis, and also improve and scale up health education for the public. In this crisis, quickly, we only develop the test, but we could increase the number of testing network, and that is more than 3,000 labs across the country, and they started doing daily testing at very high scale. Phase-wise manner it increased, and we could reach to the very, very small cities and district level laboratory. We could make many different diagnostic assays make in India assays, and finally we focus on vaccines. And there were many vaccines which was not only manufactured in India but also developed and created a new kind of standard to look forward in the country. The number of VRDL facilities was increased, clinical trial capacity was enhanced, and then we could establish INSACOP genomic surveillance across different ministries using different laboratories, and which also set up a new kind of understanding that we all the stakeholders has to be work together. Many digital apps was very useful, including COVID for implementation of vaccine program. In the COVID, first time science could demonstrate but that we can maintain the quality, we can follow the process, but we can do a production of very quick vaccine, which can be made in 8 or 9 months. Nobody thought that vaccine can be made so fast. Pandemic affected livelihood, human health, governance and security, social system, and the most important economic system across. In this process, Government of India used 2 approaches. The first was to leverage India's scientific prowess by research and development, product development, clinical trials, in-country supply, global supply, vaccine diplomacy, and that outcome of these are the Covaxin and Zyco DNA vaccine. The approach to was leverage international science by technology transfer, augment productions, in-country supply, global supply, COVAX facility, vaccine diplomacy, and the outcome of these, there were many vaccines which could be produced in India, and then not only it supported countries' need, but also under the vaccine method, more than 100 countries got benefit of it. Then it came to the developing indigenous vaccine. The first thing was to isolate a virus, to develop different test assays, but also the vaccine. As in an inactivated vaccine has set up a very good safety and efficacy, and it was in the beginning when we were not knowing much about the virus, it seems to be very easy and deliver task considered to be. But before we do that, we need to isolate the virus, and we have to develop many assays and establish and publish quickly to build the trust that we are doing right science and we are going in the right path. Quickly, ICMR and Bharat Biotech entered into a MOA, and we could not only do the genomic sequencing, growth kinetics, virus titration, genetic stability, and electron microscopy and product analysis that help us to understand that we should require 3 vaccine initial compounds, which was used with L-gel IMDG and also with L-gel. 2 doses of L-gel IMDG was initially formulated, and third was only L-gel. And these 3 products were tried in 3 different small animal models, and we quickly could see not only the safety but also the very good neutralizing antibodies for 3 and 6 microgram with L-gel IMDG concentration of SARS coronavirus candidate. These 3 candidate and placebo was taken through the hamster model, which was considered and established as a very good model to understand SARS coronavirus. We could give 3 doses of the vaccine to hamster and then challenge with the virus. And these studies lead to understand that those group of hamster which was given vaccine, they did not develop disease and they were protected against SARS coronavirus in compared to the control group where the virus was is spread across the lung, and we could see high viremia in the hamster model. The next one, that was a time when non-human primate challenge model was compulsory to get a phase 3 trial. Quickly, we set up the protocol, and we provided 2 doses of vaccines to non-human primates at 0 and 14 days. And on 28 days, we challenged these monkeys with the live virus. And we studied in detail the clinical parameters, and we observed through the X-ray and different criteria which we derived for the vaccine monitoring. And we could see that those monkeys whom had a vaccine, the clinical sign resolves very quickly. No radiographic abnormalities were detected in these animals. We could observe the SARS coronavirus antibody response in those animals where they got the vaccine, and post-challenge this response was enhanced in compared to the control group. We could also see the neutralizing antibody response generated after the second dose of vaccine, and which was constantly increasing post-challenge of the virus. We monitor the viral genomic and subgenomic RNA in respiratory tract, including the nasal, throat, and in the bowel fluid of the monkeys. And we could see that those monkeys who got vaccine, they clear the virus very quickly, not only for genomic RNA, but also for the subgenomic RNA. Quickly, altogether, this data put to come up in the results that those monkeys who got vaccination, they could quickly, the virus in the respiratory tract, in the lung tissues of different lobes disappear, but in the control, it was existing for a very long time and for many days. We could also look at the viral RNA in extrapulmonary organ, and similar kind of observation we could see. Gross pathology and also histology, histochemistry could quickly show that lung showing extensive involvement of all the lung lobes affected with virus in the case of the control group, but in the immunized group, we could see the clearance very quickly. Histopathology and immunochemistry further and make us understand that none of the vaccinated groups showed any feature of pneumonia or viral antigen detections in all 3 vaccine candidates in compared to the control group. Based on this, we could conclude that among the candidates, 3 and 6 microgram antigen with IMDG immunized animals showed better neutralizing antibody response, faster clearance, and no clinical or radiological evidence of disease. With this, PM Avim launched the program in July 22 for establishing a new institute called as National Institute of One Health in Nagpur to serve as the institutional anchor for coordination of research and development activities of the One Health mission under the guidance of a principal scientific advisor, where 13 different ministries will come together to work on pandemic preparedness and preparing the country for the future response in a better manner. With this, I would like to thank once again to IMMUNOCON to invite me. And I cannot end this presentation before thanking my team, mainly BSL4 team, who work in very with the integrity and hard work to achieve the task of delivering many COVID products and also diagnosis and many aspects. With this, I would like to thank many scientists of NIV Pune, ICMR, armed forces who worked during the pandemic to support us. I would like to thank Bharat Biotech for very great support and sharing all the knowledge, know-how to develop a vaccine, which nobody thought that India can do it. So this become a new set of examples that private partners and academia can work together and develop something very quickly. But this should not be happening only for the emergency. It should be a pattern in the routine. And when we practice in the routine, then only when the pandemic or outbreak happen, we will able to quickly respond. I would like to thank the former Director General Dr. Bhargav sir, Dr. Nivedita Gupta, ECD Chief, Dr. Ganga Ketkar sir, former ECD Chief, Dr. Priya Abraham, my colleague Dr. Gajanan Gurraj, Dr. Varsha, my colleagues Dr. Reema, Anita, and Sri Lakshmi, who worked with a lot of zeal and passion during the pandemic to give a lot of scientific answers to the country. With this, I would like to thank all the healthcare workers, NIV and ICMR colleagues, and the people who were engaged in providing different approvals. It start from CPCSC to PCCF and the clinical trials, preclinical trials approvals to fast track the study. With this, I would like to thank all of you for listening. And I'm going to miss meeting you personally, but hope to see you in future sometime soon. Thank you very much.
Thank you very much, Dr. Pragya, for an excellent historical perspective to this amazing achievement that India made. And we express our sincere gratitude to you and your team at NIV Pune, as well as at ICMR headquarters, and all the industry personnel also who collaborated with us to bring out this venture, and the regulatory authorities who supported you to develop in a record 1 year time and then release it for use during the pandemic times. So heartiest congratulations to you on getting the Shanti Swarup Bhatnagar Award, and we wish you many more such laurels to come, and we wish you very good luck for all the One Health and pandemic preparedness exercises that you are doing. And the recent one I think we did was very successful in Jaipur, where we found out that actually we are prepared. Thank you so much.
Thank you, Dr. Tarna.
To all our delegates in the conference, and
due to physical absence, we wouldn't have any questions answer here. Okay, then let's thanks Pragya Yadav for her excellent presentation. Thank you very much. Yeah.
Professor Upendra Nangtamba, please come on stage for a group picture. We will now take a short break for tea. Kindly reconvene by 5:50 as we continue with the next exciting sessions. Enjoy your break. After tea break, we will be having 3 parallel sessions happening simultaneously in different halls. We request everyone to kindly go through the schedule for the parallel session. Thank you. Enjoy your break.
Everyone, I would like to invite Professor Nagasuma Chandra from Department of Biochemistry, IISc, for the next talk. I would also like to introduce the chair, Professor Girdhari Lal, to kindly chair this session.
All right, it's my privilege to introduce Dr. Nagasuma Chandra, and I can tell you that I have to learn something bioinformatics, I have to go to her. So we understand the biology, physics of the molecule, and she is the person who will tell you the physics behind the molecule. So she is going to, she is a professor at biochemistry department, and she is going to discuss about today in terms of multi-scale modeling of the pathogens recognition and immune response. So with this word, I invite Dr. Nagasuma Chandra to deliver her talk.
Thank you. Am I audible? Thank you very much, Professor Giridhari, for the kind introduction. And thank you, Deepankar. It's a fantastic meeting. Thank you also for rescheduling my talk. I had some personal emergency and had to reschedule that from the next session, actually. And thank you for whoever has adjusted for their talks with mine. So today I'm going to talk about multi-scale modeling of pathogen recognition and immune response, especially the CD8 T-cell response. So I must warn that it's very different from all the other talks that you may have heard, because this comes in from a data perspective. And I'm not going to talk about the algorithm, but I'm going to talk about how we have used it and what insights we can gain from that. So despite extensive amount of work on diseases like influenza H1N1 and the latest COVID-19, which don't need introduction to this audience surely, there are some questions that remain open because they are very large questions, difficult to answer questions, and so on. Very broad questions such as why do some strains cause outbreaks but not all? What in the strain is actually making the difference? And likewise, why are individuals differently susceptible to the disease? So we can't really define a pathogen without its counterpart, which is the host. So host and pathogen go hand in hand. So while we broadly say human host and this virus or whatever, even a bacterium as a pathogen, we have to go into the nuanced differences and say exactly which strain and what in this particular thing defines it as a pathogen, which is what I'm going to address today from one angle alone, that is that of CD8 epitope recognition. So there's a lot of complexity in understanding CD8 epitope recognition. That's because, so I'm not sure if I have the laser pointer here. This is the one, I presume. Yeah, okay. So we have to look at multiple aspects. I'll just run through this in the interest of time. So we need to look at multiple HLA alleles, and the previous talks have given such a wonderful introduction to that, so I don't need to talk about that. There are thousands of alleles, obviously, HLA alleles in the A locus, in the B, I'm talking only of the class 1, A, B, C, and loci. There are thousands of alleles in each of those, right? And that leads to HLA genotype diversity in an individual, which we'll come to in a moment, and variation in antigen recognition across individuals. So that actually brings in a huge combinatorial problem of how variation can be, how antigen recognition can be varied among individuals, which is what we'll see, and how does that manifest across populations is what we want to look at. And as if that's not enough, we have variants in the strain as well, especially in viruses like H1N1 or COVID. We saw a rapidly changing epitopic regions and so on. And that contributes to additional layer of complexity. So how do we take in all of these things and then make some sense out of it for the questions that I posed? Because there's a lot of data. Data is huge. Data of various kinds, clinical data, now basic bio data, immunology data. Now you have data coming from high throughput techniques such as various types of omics and so on and so forth. So where do we actually begin, right? In fact, this definitely qualifies as big data that we have. All the 4 V's that are that define the big data are satisfied with the data that we have, especially for COVID or even H1N1 for that matter. So So we had been working on H1N1 before the COVID outbreak, and we were able to repurpose some of our algorithms or computational modeling concepts into studying COVID as well. So what are the types of data that I'm going to focus on for CD8 response? So we understand HLA sequences, like class 1 allele sequences, and what does that mean for their recognition function? We understand that better from a three-dimensional structural point of view, and so antigen recognition modes and so on. And HLA alleles in individuals can be now sequenced or can be determined serologically or with whatever method that is available. And there's a lot of data, and a very good databases called IMGT, where HLA distribution across ethnicities and populations are available. And of course, there's a whole lot of data on the disease spread across geographies where ethnicities can be mapped largely, and so on. And there's also data on CD8 response to COVID-19, more from a immunology perspective. Like I said, omics and other mechanistic data are also available. So to address all of these, okay, one way to come out of this jungle of data and make some sense out of it is to use a multi-scale approach, because a single level will not fit all. You can't answer all questions. You can't even fathom how to go forward with that data unless you break it down into a hierarchy. And that hierarchy, the best hierarchy to use is the hierarchy of biological systems themselves. So look at the sequences as the starting point for anything, like protein molecules as a starting point. In this case, HLA molecules on one side for the host, and on the pathogen, whatever is giving rise to the epitope for recognizing the HLA molecules, that will be the molecule out here. So we have sequences and structures, we have pathways, we have various types of interactions, a cell and a host pathogen interactions to take it all the way forward. In a nutshell, this is like a middle out approach. You can start either from a bottom up or a top down. Most of the theoretical immunology that we have seen in the past decades would come from a top-down approach. Most of COVID-19 projection models have been from a top-down, where there are epidemiological models using a small set of equations which capture the, of course, I mean, all models have their merits and demerits. So this one was useful to say whether lockdown should happen or not happen, and will it spread, will this become an epidemic or not, and so on. That was the kind of projections that people had. But that loses out on the detail which is required for a more mechanistic understanding and more, rational intervention at various things. I won't read out what's on the right-hand side. These are examples of different types of modeling methods that one can use at each level, different level. So we started with a COVID-19 data set that we were able to put together along with the Victoria Hospital and Bangalore Medical College colleagues. So we were it's a small cohort, but we were looking at a healthy group where they tested COVID-19 positive. This was during the peak of COVID. And we had an asymptomatic group. We had a mild, moderate, severe, and I won't talk about the last one that was a co-infection. I'll leave that out for the time being. Of course, this is based on strict inclusion-exclusion criteria and all that. So what we did from these patients was to collect blood samples, and the blood samples were sequenced, not for the genome, but for the transcriptome. So we had transcriptomes, whole blood transcriptomes. The bulk RNA was extracted, and then subject libraries prepared as per standard methods, and then they were sequenced. So what did we gain out of that? So we were asking questions for a different project, but then that's useful as an input into what I'm talking about today. So we were looking at what actually differentiates. What is the tipping point to say when it is remaining as asymptomatic, or when it becomes mild, and or when it is severe, for example. So we looked at the gene expression data shown on the left-hand side here. This asymptomatic, mild, and moderate, severe, which we grouped into one because the distinction was not easy. Clinical distinction was not easy among them. And we saw a large number of differentially expressed genes which belong to various categories. But to understand these better, we've developed a pipeline where we contextualize a knowledge graph, like a protein-protein interaction network at a genome scale. These are methods that we've had in the laboratory for quite some time now, and we applied to several projects. So we used the same, and then we asked, this is like modeling a large city, like Bangalore, modeling the traffic patterns in a large city, and then asking what happens in this city when, let us say, the city is asymptomatic versus the city has severe infection or something of the kind. What is the difference between the two things? Where is the traffic flowing differently between the two conditions in the same city? Because the knowledge graph, which is essentially a protein-protein interaction network, is the same. We contextualize it using specific patient-specific or cohort-specific transcriptomic data to come up with these answers. And then after contextualizing, we have to mine using graph theory methods very sensitively to look at the top layer of perturbations and so on and so forth, which is what we do. What we found is summarized on the right-hand side there. So we do see several things, of course, the innate immune system, but what we also see is the down-regulation of the CD8 or the HLA class 1 genes and downstream signaling of the recognition of these HLA class 1 molecules. So definitely the CD8 response is downregulated in the severe group, which is as compared to the asymptomatic group, and it's somewhere in between in the mild severe. So like I said, we had been working on this for influenza for quite some time. My former student, Sumanta Mukherjee, who now heads a group in IBM research, he came from an AI computational data science perspective, and Vishal Rao, who's currently in Mount Sinai, is a PhD student. So Sumanto had established several of these for H1N1 influenza, basically to characterize allele diversity, to look at genome-wide epitope prediction, characterizing host heterogeneity, and modeling CTL response in different populations, and also simulating infection spread over a model population, which I'm not going to touch upon today, but I will just quickly tell you these things for COVID, and I'll bring in similarities with H1N1 wherever it's possible. Of course, several papers have come out of the lab on these techniques. So our starting point was to look at three-dimensional sequences and three-dimensional structures of HLA molecules. What you're seeing are those here. Only a portion which is relevant for recognizing the antigen is shown here in red in this thing. What you can see as the stick model out there is the actual epitope or the antigen from a virus that is sitting inside this pocket, a nice, beautiful binding pocket that happens in this HLA molecule. And alleles differ from each other only in this red region, which is why one allele is different from the other. And if you understand what the differences are, you're able to rationalize and then even predict whether a given epitope, whether a peptide is an epitope for this allele or not. Right? That is the basis for many of our predictions, which is very, very well established today. Now, there are many experimental studies have happened to prove it, and there are several databases, several algorithms, and the latest algorithms obviously use a combination of the physics behind this recognition, the biophysics, as well as AI application to handle large quantity of data. And that picture on the right-hand side in black that you see shows that the presentation of these epitopes is very similar in all alleles, in the sense these epitopes, which are 9 amino acid peptides, are basically stretched out. They don't, it doesn't really depend on the native structures on the proteins, whether it's a spike protein or ORF3A or 3B. It doesn't really matter because they're pulled out. They're stretched, elongated, and pushed into this cavity. So the principles of recognition are somewhat similar out there. And what you see in the diagram below is a sequence logo, which tells you what are the alleles in this recognition site, in this red region that you see here, on the HLA alleles. You put all the HLA alleles together, do a sequence alignment informed by structure. So it's a structure-based sequence alignment that you see. And the higher the height of the letter, the more conserved it is. So only those that vary among alleles are shown as multiple letters in a given column. So you, in a nutshell, you're able to identify what contributes to the basic functionality of an HLA molecule binding to your peptide and what contributes to specificity differences among them. So this forms a fundamental layer. And of course, like I said, many other computational tricks and algorithms have to be in place to handle this variety of data. Then we asked a simple question at that time, quite some time ago, to ask, can we group all of these together? Right? All these binding sites, based on binding sites, can we come up with some sort of a grouping scheme? Now, grouping scheme is very well known for HLA alleles. People use serology, serotyping. That is how the classification has come. If you do it purely from a data perspective, agnostic of the serotyping, you get somewhat very similar, but you get a refined typing. And the colors, the levels actually tell you how much hierarchically you need to go in terms of comparisons. I won't get into the algorithm, like I said. But by and large, S1, S2 are the super types that are well known to immunologists. So it works quite well. but you do see even similarities across the loci. A alleles, some A alleles look very similar to B alleles in their capability, and C alleles as well. The shapes are the alleles. The color is actually programmatically generated based on the extent of agreement with serological data out here. This figure alone took like months of work, right? Okay, to generate. Anyway. So now we know that pathogenic viruses are engaged in an arms race with their host. That's why I said host and pathogen can't exist without the other. Evolving rapidly, often under negative selection. So this is a well-known concept. Now from a CD8 perspective, we wanted to understand, is it really contributing to this pressure, to the selection pressure? So we know, for example, if you look at the middle diagram of here, these are a phylogenetic tree of all strains of COVID virus that we see up to that date, at least, sometime in 22. Oh, okay. So I'll finish very quickly. So there's a ladder-like phylogeny that you see here. And then the plot that you see on the right-hand side shows you that the epitopic distances are evolving much more, epitopic regions are evolving much more rapidly than the rest of the amino acid sequence out there, indicating that CD8 epitopes are under immune pressure. Otherwise, they should have been a diagonal line. Now they are on the left-hand side. The red dots that you see there are showing that. Okay, couple of minutes only I will take. So now we use this as a starting point to identify pressured epitopes. So which are the actual pressured epitopes in SARS-CoV-2, the Wuhan strain? And we mapped it back to the cohort that I showed you to show that the epitope strength derived from multiple methods is actually much lower in the severe cohort as compared to the asymptomatic cohort. So we were actually seeing that there was a difference not only from the epitope strength but also from downstream signaling part. We were able to get a pressured protective epitopes list. And from a big list of possible epitopes, we narrowed down onto 6, which go according to these. These are the sequences and those are the proteins where they come from. And those are the alleles that actually recognize that. Now, so the next thing was to move from HLA alleles to individual people and carry out what we call as response typing. So here it says this group, if you take all the people together with their respective HLA alleles and look at the epitopes, each group recognizes or sees the virus in a particular way. That is what you see here, irrespective of the individual HLA alleles, because there is correspondence in their functional ability, which is missed if you go only according to serotyping. Right? So this is what actually happens. I won't go through the numbers. I know I'm running out of time. So we have 6 alleles in each individual, and there's a lot of data, and this number of combinations is a mind-boggling number. So we carried out this response typing and then get various types of types. A large number of people can be grouped into a small number of groups. Each dot, each circle that you see here is a group of people. It's a type. It's a response type. And they recognize the virus in a particular way. And what we are actually seeing in influenza H1, we use this kind of a thing to say that this is pathogenic strains across ethnic groups here. Height of the bar tells you who has a good response, who has a low response, and that tells you directly the susceptibility. And that matches quite well with the epidemics that have come out. The picture for COVID-2 is different, but it's a similar kind of insight is what we will get. Right? Now we want to move from individuals to populations. So we had this algorithm set up. Yeah, so I think I will close very quickly. So this is like we group the population into low CTL and high CTL. And what you see here are individual alleles, and the colors tell you who is mounting a response and who is not mounting a response. I think I should close with this. So various insights from this work are we identify key HLA alleles, key epitopes, impact on individuals, effect on variants, and on populations and impact on disease spread. What I didn't show you, but what we have actually done, especially for H1N1, is to use this and do epidemiology spread models by incorporating genetic heterogeneity into what are known as SIR models. And that gives you a lot of further insights, but I'm completely out of time, so I will stop here and thank you for your attention.
Thank you. It's a wonderful talk. We can have 1 or 2 questions, quick, if any. Yeah.
Very nice and thought-provoking talk. It is well known that and is established by some groups that there is a co-evolution between the pathogens and the HLA alleles. And did you ever thought of calculating what happened during this COVID period? New HLA alleles, possibilities of identification?
Okay, the time scale is an interesting question, but time scales are very different. Humans don't evolve as fast as the viruses, obviously. So in the time scale that we are seeing from COVID initiation to now, unlikely to have any change on the HLA side. But it is the pathogen side which will change very rapidly.
Evolving. Okay. Thank you.
Yes.
Very nice talk. Thank you very much. So my question is purely clinical. We know that there are several comorbidities which influence the ultimate outcome of COVID, whether they will remain symptomatic or die or the severity. So have you kept that into consideration while generating your models? Like, have you looked at what percent, depending on the age, gender, the comorbidities, etc.?
Yeah. Comorbidities we did not include. We had we had a separate group for that. So none of these had any, at least, recognized comorbidities.
Okay. What about smoking? Did you look into that?
Sorry, what?
Smoking.
Smoking. Yeah, I think that was also recorded as one of the phenotypic characteristics.
All right. Yeah.
Thank you. Thank you very much.
Thank you, Dr. Nagasumachandra, for a fantastic talk.
Thank you.
Thank you. Now we'll move on to the next session. This was just a quick thing. So I think next one is clinical immunology. I'll hand over to her.
Sir, I would request you and ma'am to kindly come up on the dais for a group picture. Next, we will be having panel sessions in different halls. For those of you staying in the main hall, we have an exciting session lined up titled as Clinical Immunology. I would like to invite our session chairs, Dr. S. Chandrasekhara from Chandray and Professor Swaminathan from Medical School IISc to kindly take over and lead the discussions.
Good evening. Welcome to the session on clinical immunology. So the first speaker is Professor Krishnamohan Atmakuri. He completed his PhD at MKU, and his work he's going to present today on neonatal sepsis and the challenges it imposes and potential solutions.
Thank you very much. Good evening. It's first I should thank Dipankar for a great meeting. And this is my first IMMUNOCON because I'm not technically an immunologist. I'm a bacteriologist. I do collaborate with a lot of people here in terms of the immunology work so as to make my life easier. That doesn't mean that bacteriology is terrible. Microbiology is very good as immunology. It's nice to actually see so many people still in the auditorium on a Saturday, especially in the evening, when you should be in the theaters, possibly. Okay, so my lab is looking at a few things in terms of the pathogen. If you will, you can call it as a toolkit, which every pathogen possesses. It equips itself to answer, in order to subjugate the host cellular processes.
So we're trying to understand what this toolkit is, when basically they utilize this toolkit, how they basically use this in order to modulate the cellular processes, where actually this happens, what is the spatial and the temporal kind of secretion and regulation that happens. In fact, we also look at what are the kind of molecular machines which basically are involved in terms of utilization, rather the pathogen utilizes in order to secrete something which it encodes. And the question is, of course, why and how the host is able to, control this. So we are using two disease models, the tuberculosis and the sepsis. In general, we do a lot of host pathogen interactions. And of course, there are a few fallouts which basically come out of these particular studies. And either we try to use them as either therapeutic targets or possible biomarkers or vaccine antigens and subunit candidates. It depends upon the particular candidate that we are looking at. So I'm not going to discuss anything about the TB part, but in the last 3-4 years, my lab has kind of shifted gears towards more of the TB, to more of the sepsis, depending on this moving of going from TB or sepsis depends upon the amount of the funding you get and the kind of data you get. But in terms of the definition of the sepsis, because wherever I go and present it, not many people know what is sepsis. The definition of the sepsis per se has changed over several years. And in 2016, approximately 35 to 40 clinicians and immunologists and bacteriologists sat together to come up with this latest definition, which is the life-threatening organ dysfunction syndrome, because this is not a particular set of symptoms that you see. Depending upon what pathogen, you could change a little bit of the symptoms, sort of a syndrome, and it is because of a dysregulated host response. I don't say host immune response, it's a host response to an underlying infection. So infection is certainly there, and it is in the question of how much is the dysregulation in each of these individual hosts. The sepsis basically can infect, rather the infection of the pathogen to different organs can trigger sepsis. Not every infection is leading to sepsis. There are a lot of pathogens which cause sepsis. Most of them are opportunistic. In the Indian context, there are quite a few escape pathogens, including the Acinetobacter and the Klebsiella. They kind of fight with each other in order to be the number one in every particular season that we see the sepsis. Quite a few viruses also can do this. In fact, some of the ICU-based deaths which happen because of either TB, dengue, SARS-CoV-2 influenza, basically are all classified into sepsis. So it also happens that when somebody goes into the clinic and the doctor basically suspects of sepsis, then they end up giving a broad spectrum antibiotic. There is a possibility of some of the fungal organisms taking over and continuing to trigger the sepsis. The sepsis burden, surprisingly, is the most highest of any infectious disease you can think of. It's because you have multiple pathogens which can trigger sepsis. So approximately 50 million cases. It is thought that it is underreported, and possibly 20% of all global deaths are accounted because of sepsis. In India, the death accounts could be as high as 35%. Children occupy almost 50% of this. In India, almost 40% of all deaths is because of sepsis. So the funniest thing about this is the disparity in terms of 85% of all these cases happen in the LMICs. So indicating there is a large amount of hygienic issues, MDR, XDR issues, which possibly are triggering this particular kind of a disparity. It's predicted that if ignored by 2050, 50 million deaths could happen. This possibly is not an overestimate simply because of the emerging AMR problem. So largely, the clinicians, in terms of the sepsis diagnosis, look at a lot of hematological parameters, the biochemical, the physiological, including a large plethora of symptoms. And that's one of the reasons why it is so challenging to basically suspect sepsis. And there are a lot of clinical symptoms which are taken into consideration. And also there are a couple of biomarkers which are being utilized in most of the clinics, the procalcitonin, the C-reactive protein. None of these are great specific biomarkers for sepsis. So the current gold standard, at least in this country and the LMICs, is that you do a blood culturing whenever anybody is suspected of sepsis, adults, neonates, whatever. And you do the blood culturing, then you also do the antibiotic cards, and it takes anywhere between 2 to 7 days before anybody is able to go back and tell the clinician that this is the pathogen, this is the kind of resistance the pathogen carries. But neonates are not going to be waiting for 2 to 7 days in order to get that. And most of these neonates can pass away within 3 to 5 days. So it's a huge challenge. So what is done by the clinicians is simply give a broad spectrum antibiotic and basically cross your fingers to see whether this would somehow adjust to survival. So what is known also is some of these terminologies are important because that's the reason I'm trying to show you all this. So 20% of all the neonates in the LMIC conditions, LMIC hospital, are blood culture positive, which means that you take a ml of blood from these neonates, you culture them, and you end up seeing the pathogen. What is normally seen is that while 20% of them culture, 80% of these cultures don't show you any pathogen. It's also because of the media being not supportive of the growth, but it basically raises the question that you don't know what the pathogen is, whether it is bacterial, viral, fungal, whatever it is, and what is the kind of therapeutic strategy one has. And so sepsis diagnosis is more of a rule out rather than actually a rule in. That's a big, big challenge. And in my collaborators from AIMS in 2010 to 2015 came up with the surveillance of the organisms within the NCR region and found two things. One is the early onset sepsis, which basically is from the day of the birth till 3 days. So approximately 60% of these neonates who are blood culture positive can pass away within these 3 days. While you get approximately 90% of these neonates who are in the NICUs who get 90% of the sepsis cases. These are primarily the preterm neonates. So the neonates who are born with a gestation period of 28 to 34 to 36 weeks. They're approximately less than 1.5 kilos and very, very tiny with a very poor immune competence. So there are a lot of pathogens which basically cause the late onset sepsis. So the late onset sepsis is thought to be coming from the hospital. Classically, this is dogma. So whatever is coming as a late onset sepsis, that is from 3 days to 10 days or 20 days or 28 days, is all primarily because of the hospital driven, while what is coming from the early onset sepsis is primarily coming from the mother. But in the Indian context, we don't see any difference. We see almost the same pathogens causing both the early onset and the late onset sepsis. And of course, these pathogens come with a plethora of cassettes, which basically can dictate the multi-drug resistance in them. So a lot of challenges in terms of how you can treat and manage these particular sepsis cases. As I said, quite a few challenges. One is in terms of understanding what are the kind of pathogen reservoirs. So the pathogen transmission and the dynamics. What is the kind of timely, quick, accurate, and cheap diagnosis that one can give? Because most of these individuals, especially in the government hospitals, can pay even 10 rupees because they earn less than 50 rupees per day. So then, of course, you're looking at, as I said, because we don't know what the pathogen is, targeted treatment is going to be a big challenge. Other things, the other thing is in terms of the negative blood culture negative sepsis, given that you don't know what the pathogen is. That's the true nature and the horrific face of the sepsis. So indicating that this is a big, big challenge, especially in neonates. So because of that, with the generous funding from DBT, we were able to set up a big multidisciplinary, multi-institutional research. So wherein we have 5 hospitals and 7 research institutions addressing approximately 11 questions. The program goals are primarily to, of course, reduce the burden and also see whether you can improve our current understanding of the sepsis. Some of the projects that we have, I'm just going to highlight, I'm not going to go to the details. One is in terms of doing a GWAS of approximately 650,000 SNPs by using the Infinium Illumina. This is in collaboration with IGIB, Rajesh Pandey, so as to see whether we can identify some of these SNPs which possibly can associate with the susceptibility to sepsis and possibly death. The other is, given the plethora of the pathogens which are opportunistic, one can think of looking at the host genes, and there is a plethora of, again, transcriptome data coming from a lot of global cohorts. And so there is a group at Stanford which basically crunched all this data and came up with a particular set of genes that we basically are trying to see whether we can do a targeted gene expression so as to say whether we can suspect sepsis even before the symptoms kick in. And the other is in terms of looking at the immune markers. Veena is here. Veena, Anmol, and Dipankar from JNU are looking at some of these immunocomponents. And Ranjan from IJIB is looking at the metabolite biomarkers. So the other is, of course, as I said, how do we diagnose the culture negative sepsis? The other is in terms of the progression of the microbiota. You start monitoring the microbiota of the neonates right from day one, and as they enter into sepsis and post-recovery. Can you basically monitor the microbiota and say that is this microbiota is going to be dictating whether somebody becomes susceptible to sepsis or not? And of course, given the kind of volume of data this program is going to come up with, we possibly are going to look at the omics. Sorry, this seems to be having its mind of its own. And then, of course, we have a biorepository in the institute, which basically will be sampling, or rather storing all the samples that we have in case we don't utilize some of that. So I'll just touch upon the dynamics of this pathogen transmission and how worrisome this can be. And so bear with me because there is no CD8, CD4 information here. But so the question is, in terms of the transmission, as I said, is the mother transmitting it? This is in utero. This is considered as vertical transmission. This, of course, it is coming. Where are the pathogens? It has to be coming from the environment. So the mother could be bringing it into her even before the delivery. And so the neonate, after the neonate is born, one can have the pathogen transmitting from the hospital into the neonate. Of course, this is a big, big question. And this kind of a experiment, rather understanding, has not been dealt in this country. And this is the first, so there's a lot amount of expectations. Hopefully we'll be able to meet some part of it. So the reason why this is important is this possibly will help us to understand whether this is the management of the pregnant women or the hospital, or possibly both. It's quite possible that every pathogen is not going to be horizontally transmitted or vertically. So it depends upon the pathogen. So we, in this particular study, we are looking at four important pathogens. So the Acinetobacter pneumoniae, Klebsiella pneumoniae, then Pseudomonas aeruginosa, and E. coli. Because these four pathogens cause approximately 70% of all the blood culture positive cases in India. And approximately 60% of the neonates who are blood culture positive in any of the NICUs across the country, primarily possibly the government hospitals, die because of these four pathogens. So what we do is we basically sample the environment every single month of the study. So the study is for 5 years. The sampling will happen possibly for 2 to 2.5 years. Some of the hospital sites, we have approximately 36 samples, some 24 samples, and 18 samples, and so on, depending upon when the hospital started the enrollment. Simultaneously, even before, before the neonate is born, the mothers are enrolled, and basically their rectal vaginal samples are collected, because that is, sorry, I didn't realize I was so slow. I'll try to be as fast as possible, sorry. So we sample the swabs and basically culture them and look at these pathogens. We do the same thing with the neonates, whoever come into the NICUs, isolate the blood, then again collect the samples, and then stock them, and basically take these samples back, depending upon the cognate mothers, the environment and during that period, and the blood isolates, and do a comparative genome sequencing. So far, we have screened approximately 120,000 mothers, enrolled approximately 4,300 mothers, and neonates close to 5,000. So what I wanted to emphasize here is every single tick in these particular sites is basically different reservoirs. There are 54 reservoirs which we have sampled from each of the hospitals, 54 reservoirs from every hospital, every single month. And every tick indicates that every time this is cultured, you end up getting one of these 4 pathogens. Not all of them, but one of these pathogens. And you can see how many times these pathogens come back. And this is post whatever the management the hospitals are doing at their end. The same thing in the labor rooms, same thing with the mother OTs. And we then also take the swabs to do a metagenomic analysis. And what we see is these environments are enriched with a lot of the pathogens that basically are causing sepsis. And most of these pathogens are MDR and XDR, as I was saying. And we do a lot of comparative genomics. This is in collaboration with Baptosh, who is great with sequencing. In terms of MLSTs, 5S, 16S, 20S, 23S, and also several ribosomal genes, and also the core microbiome, the genome comparisons. So a lot of comparative analysis is going on right now. Because of the interest of the time, I'm just going to go quickly to the summary. So what we know is that there is a lack of sterility in all these hospitals, in the hospital reservoirs. And approximately 75% of these reservoirs clearly have bacterial flora. The analysis, the metagenomic analysis indicates that it is rich in Enterobacteriaceae, which has a lot of pathogens. And as I said, all these 3 main reservoirs are replete with these pathogens. And mothers harbor E. coli, most mothers harbor E. coli in the rectal tract. Interestingly, not every neonate or every hospital has the E. coli as the major burden pathogen. I also talked about the antibiotic resistant genes. What is interesting is the mothers who basically have either Acinetobacter, Klebsiella, or E. coli in the rectovaginal tract do not have the neonates having that particular pathogen, rather getting sepsis with that particular pathogen. We don't know even if we get it, that is the one which is exactly, very highly homologous in terms of the genomic content. We will have to look at that. But in general, we don't even see that. On the opposite side, on the contrary, those neonates, so the mothers with the blood culture positive neonates for these pathogens do not carry them in the rectal vaginal tract. So we predict that the vertical transmission possibly is quite low and the horizontal transmission is predominant. And of course, our comparative genomic analysis is what is going to give a lot of answers, and in terms of some of the recommendations that we possibly will be able to give to these hospitals. Of course, there's a lot of other things which are going in the lab. I won't go into the details, but that is my group. And thank you for your time. Thank you.
Thank you for a very nice presentation. And the statistics are staggering in terms of, 70% of the hospital samples having these resistant pathogens. I have a question, but we'll first go to the audience for a couple of questions.
It was a nice collection of information as far as sepsis is concerned. What is, the earlier speaker also spoke on that the definition of sepsis is not clear, he was mentioning. Okay. But if you could have made one slide of that, that would have, made all of us knowledgeable on that.
I did give the definition, but possibly.
I might have, yeah, okay. One thing I would like to know, when the culture is negative and you are calling it as a sepsis and your metagenomic is showing that this is the organism, and if you treat the patient with that, will it be helpful, and how much it would be helpful?
That's a great question, because it's not as easy as your question was, because let us take, for example, even a culture positive to begin with, and you do a metagenomic analysis of the syrup. Just because the case was Acinetobacter, for example, in the blood culture, when you do the metagenomic analysis, you don't see Acinetobacter as the number 1. It could be anywhere between 100, 150, 50. It could be any organism, because you're looking at the abundance of the genome content. But when you are looking at the blood culture, you have cultured it. And that's why, because the media supports it, you see that organism. So one of the challenges in terms of use of NGS to look at metagenomic analysis and predict that this is the pathogen is going to be challenging. But if we see the pathogen, one can think of presuming that this is an opportunistic pathogen among a backdrop of quite a few other commensals. So you should be able to think of treating it. But it has that limitation in terms of the cost because, even PCRs are not being done in these hospitals. So having to ask somebody to do per se every sample and NGS is going to be impossible. We have to come up with something much, much cheaper and better. But because of the research, yes.
Thank you. So in view of time for the next talk,
you had some
thank the
questions.
speaker. Yeah, I'll catch up with you later. Thank you. So we'll go to the next presentation, and this is Dr. Chandrasekhara, who is my co-moderator here. And he's a renowned hematologist, and he did his MD from Mysore Medical College. And subsequently, he was an assistant professor there and a consultant immunology at MS Ramaiah Medical College. And he has been pioneering in the field of rheumatology research, and he established the Chandray Rheumatology and Immunology Center for Research, a specialized one-stop center providing care and treatment for diverse autoimmune diseases. He's going to speak to us about personalized and precision medicine for autoimmune rheumatic disease. Is it a dream or an expected reality? Welcome.
Thank you, sir. At the outset, I thank both the scientific committee as well as the organizing team for giving me an opportunity to share some of the concepts or the perceptions that we have, both as a clinician, to some extent, probably few of the basic science work which I have done. What do we anticipate when it comes to the management of autoimmune diseases? I think this is my first concept paper, which I published in 2012 in Indian Journal of Pharmacology, where I just tried to address or understand what it was when it was in 2014, the way we were thinking or considering managing the autoimmune disease. In medicine, if we have a disease, we broadly have four perspectives of approach. I think my previous speaker who was speaking about sepsis made it very clear when it is an infectious disease, what we try to do is identify the pathogen, figure out the antibacterial or antimicrobial, treat it with the antimicrobials. And probably the major perspective with which we treat there is to eliminate the bacteria or the pathogens as much as possible, simultaneously hoping that the host also attempts to clear it without rolling or going into sepsis. The next model which we use in the management is what I call as oncology therapeutics or oncology management protocol. There we have an aberrant cell which has gone abnormal in its metabolic process and regulation, and you can focus on the cell's metabolic process and try to hit it and eliminate as much as possible, either by surgical method or debulking, or use chemotherapeutic drug or any other drugs to get those cells eliminated. And then comes the metabolic or defective causes for the diseases like hypertension, diabetes mellitus, or any such. You know, there is a specified or a broadly specified metabolic or a regulatory dysfunction. Try to hit on that, try to modify that, bring it to the best level where it can manage a homeostasis process and control the process, as a consequence of which you can do good with the disease. Then comes the transplant rejection model. That's what we most of the time use with reference to autoimmune disease. Here, I think the speaker has fairly well said 2-3 before, that we transplant the organ and we try to see to that the immune system do not harm the organ that I have transplanted. So that's the fundamental on which we tried and we started attempting in managing the autoimmune rheumatic disease. And over a period of time, we learned a lot. I think I can still remember in 1982, when I got interest into immunology as an MBBS graduate, The first case which was shown was a systemic lupus erythematosus. During the rounds, my medicine professor said that she has a nephritis involving because of systemic lupus erythematosus. She survives for next 4 or 5 years. And that time has changed. And now we can say that the kidney can survive more than 15 to 20 years if it is properly treated. Why that happened is we had a reasonable good learning curve. In earlier days, we used to talk of giving disease-modifying antirheumatic drug or immunomodulator very hesitantly. We used to stray as much as possible symptomatic, start with a very small minor doses of immune modulators, and gradually increase it over a period of time. Slowly, we learned the safety of it. We started working with combinations. We also learned that immune intervention is going to make a huge difference in the outcome. Then targeted therapeutics. Here, I think I would like to just get the attention of everyone. We use a term targeted therapy or targeted intervention, which is completely different from what we talk about in an oncology. When we talk of targeted therapy in oncology, we talk about the metabolic process which is abnormal, and we are very clear targeting that particular process is going to eliminate the cell in question. Here, we talk of targeted immune suppression is on the immune limbs. It's anti-TNF, we are going to block the TNF pathway. Anti-B cell, I am going to block the B cell pathways. Here it is not that, we are not really focusing on the autoimmune process or the process which is completely destabilized. So, however, those drugs have really improvised our outcome. Slowly, we started realizing we have come to a point where we can achieve a state of remission in a good proportion of patients. Overall outcome of these patients have significantly improved what it used to be 2 to 3 decades, where we used to say that he lives for a few years, to where we are now very clear how long he lives, how best we can make his life, and can we achieve a state of remission. That's where the pyramidal approach, where we used to start from symptomatic to go into the disease-modifying antirheumatic drugs or any other thing, has been changed into early introduction, early intervention with immune modulators and immune suppressants. But we still have a challenge. Immune system, as we say that, I always say that do not mess with me, because anything and everything can go wrong. Because the moment I go with immune suppression, the infection becomes a major challenge. When I go with higher immunosuppression, possibility of developing malignancy, and we do sometimes see the patient developing a secondary autoimmune disease is always a possibility. However, what is the current working model? How do we treat or how do we manage most of our autoimmune disease? How we normally manage is, we first make a diagnosis. We call it either it's rheumatoid, systemic lupus, vasculitis, or whatever so. Then we look about the organ that is involved. Say, for example, if it is systemic lupus erythematosus, if it is kidneys or CNS are involved, we go with a higher immunosuppression. If it is only MSK, musculoskeletal involvement, or the skin involvement, we go with a lower profile immunosuppression. And we manage and monitor the activity of the disease based on the organ systems functions. For example, if it is a joint disease, how much is the joint pain, what is the swelling, what is the tenderness, and if there is inflammatory parameters like ESR, CRP, and C3, C4, how much it has been modified. Based on that, with previous experience, previous trials, and previous available literatures, we try to modulate and manipulate, and overall, we treat the disease process. But at we very clear about that we have not really focused on what is the exact pathogenic process which is triggered, maintains the autoimmune disease. So that being the scenario, are we really, or can we think of a personalized treatment? And why do I really need a thought process of why do we really need to think about a personalized or a precision medicine when it comes to autoimmune rheumatic disease? I'll just give you a simple example to go through. This is the current recommendation what American College of Rheumatology recommends when you want to manage a rheumatoid arthritis. Make a diagnosis of rheumatoid arthritis. Once you are sure it is rheumatoid arthritis, you classify whether he has a bad prognostic or a good prognostic. Good prognostic, start the patient with methotrexate, or if has a bad prognostic, start him with little aggressive combination of DMRs and move along. And if patient doesn't respond, then go to the next stage. You use biologics, you use target-specific DMRs, or you combination thereof, not still doing good, then you go for experimental therapies. So now, the major challenge that we face is, when I start treating 100 patients in the very first stop, only Still 40% of them don't achieve what I am looking for. Patient doesn't get a remission. His pain still continues. He continues to suffer from the disease and disease symptoms. If not 100%, at least maybe 30-40%, because we would love to have 100% subsidence of the disease activities. So now what I have to do with this 40%, then I go to the next step. When I go to the next step, here again, 20% do good and another 20% or 40% among them may not do good and have to go further. So when that is the situation, what I need is, if I pick this 40% or 20% the very beginning, if I start in the very point when he enters, if I could pick or identify these patients, I would love to jump or introduce these patients into the second level medication rather than working him with the patients or trialing him with the drugs of either mono or say or combination therapies, which otherwise for last 3 to 6 months we have unnecessarily exposed these patients in a drug which was not otherwise useful to him. So going and moving towards personalized and precision medicine is definitely going to make a huge difference when it comes over to these set of scenarios. So now, what is the real positions we have from most of our trials? If you can just quickly go through, you can see these are the different compounds that has been all approved, except anakinra. So if you see most of these biologics, say abatacept, it was successful in 44 people out of 100. What I'm talking about is ACR, more than 70% response, because that's a best response that I expect. It happens only in 44 out of 100 patients, and the same thing in 21 out of other 100 on the placebo also had a good response. So we have a lot of issues with reference to each of these. What it makes us to, in a nutshell, is few percentage of patients respond and a certain percentage of patients do not respond. Few percentage of patients who didn't respond to abatacept responds to the another biologic therapies, and subsequently he may do good with that. So in a nutshell, what it happens is currently what we are working is one size fits all. Once I diagnose a rheumatoid arthritis, start him with these ABC pathways, doesn't work, then go to the next pathways. And this is one of the major challenge I face and any of our clinical staff faces. The first question the patient asks is, "Doc, are you sure this drug works for me? And is there any other better drug for me?" So here the question is very difficult for us to answer. This is where we are now looking forward, because we have significantly in past 2 decades, we have really improved with reference to therapeutics, but we are still not able to address these questions. The reason I still feel is we believe in what we are calling as Mahajano Enogatasapanta. I think this is a Sanskrit term which is used with 2 different contexts. Mahajano, it means the great people. It's always best to follow the pathway of the great people. The other term for Mahajan means is a group. Follow the big group. And most of our clinical trials, what evidence-based we talk about is, if more than 60% patients do good and achieve the target, the drug gets its approval. If p-value between the group A and group B is more than 0.05, there it is. I have proved the drug works in good proportion of the patients. But we are forgetting the other 30-40% have not shown a good response. And what should we do with reference to these patients? This is where the role of precision or the personalized medicine comes. Just to make clear between these 2 terms, precision medicine is totally different from what we talk about personalized medicine. When we talk of precision medicine talks about the defect in the disease process. For example, if it is an oncology or a cancer cell, if ABC or BCR-ABL is the defect, the drug you use is to alter or modify the BCR-ABL functioning, and that's why you are able to get the things. That is where a precision medicine, what we talk about. When we talk of personalized medicine, in addition to the precision with the drug, you also have to take about the genetic aspect. How does that patient metabolize? What is the possibility of adverse reaction that he's going to have with these drugs? All these issues are also come into play before the precision medicine translates into the personalized medicine. So one of the major challenge, I think the previous speaker also was mentioning about, is the cost factor of it. If I develop a biomarker test of all these things, the cost of therapy is going to increase phenomenally. But if you go back to my previous 3 slides, if I save a patient's being exposed to a drug which was not otherwise useful for a 3 to 4 months, and if I save 3 to 4 months of his time in getting into a good active life with a perfect drug, that whatever the biomarkers investment that has been done could be completely saved. Because if you look from the different perspective, certainly these biomarkers, if one move into the personalized medicine, can significantly improvise both the patient's outcome as well as from the economical perspective. But it's not that simple as we talk about when it comes to the autoimmune rheumatic disease. So just to get into the problem, crux of the problem, see, what are the questions or what are the possibilities which we can differentiate between an individual who responded versus an individual who didn't respond? One is, it could be refractory because of the immunological mechanisms, or the autoimmune response which has been developed must have been high affinity, or because of the varieties of different immunoglobulins profiles, glycated, non-glycated. I mean, you have a lot of factors which can influence and alter the immune responses. And the immune system or immune response has left for a longer time, it would have created pannus, it would have created peripheral lymphoid tissues, it could have become an autonomous lymphoid features that itself could have added or contributed a lot with reference to the disease resistance. And drug factors, of course, the development of resistance, the faster clearance, and allow deviations. In addition, we have two other factors which we need to seriously consider. That is doctor's factor as well as patient's factors. Patient's compliance matters. Doctors picking up the right drug matters. So barring that, there are a lot of immunological factors which one can help in working out, personalizing, and precising the medicines. As all of us know as an immunologist, autoimmune disease has multiple steps. It starts with the genetic environmental trigger, gradually gets into an autoimmune response and establishes itself, starts becoming pathogenic, and over a period of time, it grows. And in a nutshell, what we talk about is, it's just the sensitization of the self proteins, either B cells, autoantibodies, or any of them start aggressively attacking the tissues, and thereby also work up with the lot of innate cells. So now the problem is, it's not just few medicines I have. We have plethora of medicines. We have a name a pathway. We have a antibody or a mechanisms in which we can block that. That being
Time
the
is
case,
up,
I have 2 minutes. So that being the case, we have a lot of things to work with. So, but one of the major crux currently now we have is, there are very few publications. We need to work and explore this. And one of the major limitation is, it's a very complex process. Autoimmune disease is quite a complex process. Multiple steps are involved, and developing a model or developing a mechanism seems to be a little difficult. So you need to use multiple biomarkers, use composites biomarkers, which can over a period of time should help. Hopefully with the development of AI, ML, and other modeling techniques, we should be able to achieve this. Currently, it looks as a far distance achievable things. But it's just not that. If one puts it in mind, probably we should be able to achieve it. But this can definitely alter and revolutionize the way we are going to treat and manage autoimmune rheumatic disease. Thank you very much.
Thank you, Dr. Chandrasekhara, for a nice talk. Open to audience for 1 or 2 questions.
It's always wonderful to listen to Dr. Chandrasekhar. I have one or two clarification rather than question. When you said the bad prognosis in RA, rheumatoid arthritis, it gave me some sort of shock. Though I am not a clinician, but I manage a lab. So this is something maybe we can put a word to that. And the second thing is, in RA, quite often we have seen anti-TB antibodies in about 30 to 40%. So if you can look into that aspect, probably, it may not go to the bad prognosis if you can control that aspect. That's my feeling.
No, I think when it comes to the prognosis, what we talk about is the way the patient presents. Patient has a very, say, multiple joints involved with highly swollen joints, with the functional activity being significantly suppressed, and his rheumatoid factor anti-CCP being positive in a very high titer, if his X-rays already start showing an erosions, that suggests that the disease is very aggressive and prognosis is not great. So that's where we try to use combination earlier than going for a single drug. With reference to TB autoantibodies and TB antibodies, I think it can be seen in Indian populations for a larger extent. And even for that matter, where we associated Takayasu with the TB positivity, we have now come out saying that it's not really so much associated. Probably there could be, but 1 or 2 patients may be related, but we need to see how it, when it comes to personalized precision medicine, probably the presence of infection in the patient may significantly influence the outcome. So we need to work from that angle too. That's why one of the multiple markers that we may have to have is the presence of chronic viral infections and chronic persister infections.
All right. If there are no more questions, thank you, Dr. Chandrasekhara, for your presentation.
I welcome the next speaker, Professor Dr. Vinita Shobha. She is professor and head of the Department of Clinical Immunology and Rheumatology at St. John's Medical College Hospital. She is both an astute clinician and a researcher. And we can say that she has literally driven our Karnataka Rheumatology Association for a lot of collaborative research. And she has published quite a good number of papers, both in basic science as well as on the clinical areas. She finished her both post MBBS as well as MD from PJ Chandigarh, and subsequently completed her DM in clinical immunology from SGPJ Lucknow. Subsequently, she has been with St. John's, and she has developed an excellent, very good department of rheumatology and immunology at St. John's, which is also running DNB courses. And over to you, Dr.
Hello, good evening, everyone. Thank you, Dr. Nandi, for inviting me here for IMMUNOCON. This is my first time here. And thank you, chairpersons, Dr. Chandrasekhara, for kind introduction. So I have chosen this topic to talk to you all about today, which is titled Mastering the Lupus SLE Obstacle Course, Strategies for Doing Better and Going Further. So I'm not sure how many of you would know about this disease, which goes by the name SLE. Full form is systemic lupus erythematosus, frequently just called lupus. It is a multi-system autoimmune disease, which is characterized by multi-system involvement. That means it affects not just the joints or the skin or the particular organ, it affects almost every organ system in our body. And it also has plethora of autoantibodies. In rheumatoid arthritis, what Dr. Chandrasekhara talked about, there are just 2 or 3 defined autoantibodies, whereas in condition called lupus, there are almost 10, 15, 20 autoantibodies which have been described. Unfortunately, this disease also, like several other autoimmune diseases, has a chronic disease course with frequent flares. So basically, in this disease, SLE, the immune system goes rogue, and the human body is considered as a foreign organism or a foreign tissue against which the immune system starts attacking, causing disease. There are some classical manifestations in lupus. For example, we can have what is called as the butterfly rash in these people. I'll show you a picture here. So this is, if we see this, then our diagnosis is very easy. But several times we do not see this classical rash. And I also want to mention that this disease affects usually young girls, usually in the age of 15 to 35 years. 90% of the patients affected are girls, and boys are only 10%. Untreated, this disease has a very high morbidity as well as mortality. So why did I choose this topic today to talk to you all? Because we need information about this disease from our country, because the clinical manifestations as well as the pathophysiology differs across the geographical regions and ethnicities. So we need data on this disease from our own population. Therefore, it is imperative that we design our own diagnostic algorithms, therapeutic protocols, and that requires collaboration between scientists and clinicians. And that is why I have chosen this topic and I'm here today. So despite several advances in our understanding of pathophysiology of this complex illness and discovery of newer therapies, we are unable to achieve sustained deep remission in more than 3/4 of our patients. So there is still a lot of distance between the cup and the lip. You know what I mean here, that we are not where we should be in reference to this disease. So I'll just tell you a story of a SLE patient who's under our care for last 10 years, and we still haven't found a solution for her. So I've kept this talk extremely light so that all of you can keep awake and not feel tired. So the first obstacle we face in the context of lupus is a delay in diagnosis. So I'll tell you this story of this girl whom I just mentioned about. She was very lucky that her diagnosis was established within the first 2 to 3 months of onset of symptom because she had fever. So she was 22 years old when we saw her in 2014. And she had, as I mentioned, constitutional symptoms, which is fever, weight loss, anorexia. And she had this butterfly rash on her face. And she had inflammatory polyarthritis, somewhat like rheumatoid arthritis. But in addition, she had inflammation of her serosal surfaces. She had pericarditis, she had pleuritis, and she had anemia, which was immune-mediated, autoimmune hemolytic anemia. She had low counts, leukopenia. She also had affliction of her kidney, lupus nephritis, and she was predisposed to thrombotic manifestations because of presence of autoantibodies, which are called as anti-phospholipid antibodies. So in this patient and in all patients, we've already wasted 2 to 3 months of time. So early diagnosis is a challenge in our country and all across the world because of the heterogeneity of the symptoms and signs. And this is one story of one patient, but no two SLE patients are exactly alike. So that makes our life extremely difficult. The next obstacle which we face is that there is no single diagnostic investigation. So apart from the diversity of this clinical picture, the lack of a single diagnostic investigation also contributes to a delay in diagnosis. So what we frequently have to land up with to look at a constellation of clinical manifestations, laboratory parameters to confirm the diagnosis. The laboratory parameters which we have to look at are different in different people based on the organ systems which are affected in these people. So why is this early diagnosis so very important? We want to hit hard. We want to hit with the efficient treatment before the web of the cytokines and the autoreactive cells gets entangled and knotted together beyond repair. That is before the damage has set in. So we want an early diagnosis, and we are very lucky that we are in India. In Western countries where they have functioning on appointment system, it takes as much as a year for diagnosis. Whereas in India, we recently published this study, our median time to diagnosis is only 6 months. And in about 1/3, we could diagnose as early as 3 months. So what happens is that, we actually, when we saw this data, we were very happy, but still, unfortunately, our disease is very different from the Western population. And we do not have this early diagnosis has really not transcribed into a lower mortality in our country yet, possibly because the aggression of a disease is much more than what we see in some of the Western counterparts. So why does this disease occur? The patients, the first question they ask when we give them a diagnosis of lupus is, why did this disease occur? But we don't have an accurate answer to it. We are aware of several risk factors, triggers such as genetic and environmental factors, especially viral infections, but we are not able to reach the root cause of why this illness happens. Genetic makeup of individual does seem to play a role, but we haven't caught it yet in a most perfect manner. This paper was published almost 12, 13 years back, and I took this very simplistic figure just to say that SLE associated foci on the chromosomes are widespread across several areas, and they're related to functionality of several cells, such as T cells, B cells, dendritic cells, and several cellular mechanisms. So this is again another simplified slide just to highlight the complexity of the pathogenesis of SLE, wherein both innate and acquired immune systems are aberrant along with defective immune regulation, eventually resulting in permanent tissue damage. So for the students here, if you want to learn immunology, dive into the pathophysiology of lupus, because this disease tells us and shows us that anything which can be going wrong in the immune system goes wrong in this disease. So what about the treatment options? That's another massive challenge for us. As a clinician, we are only having immunosuppressive therapies in our armamentarium. And there are no perfect guidelines, as Dr. Chandrasekhar also was trying to allude to, that it is not possible to choose the right drug for a said patient. And so basically we go just by the severity of the disease and try to match the degree of disease severity and with the degree of immunosuppression. So if a patient has severe disease, they get more severe immunosuppression. If a patient has less severe disease, they get less severe immunosuppression. So choosing the right treatment actually requires us to become detectives so that we can deliver the best optimal therapy for our patients. Now coming back to our patient, as I said, we saw her in 2014 and she had nephritis, she had involvement of the kidneys. And so we treated her with glucocorticoids, steroids as a popularly known as, and with other immunosuppressive treatment. She did very well. In 1 year's time, her kidneys were into remission. But in 2018, she had a cutaneous flare. And as I said, she was a young girl. She had a baby also in 2018. In about 2 years after that, we got frantic calls from her family saying that she's not able to move her legs. So she had developed paralysis of the legs because of the autoimmune disease, which was now affecting her spinal cord, causing transverse myelitis. So we had to change gear. We had to give her different kind of immunosuppressive medications, which we usually do not like to give to young girls, that is cytotoxic drugs. Last 2 years back, again, she had a flare in her kidney, and we had to again switch gear to multi-targeted immunosuppressive therapy. Currently, she is not doing too good. She's just recently been discharged. She was given rituximab a couple of months back because of ongoing disease activity, which is a B cell targeted therapy. The autoantibodies are being produced by the B cells. So what we try to do here is blindly kill all the B cells so that the production of autoantibodies will stop. And we hope that there will be a reset of the immune system. And when the B cells come up, they will not produce the autoreactive antibodies. So she was recently admitted with septic shock. We've heard a lot about infections, along with acute renal failure. And as was being told in the neonatal sepsis, that her cultures were negative. So we do not know what to do. So we've given her antibiotics. She's not improving. So we have given her IVIG. And we just discharged her last week. And I'm hoping to see her alive in next 2 weeks' time. So this is lupus story. Ups and downs are part of constant vigilance. And so my reason for being here is to induce all the young scientists to start working on autoimmune diseases, move away from infections, not entirely, but partly, and move away from oncology and come to autoimmune diseases. So it's already been said that one treatment is not the right treatment for all the patients. And unfortunately, so for you scientists, the pharma industry and the research teams have largely focused on developing new medications, but the biomarker discovery has sadly lagged behind. And so we are not able to choose what Dr. Chandrasekhar also alluded to, that we do not have a right biomarker which can tell us this is the right drug for this patient. This is what is going to happen to this patient. This patient is going into deep remission. This patient is more likely to die. We do not have that information because the biomarker discovery has actually lagged behind. So basically, simply speaking, we are throwing medications blindfolded, not really knowing whether it will work or not. So this story was just about one patient, but as I said, no two SLE patients are alike. And by help from you, we hope that we will be able to group patients into different subsets, which can help us to choose the best treatment. Now, I'm only talking about the therapies, the diagnosis, but these patients actually suffer a lot because it's a chronic disease. So they not only have to deal with SLE, the lupus, but also handle damage. They also have to handle comorbidities, like they might develop secondary diabetes, hypertension, atherosclerosis, infections, and of course, the adverse effects of the drugs. Infections are something which we are massively worried about all the time because we are immunosuppressing all of these patients and we are exposing them to a high risk of infections. So I just want to give you one example of side effect. So my girl, she was 22 or 24 years old when I first saw her. She was a beautiful young girl. And what happens with glucocorticoid therapy? So she becomes obese, she develops moon phases, she develops hirsutism, she develops TRY, and she doesn't want to go to college anymore. She doesn't like to go to work at all. So this tells us that there is a big gap between what we could do for our group of patients with autoimmune diseases as compared to where we are today. So from the patient's perspective, it's an unending roller coaster ride. One thing or the other is always going wrong, as I showed you in my patient. And just imagine a young girl being told to take heaps of tablets every day, right? So not just 10 tablets, sometimes as many as 20 tablets in a day is what these patients need to take. And some of you might be able to recognize this girl, Selena Gomez. She's an American singer and actress. And she had lupus and she landed up getting a renal replacement. Partially because of her busy lifestyle, she did not take her medications properly. She probably had poor compliance in addition to an aggressive disease, and she landed up getting a renal transplant. So that is what can happen to these young girls. Economic burden, that's something now beyond the boundaries of this room. But I want to mention here that the treatment is not easy. It is not inexpensive. It is extremely expensive, leading to catastrophic healthcare expenditure in as many as 1/3 of our patients with lupus. And the major cost is of the medications. High mortality, so there is a cohort of lupus being built across India, across 10 centers. And in that we have found over a 18 months follow-up, the mortality rate is as high as 10%. And now when we looked at our data at 36 months, the mortality is close to 20%. So here we are talking about a disease which is affecting young girls and is having a high mortality. I'm not talking about the morbidity. I'm talking just about the mortality. So I want to induce my young friends again to start working in autoimmune diseases. So this is about the INSPIRE cohort. And this was a DBT funded study. And I'm showing this picture here because I want to tell you that in this cohort study, with the vision of the leaders who led this study, we have the samples kept for these patients, 2,500 patients. We have their DNA. We have their serial urine and serum blood samples, which can be used to study this complex disease and support us clinicians in doing better in taking care of these patients. So before I end, myself and Dr. Chandrasekhar would like to invite you to participate in our conference, which is going to be held in Bangalore next month. It's called Applied Immunobiology. In this session in Indian Rheumatology Association conference called IRACON. So I request all the young scientists to please join us, wherein we can try and join hands with you and tell you what we need from the scientists, what we need from the bench, rather than bench coming to us and asking for samples. Thank you very much. Thank you for your patient listening.
Thank you, Dr. Vinita. It's a fantastic summary of treating and dealing with the lupus and its complications. Very informative. Any questions from the audience? Maybe 1 to 2 questions. Yes, I think there is somebody there.
Good evening, ma'am. I'm Pratyusha. I'm a PhD student at IIT Mandi and I work on autoimmune diseases. One thing that bothers me a lot is that on Indian population, we don't have the stats available for autoimmune diseases. We don't have, we are not very attentive towards autoimmune diseases, so we do not know what is the incidence rate, what is the prevalence rate. And we keep calling this as rare diseases, but I think it's misleading to call this rare diseases unless we have the stats on the table. So I wanted to ask if you are aware of any database which can provide us about the stats in the Indian population at least.
So ICMR is particularly not in favor of autoimmune diseases. And if I want to write a grant to ICMR, there is no option for me. That section doesn't exist in the in their naming of the diseases. So, but there are certain data. There was a COPCOD study for autoimmune rheumatic diseases. So we have some data regarding the prevalence. And from Karnataka, with in collaboration with several other rheumatologists, we do have some data sets which has been published. But if you're talking about the samples, the cells, that is there in the INSPIRE cohort for SLE patients. But I don't think any other center or any other registry is at the moment saving any cells or sera for most of the autoimmune diseases.
Database too. We have another database called Indian Rheumatology Association database, which has with reference to autoimmune rheumatic diseases. And similarly, if you want on multiple sclerosis, etc., I think NIMHANS and its team has with reference to that. Encephalitis of autoimmune type, again, NIMHANS with its other branches have its an encephalitis, autoimmune encephalitis data.
Yeah, NIMHANS also has brain banking.
Thank you, sir. Thank you, ma'am. I'll take note of it.
Open up to get that.
Dr. Vinita, I have a couple of questions actually. One is, do we see any ethnic, like let's say India is a very multi-ethnic population. Do you see a higher incidence of lupus or severe lupus in certain ethnic groups versus others? Because that might give a clue towards unique genetic predisposition for our population.
So there are very few studies with respect to genetics of lupus in India. I think there is one from JIPMA, Dr. Negi's group, where he looked at the Dravidian population. I don't often remember how different was it from the rest of the world data. But this INSPIRE cohort, actually it had started with the premise that there will be differences in the genetic makeup of, or the manifestations of the SLE across different parts of India. But the data analysis, what we have done now, it is not showing any major differences in the north, south, and east.
And the second question is voclosporin and belimumab, they are fantastic addition, to the management of patients with the lupus and lupus nephritis. So we have been using it, in the US. Are they available in India?
No, they're not. Both of them are not.
Okay. There is one more question.
Ma'am, in SLE, there is one enzyme, is lack of enzyme in many patients. Like that, in FDA also implicated that that enzymatic therapy in most of the SLE patient, which is improved. That enzymatic therapy is not available in India.
I'm not aware of any enzymatic therapy for SLE. I think you might be confusing with some other disease.
No, there is a circulating DNA is the most of the patients.
So the anti-dsDNA therapy, that trial failed. It didn't meet its primary endpoint. It was called Abetimus. That trial happened even in our center as well, in India also, but it didn't meet its primary endpoint.
Thank you, Dr. Vinita, for a very nice presentation. Thanks, everyone. Thanks,
Thank you.
Our thanks to the speakers and the chairs. I would request all the speakers of this session and the chairs to kindly get up on the dais for the group picture. It's my pleasure to welcome you all to the next parallel session featuring a series of engaging talks on the latest advancements in systems biology. I would like to invite our session chairs, Dr. Pramod Kumar from BARC, to take over and lead the discussions. I also welcome Dr. Narendra Dikshit as a chair for this session. This is the pointer, ma'am. Do you want me to use the one on the screen also? No, I'll do it. This is the pointer. This is the
Good evening, everybody. Welcome to this session on systems biology. Our first speaker is Dr. Shailja Singh. She is a scientist at the National Center for Cell Science. She's done extensive work in systems immunology. She serves as the academic editor of PLOS ONE and is on the advisory board of FEBS Letters. Today, she's going to tell us about EGFR signaling. Dr. Shailja Singh, please.
Yeah, thank you for the kind introduction. And moving from models to therapeutic intervention, once again, we are back to model system. So wherein I will expose the Leishmania disease model system through the systems immunological part. The first few slides are for the students over here. So please bear with me as I was asked by the organizers to brief something about the systems immunological model. So as we all know that immune system is basically a complex network of cells and soluble mediators which actually evolved to defend in the host organism. And the complexity and the multi-scale nature of the immune system basically necessitates the use of sophisticated modeling tools. As we all know that as a biologist, we all get intimidated by the mathematical jargons and the seemingly complicated computational techniques. So the strategies are there in the SimBiology toolbox with the systems biology or the systems immunological approaches, wherein you can discover the unknown targets, unknown relationships, and also cause out the mechanistic insight to understand the immune system as a whole. So basically, systems immunology is able to provide a new strategy for understanding the human immunity. Now, when we go for the systems immunological part, the most important thing over here is that we focus on the modeling technique, approximations, and assumptions. Now, assumptions are very, very important because some of the kinetic parameters into the literature for most model system are missing. So how to start with? At some point of time, you do get an idea about the kinetic parameters. At some point of time, you have to do the discretization of those particular steps. The thing is, even if you develop your model structure, how do you define with the subsequent motives? You are listing down the components in a signaling network through a repeated iteration of pre-tested domains, modules, and motives so that you identify the target and deliver the target in a controlled and predictable manner. So this is how we go. And to set the stage, we start with an aphorism which is attributed to the famous statistician George Box, who said that all models are wrong, but some are useful. So this is very, very important because whenever I go for the presentations and the model system, when the audience say, then they say that certain part of your model has experimental data. And at some point of time, you have done the discretization, which is very much allowed because you do not have the sufficient background experimental data to start your assumptions or making your biological assumptions done. So, and many or most kinetic parameters, if you will encounter in the literature, they will coin it as sloppy parameters. So what do I mean when they are called sloppy? Means you have the flexibility to adjust the parameters with some scaling, with some orders of magnitude, and also the flexibility to provide the certain set of parameters in your model system which are stable, which are not transient to the dynamics, the signaling dynamics you have chosen with. And so you can say that those parameters have to be robust. So the systems immunology, the main thing is developing the multi-scale understanding of the immunological responses, as Professor Nagasumachandra also mentioned a few minutes ago about the multi-scale modeling in her SARS-CoV-2 infection model system, and also the parameterization given. So I will discuss about the mechanistic model of a macrophage-associated infectious disease leishmaniasis. Why? Because it has the ability of the macrophage has the ability to reprogram itself, and which can be tapped for application in immune therapeutics by stimulating them with chemokines or with immune stimulants like lipopolysaccharides. All of you might be knowing that there are three forms: visceral, cutaneous, and mucocutaneous. We had Christian over here who was demonstrating the visceral form of the disease. My lab actually works on the cutaneous form of the disease, which is caused by L major. If you can see, the global prevalence is very much in Africa, Brazil, Bangladesh, Ethiopia, South Sudan, and Sudan. In India, it is very much prevalent in Bihar, Bengal, Jharkhand, northeastern states, and some of the states like MP also has this visceral form of the disease. Now, the most important thing is, in absence of the drug, miltefosine is not an anti-leishmanial drug, it is an anti-cancer drug, and which is the only oral source of drug available for L major, L donovani, and L brasiliensis. As we all know, there are no vaccines till date which is available for this form of the disease. So just to give you a brief introduction, you have the infective stage, which is the pro-mastigate. The diagnostic stage is the a-mastigate stage. So when the infected sandfly bites the human host, it injects the metacyclid pro-mastigates into this skin, which are eventually released into the phagocytotic cell. Inside the phagocytotic cells, these get converted into a-mastigates, which are eventually released into the extracellular space to be taken by the another phagocytotic cell, or can be taken by the blood meal in the sandfly to begin a new cycle of infection. So this is the survival of Leishmania parasite inside the polymorphonuclear neutrophils. So you can see there is a release of leukocyte chemotactic factor by the parasite, which eventually releases the monocyte attractant chemokine 1b, and which is releasing your Trojan horses, that is the apoptotic PMN that harbors the viable parasites. So uptake of apoptotic PMN silences the antimicrobial function of the macrophages, and parasite survival is enhanced, and the survival of Leishmania parasite is continued, giving rise to the M2 phenotype. We all know the Leishmaniasis immunobiology with classical activated releasing the M1 phenotype with the release of IFN gamma, IL-12, and other pro-inflammatory cytokines. Alternatively, activated cytokines with the Th2 response with the release of anti-inflammatory cytokines such as IL-4, IL-6, IL-10, TGF-beta. So you have to have the Th2, Th1, that is your protective and susceptible immune response balance. So our goal is, can EGFR, which is one of the most important inflammatory signaling pathway associated with CD14 and TNF modulated by Leishmania, can we have a cross-talk point of intervention? So why CD14? Because TLR recognizes LPG, which is the PAM of Leishmania, TNF, which is this output of CD14 TLR activation, and EGFR, why? Because it causes the inflammation and skin-on infection. So we hypothesize that there could be a crucial cross-talk point of intervention which can be regulated by using the synthetic gene circuits, and that can be giving you acting as a positive feedback loop for a disease resolving inflammatory response. So in my lab, what we focus is on the systems aspect. So over here we are linking your signaling network to a logic based gene circuit which will actually give you a disease resolving inflammatory response. So the crosstalk point of intervention is being inserted into a circuit, and that circuit is being hypothesized in the form of a construct, and we do our wet lab experimentation to prove the disease resolving inflammatory aspect. So the theme is basically you have your molecule, you design the crosstalk, you embed it in the circuit, engineer the designer cells containing containing your therapeutic targets, and implantation of the designer cells into the mice model system. So the goal of the modeling in systems immunology is to provide the mechanistic insight, and basically you engineer the cells which can be used in various delivery platforms to sense the specific molecule. And integration of this biological device will help you to better understand and gauge the kinetic aspects of the signaling dynamics. Now, this is not the first time which is being done. Charles Martin Fusinager in UTH Zurich, he has sufficiently done work under this theme with urate homeostasis. He also has a synthetic circuit which takes into account the obesity. He has a synthetic biology-based device for liver injury in mice, and so on and so forth with the hypertension model also. So with this, we we had to have the signaling network of CD14, TNF, and EGFR pathway. So the downstream, we did the TFTG network, which was being regulated by these three pathways, and the crosstalk point of whatever we have identified, we tried designing the circuit with its validation in the experimental conditions. So how did we do the reconstruction? The reconstruction, as we say, we had the protein-protein interaction network from the various databases of all of these CD14, TNF, and EGFR. We had the two PPI networks with and without positive feedback loop. And we did the network analysis with certain statistical measures, as said, diameter and shortest path and clustering coefficient. Now, what do I mean by this? Clustering coefficient, if it is low, these are all statistical measures, students, which we adopt in systems biology. So it says that the transmission of signaling information is in one direction, okay? And diameter and clustering, shortest path, if you can see over here, with positive and without positive feedback loop, 11, 7, 10. That means local density of interactions, which has been calculated for the average, for the closest neighbor, averaged for a particular node for the entire network. Okay, so we are averaging the closest neighbor of a node for the entire network, and that is how we calculate it, and your software is able to, your statistical measures basically are able to give you these outputs. And if you can see, you have the crosstalk point of identification with NF kappa B, IKK beta, MKK173647. So all of these, and in the Symbiology toolbox over here, you can see this is the layout of the box wherein we had the reconstruction of all of these three pathways. As I proceed, I will tell you all of the crosstalk point of intervention was validated with different combinations of ligand and inhibitor of CD14, TNF, and EGFR. So you can see after identification, we had this question: how do these nodes behave in a dynamic condition? Do they influence the other nodes in the network? So yes, they do, because you have the reconstruction, you generate the time series data of each of this signaling component, and these databases, actually, if you can see on the right hand side, you have this ordinary differential equations. So what do they say? What they are actually taking into account the time evolution of the dynamical variability of the kinetic parameters which are associated with these reaction. Over here, it is the association and the dissociation kinetics of the two proteins that are physically interacting in your signaling network. So what do we say? We have mass action kinetics for association, dissociation. We have Henry-Michaelis-Menten for phosphorylation, dephosphorylation. And when you have a product, we apply Hell-Heinz equation. So all of these parameters, you can say, we have with without feedback, with feedback, the concentration range is there. And we deposited this model in the biomodel databases, which is curated by the curators. So we do the statistical analysis and find all of this flux sensitivity PCA. So what do I mean? I'll just say that you are finding any change in the parameters which is associated with these species should not dynamically change the output of the system. Supposedly, if I combine my flux and sensitivity, and you have certain components like STAT, you have ERK, you have cytoplasmic JNK, they have a high sensitivity. They have a high flux, but no sensitivity. Means what? They are not contributing towards the dynamics of your output. So you can just get rid of those parameters in your signaling network, which you have reconstructed. Now, if I combine my sensitivity with PCA and your certain components like ERK 1,2, which is downstream of MKK 1,2, and if you have JNK, which is downstream of MKK 4,7, and you have the other component, which was AKT, which is downstream of PI3K. So those are the crosstalk point of intervention, which should be looked for further interventions, So those also you just cannot debar. So what we do is fish out and churn the important parameters in your signaling network. So over here, these values you can see, the parameters are mentioned, the kinetic parameters, the initial concentration, and this says that only 20% parameters are important. So the 80% parameters which were redundant. We got rid of them and then we are focused. So this is the beauty of systems biology and immunological modeling, that you get rid of the spurious parameters by using the statistical concepts which are embedded in the MATLAB Symbiology toolbox. So further we went downstream and reconstructed the TFTG network with the TREAD databases. So these are the transcription factors, TREB, ELK, ETS, FOS, JUN, etc. with the target genes, constructed the transcription factor target gene and found that NF-kappa B is getting modulated. And if NF-kappa B is affecting the other cytokines also, IL-10, IL-2, IL-12, which were reconstructed, and over here, it is not visualized clearly, but they are there in this network. So if NF-kappa B is modulated, we can say that we can, take care of the other cytokines which are existing. Now, as I said in the beginning also, for the parameter estimation and the parameter values, you need to be taking into account the robustness. So there we do a MOGA, that is called as multi-objective genetic algorithm, which takes into account the evolutionary robustness. And over here you can see these H1, H2, H3, H4, they are the decision variables for these objective functions F1 and F2. So for the biologists, it is nothing. What we want is from M2 phenotype to M1 phenotype. The H1, H2 are the anti-inflammatory cytokines, say IL-10, IL-4, and the H3, H4 are the pro-inflammatory cytokines, TNF-alpha, and just 2 minutes, IF and gamma. So we have to have this functionality done in such a way that you converge from anti-inflammatory to a pro-inflammatory function, right? So when we did all this analysis, we could see, you can see there is a perfect convergence of IL-10, IL-4, TNF-alpha, and IF and gamma through the multi-objective genetic algorithm. And there is a overall convergence from anti-inflammatories to pro-inflammatories. There's a perfect rugged oscillatory behavior which you can see. So coming back to this, I will just say that since we had this NF-kappa B, all of us know that there is a ceramide, Leishmania virulence factor, GP63, proteophosphoglycan, PTPG, they all are getting, increasing the activation of this NF-kappa B. And L major A mastidotes, basically they induce P50 and C-rail containing the complexes, which form P50 homodimer and P50 serial heterodimer that translocate to the nucleus, altering the cytokine gene expression pattern towards anti-inflammatory state. So Leishmania is capable of cleaving the NF-kappa B P65 relay to P35 relay, which heterodimerizes with P53, and it is a GP63 dependent cleavage which is responsible for induction of anti-inflammatory cytokines like IL-4 and IL-10. Excuse me, because for 3-4 slides were for the students, so I'll take 3 minutes more. We want to have this TH2. So we wanted just that if NF-kappa B is probed, system may evolve to a new pro-inflammatory phenotype. So we designed a synthetic circuit, and over here you can see just with the video that this is nothing, that we just want that circuit. It's not plain over here. That you have the circuit. If you want to plug it, you should get the desired behavior, and if it is not, you will not be able to. So you have all of the components embedded in the synthetic circuit with CMV promoter. Downstream of the CMV promoter, you have all of the parts, components which are inserted with PKC isoforms. Why? Because Leishmania parasite is capable of modulating both PKC and NF-kappa B. PKC mainly through two domains, PB1 and a catalytic domain. So we have this chimeric construct of regulatory domain of PB1 from PKC zeta and PKC alpha. So this chimeric PKC via IKK beta phosphorylation, phosphorylate IKK beta, and then it activates NF-kappa B for translocation from nucleus to the cytoplasm to modulate the gene expression. So this is our concept, that chimeric PKC or engineered proteins are very, very fascinating to be used in for rewiring the signaling network. So this was the concept which I wanted to introduce. I will not go into the technicalities of all of these, but we had induced this circuit in the wet lab experimentation with 1 millimolar of IPTG, checked its stability, and found and always for the student over here, when you design a construct, it should be very clear that you do not follow a very complicated strategy, and it should be very simple. And the synthetic circuit should be orthogonal, means it should not interfere with the host, and modular means it should be, in precise fashion. It should give you a desired output. Supposedly, I have an electrical circuit, I plug it, it should be on and the light should not go off. So that is how the wiring is done. We did the circuit chimeric construct testing on through the Western and the mass spectrometry analysis also. And also all of these parts, you can see we got it from the Registry of Standard Biological Parts and checked its induction on the on state with induction with 1 millimolar IPTG. So further, what we have done is doing the Western
Dr. Shelza,
analysis with the chimeric.
You're really running out of time, ma'am.
And okay, and the confocal image is also showing the same, that the transfected induced phosphorylated ITK beta is high. So just coming to the inference, I will not show much. Our wet lab experimentation, the percentage in was 2 minutes. Percentage infection also was high, and anti-inflammatory response was converted to the pro-inflammatory response. So that just last slide I'll show you. The in silico design synthetic circuit is a logical abstraction analysis driven by CD14, TNF, and EGFR pathway, and the corresponding downstream transcription factor target gene. The reconstructed TFTG network serves as the effector system of the activated signal transduction system. And in nutshell, we present paradigm for the design, construction, and validation of the robust synthetic circuit, which you can use as an implantable therapeutic device with more scale-ups or reuse. Now, this is very, very important for all of the students. Macfarlane Brunet has said, I have done this to illustrate that the approach being used is not meaningless speculation, but has real possibilities of suggesting experiments that may lead to its modification or rejection. Thank you.
Thank you very much, Dr. Shelza Singh. We move to the next speaker now.
So our next speaker is Dr. Somen Basak from NII New Delhi. Dr. Basak carried out his doctoral studies on virology at Calcutta University under the supervision of Professor Dhruva Jyoti Chattopadhyay. In 2003, he joined Professor Alexander Hoffman's laboratory at USCD as a postdoc fellow. During this tenure, he began to use systems biology approach to decipher complex signaling mechanisms. Dr. Somen joined NII in 2010, and his Systems Immunology laboratory has been addressing how cell signaling mechanisms, particularly those involving the NF-kappa-B system, modulate immune responses in physiological settings, such as those elicited by viral infections, and how dysregulated signaling causes disease pathologies, including inflammatory bowel disease.
Dipankar and the team for this wonderful opportunity, and Shailaja for illustrating some of those crosstalk studies and importance of dynamical control that she talked about. And my presentation, the content of the talk today would be probably a little misfit for this session. And we will come back to molecular and cellular immunology. That is what I'm going to discuss here today. But before I go into that, so this is my team, Systems Immunology Laboratory, and today I'll be discussing the work done by Naveen and Alvin in my laboratory. We have supported by a variety of many collaborators. Amit and Vabatosh, we have collaborated in this particular work that we have been describing here today, and also Debarko from IIIT Delhi. And we had discussions with Saurabh and Carla, oftentimes during this progress of our work, and obviously Satyajit. And I'm basically standing between you and this whole end of the day. I know it's a bit precarious situation, but I'll try to be brief. So what is the main question that we've been addressing in my laboratory? You see that you have microbial cues, you have cytokines, they activate these intracellular pathways, so very dynamically controlled intercellular signaling pathways, and that would lead to controlled expression of inflammatory genes. That's important. That's important for inflammatory and immunoregulatory response that the cells would elicit. But when this dynamical control is distorted, when these pathway activities are distorted, that would lead to disease. So the question that we're asking, how do you so nicely calibrate this activity of this intracellular pathways? And people have their own theories. We have our own that we have been testing for last more than a decade now. So I'm going to describe what is the main thesis that we have been testing in the laboratory. So when I say this microbial sensing would activate intracellular pathways, we call them instructive cellular pathways. Let's call them magenta pathway, right? They are important. They are going to activate inflammatory genes. And a cell within their anatomic niche, there are other pathways too. that these yellow pathways, that these green pathways, let's call them ancillary pathways. They're not directly involved in pathogen response, but within an anatomic niche, they're going to be activated nonetheless. And what we learned in last 2 decades, these pathways are not insulated from each other, right? You have done mass spec studies, you have done genomoid shRNA studies, you learned that these pathways are interlinked. In our laboratory, we have been testing, and that allows this biochemical or direct physical or functional interlinking allows pathway crosstalk to happen, crosstalk between intracellular signaling pathways. In my laboratory, we have been testing this thesis that this intracellular cell signaling pathway crosstalk is an important immunoregulatory mechanism, and its dysfunction can have ramification for human disease. So, and we will see here that we will test this hypothesis, and I will describe how we have tested this hypothesis in the context of dendritic cells and intestinal pathologies. And we had several speakers throughout the day, throughout the sessions. They described intestinal homeostasis and inflammation. I'm not going to go into the detail, but this is the mucosal interface. And where you are often subjected, often experiencing cues. You have those dendritic cells that would activate IL-12 and other immunogenic cytokines, IL-23, for example, that would activate effector T cells. They are important to limit, to retain local infections if that happens. But you don't want those effector T cells to be too much active. What these dendritic cells would do, they would also express immunoregulatory cytokines, IL-10, retinoic acids, that would promote regulatory inflammation suppressive T cells in the colon. And that would keep this balance. This balance of Tregs and effector T cells is very key to maintain homeostasis in the intestine. And when this balance is distorted, when you have overactive effector T cell function, that would lead to colon pathologies in inflammatory bowel disease and so on and so forth. So this is where we found out in the literature, dendritic cells play an important role. Not surprisingly, they instruct T cell functions. They're important for maintaining this balance. You see, in ulcerative colitis patients, in IBD patients, there is a marked reduction in dendritic cells that produce those immunoregulatory, immune suppressive cytokines. Not only that, whatever dendritic cells are there, they would make less of those immunoregulatory IL-10 and retinoic acids. And these implicated the dendritic cells in colon pathology and colon homeostasis. And we knew NF-kappa signaling is an important player when it comes to dendritic cell maturation and activation. And this is where this NF-kappa signaling is. You all know about NF-kappa signaling, and Shailaja talked about NF-kappa signaling. This is the signaling they talked about. There's a magenta NF-kappa signaling. It's called canonical NF-kappa signaling, where this pathway activates in response to microbial cues and leads to activation of relay and serial containing dimers that are important for expression of dozens of immunogenic cytokines, IL-12, IL-23, IL-6, IL-1 beta. This pathway has been well studied, including in dendritic cell. And what we realized, because this pathway has so much important role in physiology, that there is less scope for intervention in disease, because this will cause devastating side effect. And there is this another pathway within the NF-kappa system that has been less studied. This is called non-canonical NF-kappa pathway, and we'll remember 2 or 3 words here. This is the NF-kappa gene that makes this protein called PN100. This pathway, you have this kinase called NIC. That's the upstream kinase of this non-canonical pathway that promotes proteolysis of PN100 into the mature NF-kappa subunit P52, which in association with REL-B enters the nucleus as an alternate NF-kappa dimer, REL-B P52 NF-kappa dimer. So you have NF-kappa gene, you have REL-B P52 NF-kappa dimer, you have this upstream kinase called NIC, and this pathway is activated by a subset of TNF receptor. Lymphotoxin beta receptor, GM-CSF receptor, many of them are, by the way, present in dendritic cell. So and this pathway in physiology is known to produce lymphokines that are important for lymphocyte trafficking in the draining lymph node. We asked if this pathway in dendritic cell is regulating that critical balance between regulatory T cells and effector T cells. And indeed, this pathway has been implicated. The dendritic cell function of this pathway has been implicated in various disease models, preclinical disease model. It has been implicated in psoriasis. It has been indicated in mouse models of EAE. And when it comes to the intestine, Some studies have shown that this pathway, at least this MEK kinase, the upstream kinase of this pathway, actually promotes Th17 mediated gut immune response to Citrobacter, suggesting the pathway in dendritic cell in intestine has some role. Interestingly, there's a genome-wide association studies done long back that identified two important components of this pathway, lymphotoxin beta receptor and NF-kappa B2, as a risk factor for human IBD. So we have the component of the pathway has been implicated in human IBD. Mouse studies has indicated this pathway could be functional in intestinal disease. So we asked if RelB-P52, this non-canonical RelB-52 signaling in intestinal disease modulates gut inflammation and gut pathology. So the first question was, of course, independently verifying if this pathway exists in intestinal disease. And this is a single cell RNA sequencing data. We have seen from a lot of those here today. And we looked into this mononuclear phagocytes in the intestine, separated them into dendritic cells and macrophages. And if you see, the component of this pathway, RelB and NF-kappa2, are primarily, if not exclusively, expressed in dendritic cells and not in macrophages. And when those mouse were subjected to DSS treatment that induces intestinal inflammation, you can subject them to 7 days of DSS treatment that would induce acute intestinal inflammation. And you collect cells in a time course and look into the dendritic cell component. What you find here is that during the progression of DSS colitis from day 0 to day 3 to day 6, there is increased expression of the non-canonical NF-kappa B pathway target genes in intestinal dendritic cells, clearly suggesting that this pathway is functional in intestinal disease and is exacerbated during the colitis progression. What we did then, we ablated this pathway in CD11C dendritic cells. And this is the results. And when you subject those mice to DSS colitis treatment leads to body weight loss because of aggravated diarrhea. And you see that for knockouts, there was less obvious body weight loss in acute colitis model. And we also did this chronic colitis model where you actually subject those knockout in cyclical DSS treatment. Again, you see some degree of resistance. That allowed us to conclude that this non-canonical NF-kappa B signaling in disease is intensified upon and exacerbates experimental colitis. But how is it linked? What is the function of this non-canonical NF-kappa B pathway in dendritic cell that leads to experimental colitis? So we first performed some experiment using bone marrow derived dendritic cells. We compared wild type and knockout dendritic cells in a RNA sequencing analysis, and we performed geobiological pathway enrichment assays. We found out that retinol metabolism came up as a front leader. That pathway, retinol metabolism, is likely to be affected. And this is the dendritic cell retinol metabolism. Dendritic cell convert retinol to retinoic acid involving this RDH and RALDH family of enzymes. And dendritic cell secreted retinoic acid plays a very important role in promoting FOXP3 positive Tregs in the gut, in promoting IgA positive plasma cells, and also in leukocyte recruitment in the intestine. So we examined those genes that are involved in the retinoic acid metabolism pathway. And here you go. We found out that deletion of this pathway in dendritic cell leads to increased expression of RALDH2 enzyme, a critical enzyme for retinoic acid synthesis by dendritic cells. And this is our intestinal dendritic cells data, no longer bone marrow derived dendritic cell. We're looking into the intestine, we found out that our knockouts, there's not difference in the dendritic cell abundance or their expression of CD80 and CD86. This is a functional assay to measure the enzymatic activity of dendritic cells collected from the intestine. We found out ablation of this pathway in DC led to increased retinoic acid metabolic RALDH2 enzymatic activity in intestinal disease. Did it cause increased accumulation of regulatory T cells in these knockouts, and thereby they were probably protected from colitis? We looked into this, and the results are very clear, both in lamina propria and in mesenteric lymph node, and both basally and upon DSS treatment, we found out that our knockouts were having showing increased frequency of FOXP3 positive regulatory T cells. We looked into the IL-10 level, they were not per se increased. The frequency of IL-10 positive FOXP3 T cells were increased in the gut. The other part was IgA positive cells. Again, we looked into it. We found out in the gut, there was an increased accumulation of IgA positive cells in our knockout. To an extent, that led to more luminal IgA as measured by our fecal IgA measurements. So knockouts were having more luminal IgA. And this IgA is known to instruct microbiome through its coating and differentially impacting excretion and settlement in the colonies. We did a microbiome analysis. I'm going to really cut a long story short here. What we see very encouragingly, the firmicutes, something that has been associated with healthy gut, we see an increased abundance of firmicutes. And this led us to conclude that this non-canonical NF-kappa B signaling in DC presumably elevates the suppresses RALDH2 expression. So a deficiency of this pathway elevates the expression of RALDH2, leading to colonic accumulation of suppressive regulatory T cells and also eubiosis. This next question was a little bit molecular. What is the link between this non-canonical NF-kappa B signaling and RALDH2? In dendritic cell, these are the three transcription factors that are known to induce expression of RALDH2, NOTCH2, IRF4, and beta-catenin. And results are very clear, our blot, Western blot, from in dendritic cells. You have increased accumulation of beta-catenin when you have a deficiency of non-canonical NF-kappa B pathway in dendritic cells. And when you use a beta-catenin inhibitor, this increased level of RALDH2 that you have observed in our knockout disease could be restored to that observed in wild-type cells, suggesting that beta-catenin drives RALDH2 expression in our knockouts. And what is the link between non-canonical NF-kappa B signaling and beta-catenin? We performed RNA-seq, we performed ChIP-seq analysis, and this is a short summary of it. Very interesting that beta-catenin is known to be constitutively degraded in cell. And these proteins called axin, these proteins, they tether beta-catenin to a destruction complex, leading to its constitutive degradation. What we found out that non-canonical NF-kappa B pathway, this RELB52, promotes the expression of auxin, particularly auxin-1. That in its absence, you lack auxin-1 to an extent, beta-catenin is no longer subjected to constitutive degradation, leading to its elevated level and triggering this beta-catenin RALDH2 tolerogenic gene axis. So that's the mechanistic model. We asked if it is still relevant for colon.
So many of you, maybe 1 or 2 minutes left.
2 minutes, thank you. Yeah, 2.5. So we found out that we asked this question, is it still relevant for the colon? Because those are the studies that are mechanistic. And this is here you go. We took, my students took whole 1 year to do this. They introduced an haploinsufficiency of beta-catenin in our knockout disease. Those mice shows now reduced RALDH2 expression. Those mice shows now reduced Treg expression frequency, reduced level of fecal IgA, and they show reinstatement of experimental colitis, clearly causally linking this beta-catenin RALDH2 with this non-canonical NF-kappa B signaling, orchestrating another example of crosstalk between pathways, and that can influence immunology. And this is a human data set, single cell RNA-seq. Again, we actually use the metadata. And this is again showing very nicely in human IBD. You have engagement of this non-canonical NF-kappa B pathway. So that led us to conclude that non-canonical NF-kappa B signaling in DC fuels intestinal pathologies in IBD by subverting the tolerogenic beta-catenin RALDH2 axis. So again, the crosstalk between two signaling axes, and it's influencing outcome in the mucosal interface. I'm just going to take 1 more minute to present something that we have not published yet. We asked this question that this RELB52 appears to control Treg frequency. In absence of this, you have a lot of Treg. It will compromise immunity in the gut. So how about we challenge these mice with Citrobacter? And we did that. And Citrobacter challenge led to much more pathology in RELB knockout, our knockouts. You see there is a lot more decrease in the body weight, lot more fecal bacterial load, indicating compromised immunity. So we were fine. this must be the mechanism that we have discovered already. We performed this experiment in NF-κB2 knockout. Absolutely no effect, suggesting something else is going on. And this is the results I'm going to show. Dendritic cell-derived IL-23 is known to play a very important role in the gut. And this is the analysis we did. Again, we did an unsupervised clustering analysis of gut disease, single-cell RNA-seq data. And we got these 7 clusters. We asked this question, which one of them are making IL-23? Almost clearly, exclusively, cluster 5 is making IL-23. We then derived a gene signature for cluster 5 because we were interested to interrogate it further. So this is a gene signature that demarcates cluster 5 from the rest of the clusters. And then we went to another data set where there were data for wild type and Rilby knockout intestinal disease. This cluster 5 is completely absent, suggesting there is another function of this pathway in dendritic cell to maintain IL-23 positive dendritic cells in the intestine, which is immune protective. So this is the conclusion. Signaling crosstalk matters. It actually can allow for very tuned control of the immunity. It provides for therapeutic opportunities, but as you see that these pathways have sometimes roles. For example, this non-canonical NF-κB signaling appears to be important for immunity as well. So this is where I will stop. This is the drug dilemma of the system medicine, that more information is required to accurately target pathways or the molecules for betterment of human health. Thank you very much.
Thank you, Dr. Somen, for this enriching talk. Now the session is open for questions.
Yeah, Kushag.
Somen, how does DSS induce this alternate pathway for NF-κB?
Yeah, this is a good question. Yes, very good questions. And we think that we have some data, not very direct evidence. Lymphotoxin beta receptor on dendritic cell triggers this pathway in intestinal disease, and the ligands are expressed by lymphocytes. In addition, people have shown these ligands are also expressed by ILC3, something that we have not tested yet. It appears that there is an increased accumulation of ILC3 during the progression of DSS colitis in the gut. We presume that this might be bringing the ligand and thereby aggravating signaling, non-canonical NF-κB signaling in intestinal disease. But something that we have not tested. There are gaps in the...
So why I was asking this question, that you're using DSS and this is colitis model. Suppose you use another model where you know that how your alternate pathway is activated, so it will be applicable there as well.
DSS, yeah, I think this is a good criticism. We have to perform TNBS or other models.
Yeah, something like
that. I think the microbial cues are unlikely going to be directly activating the pathway. The pathway activity of this non-canonical signaling is very restricted to only a handful of TNF receptor, as far as we know so far.
Thank you.
Sure, thank you.
Sir, how the cell decide to go for canonical or non-canonical pathway? Is it only the ligand or any other factors? Because both pathways can happen in a single cell.
Yes, it can, it does. Give an example, CD40 in B cell activates both the pathways simultaneously. But it is mostly the ligands, as I was mentioning, TLR, cytokines, they do activate canonical signaling. And it's very pervasive, activation of the canonical pathway. The non-canonical pathway, however, only a subset of receptors, and the receptors are not present in all the cell types. Dendritic cells, you do have them. Lymphocytes express mostly the ligands. So that will determine what kind of activity you see in these kind of pathologies.
Thank you, Somen. In the interest of time, we're going to bring the session to a close. Thank you all, and thank speakers once again.
Okay, thank you very much.
Our sincere thanks to all the speakers for the wonderful talk. I kindly request all the speakers of this session and the chairs to come up on the dais for a group picture.
Sorry.
Thank you to the speakers and the chair. As we wrap up day 3 of IMMUNOCON, we have few engaging shows featuring performances by the talented students of IISc. First, we have the Musical Society of IISc, Rhythmika. Rhythmika is the official musical club of IISc, started by a group of enthusiastic musicians in 2002. Rhythmika has grown to have over 50 active members every year. They have musicians with diverse taste in music, ranging from Indian classical to Bollywood to rock and pop. Let's welcome Rhythmika on the stage and dive into a musical ecstasy.
Let me just introduce our members. So we have Atharv on the tabla.
Hello.
Then we have Rishabh on harmonium. We have then Sanshray on the cajon, djembe and ghungru. Then we have Ashwin on the keys and vocals.
Hello.
Hello. We have Walling
Hello.
on keys. We have Rishabh doing vocals and harmonies. We have Kedar doing vocals. We have Swapndeep doing a harmonium and vocals. We have Vinayaka doing vocals. We have Monmita again doing vocals. And we have Anvesha doing vocals. We have Ekta on vocals. Devashmita on vocals. Shubhadeep on vocals. And I'm Samat, I'll be doing the guitar. And yeah, and Sheesh on vocals, yeah. Sorry, a lot of people to introduce.
Hello.
Yeah.
It's 2.
So we'll be doing a couple of like semi-classical, classical and pop songs.
Hello.
And I hope you like them. Again, if you know the lyrics and all, you can like feel free to sing along, clap along.
Hello.
Audience participation is always looked positively upon.
Check. It's there, no? It's there.
Hello.
केतकी गुलाब जोहि चंपक बन फूले, केतकी गुलाब जोहि चंपक बन फूले, केतकी गुलाब जोहि चंपक बन फूले। ऋतु बसंत अपनु गंध गोरी गरबा लगा, में बैठ आज पी के संग झूले, केतकी गुलाब जोहि चंपक बन फूले, केतकी गुलाब जोहि चंपक बन फूले। गैल गैल कुंज कुंजा, गैल गैल कुंज कुंज गुण गुण भवरोति कुंज, राग रंग अंग अंग छेड़त रसियान। कोयल की पंचम सुन दुनिया दुख भूले, भूले भूले। केतकी गुलाब जोहि चंपक बन फूले, केतकी गुलाब जोहि चंपक बन फूले। ऋतु बसंत अपनु गंध गोरी गरबा लगा, सुलना में बैठे आज पी के संग झूले, पी के संग झूले। केतकी गुलाब जोहि चंपक बन फूले। मधुर मधुर थोरी थोरी मीठी बतियों से गोरी, मधुर मधुर थोरी थोरी रे सा दधनि रे सा, मदनि रे सा नि ध प ध म ग म ग ध नि ध म ग रे सा 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 म प ग म नि ध नि 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 म म नि म रे सा रे सा नि ध प म ग म ध नि सा रे सा नि ध प म ग म ग। मधुर मधुर थोरी थोरी मीठी बतियों से गोरी, चित चुराए हसत जाए, चित चुराए हसत जाए। चोरी कर सिर झुकाए, शीश झुके चंचल लट, कालन को छूले, कालन को छूले। केतकी गुलाब जोहि चंपक बन फूले, केतकी गुलाब जोहि चंपक Hello, hello. Hello, hello. Check. Hello, 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 hello. ओरे पिया ओरे पिया ओरे पिया ओरे पिया उड़ने लगा क्यों मन बावला रे आया कहाँ से ये हौसला रे ओरे पिया ओरे पिया ओरे पिया ओरे पिया साजिश में शामिल सारा
Hello. Hello.
Hello.
Hello. Hello. Hello. Hello. Hello. Yeah.
खामोशियों के तरंग लिए तरंग रिश्ता ये कैसा जल्दी निभाओगा रिश्ता ये कैसा जुल्म को बचाओगा बोलो अधूरे हम दिन तुम्हारे अधूरे हम दिन तुम्हारे छोड़ दे रात की चुप्पी में शोर नट कर छोड़ दे मुट्ठियों में बादलों को लेके मुंह छोड़ दे रात की चुप्पी में शोर नट कर छोड़ दे बेबाक है बिंदास है जिंदगी की प्यास है मजबूरियां मनमनी मजबूरियां है वो अधूरे हम दिल तुम्हारे अधूरे हम दिल तुम्हारे प्यार की ये कहानी सुनो, एक लड़का था एक लड़की थी, होती क्या है जवानी सुनो, एक लड़का था एक लड़की थी, वो भी एक दौर का वक्त ही और था जब वो थे अजनबी, दोनों तन्हा से थे पर वो करते किसे बात जो दिल में थी। प्यारे की एक कहानी सुनो, एक लड़का था एक लड़की थी, वो जीता है जवानी सुनो, एक लड़का था एक लड़की थी, वो एक बार था वक्त ही और था वो थी अजनबी, दोनों तन्हा से थे पर वो कहते किसे बात जो दिल में थी। जिंदगी ये कहानी सुनो, एक लड़का था एक लड़की थी, होती क्या है जवानी सुनो, एक लड़का था एक लड़की थी।
और प्यार हो ही गया, जो मांगा था वो पाया तो होश खो ही गया।
जिंदगी ये कहानी सुनो, एक लड़का था एक लड़की थी, होती क्या है जवानी सुनो, एक लड़का था एक लड़की थी। अब मोदी बातें आए जिनके गाने सुन मेरे सारी हवाएं, धड़कन धड़कन मैं क्यों बनी, मन चाहत है मेरी सबने अपने जादू जगा।
तो जानोगे क्या है नशा प्यार का, सोचोगे यही चाहोगे, सुन रही है सदा। सुन रहे सदा,
सुन रही है
सुन रही है
सदा।
initiative.
क्यों आजकल नींद का ख्वाब ज्यादा है, लगता खुदा का कोई नेक इरादा है। कल था फकीर आज शहजादा है, लगता खुदा का कोई नेक इरादा है। क्या मुझे प्यार है यार, कैसा कुमार है यार, क्या मुझे प्यार है यार, कैसा कुमार है यार। पत्थर के इन रास्तों पे फूलों की एक चादर है, जब से मिले हो हमको बदला हर एक मंजर है। देखो जाम नीले नीले आसमान तले, रंग नए नए जैसे घुलते हुए, तेरे सामने जागे तेरे वास्ते, तेरे ख्यालों से भीगे मेरे रास्ते। क्या मुझे प्यार है यार, कैसा कुमार है यार, क्या मुझे प्यार है यार, कैसा कुमार है यार। तुम क्यों चले आते हो हर रोज इन ख्वाबों में, चुपके से आ भी जाओ एक दिन मेरी बाहों में। तेरे ही सपने अंधेरों में उजालों में, कोई नशा है तेरी आंखों के प्यालों में। तू मेरे जामों में जवाबों में सवालों में, चढ़ते चुरा तुम्हें मिलता ख्यालों में। क्या मुझे प्यार है यार, कैसा कुमार है, क्या मुझे प्यार है यार, कैसा कुमार है। আমি যে তোমার শুধু যে তোমার মেরে ঢোল না मेरे ढोल ना सुन मेरे प्यार की धुन मेरे ढोल ना सुन मेरे प्यार की धुन मेरे ढोल ना सुन मेरी चाहते हो फिजा में बहे जिंदा रहेंगे होके फना मेरे प्यार की धुन मेरे ढोलना
साथी रे तेरे सर में है चलेगा तुझे ही बेचैनियों में आएगा दिल साथी रे मर के
तुझको चाहेगा दिल तुझे ही बेचैनियों में पाएगा दिल मेरी खुशियों के साथ में तेरी राहतों की खुशबू है तेरी बगैर क्या जीना मेरे रोम रोम में तू है मेरी चूड़ियों की खन खन से तेरी सगाई आती है दूरियां हमेशा ही मस्ती का मुस्कुराती है नीधा मग सनि दरे सगमा मग सनि दरे सगमा सगमा मदनी मदनी सा नीधा नी दरि सा दरि सा दरि सा दरि सा मदनी 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 सा मदनी सा मदनी सा मदनी 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 सा मदनी सा मदनी सा मदनी सा सनि ध सनि ध सनि ध सनि ध सनि नि सा सनि नि ध नि नि सा सनि नि ध म म ग ग सनि नि ध सनि सा 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 नि ध सा 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 मेरे ढोल रास मेरे प्यारे की मेरे ढोल
सांसों में तेरी सरग में है रात दिन जिंदगी मेरी तो कुछ ना अब तेरे बिन सांसों में तेरी सरग में है अब रात दिन जिंदगी मेरी तो कुछ ना अब तेरे बिन तेरी धड़कनों की सरगोशी मेरी धड़कनों में बजती है तेरी जागती निगाहों में ख्वाहिश तेरी ही सजती है तेरे ख्याल में हर पल तेरे ख्याल शामिल है लम्हे जुदाइयों वाले मुश्किल भरी ये मुश्किल है शुद्ध जे तुम्हार मेरे ढोल
अगलावा मजबूरी नो आन जान दी वसूरी नो सैर बने जाओ मैं पूरी नो आना से ओ नहीं आया दिल भाग भाग में टकराया कहाँ बोल के दस जावे दी चूरी नो रावाच बावाच उनो लुगावा कोई बैनू ना रोके मेरे ढोल जुदाइयां दी खबर की वे हो मेरे आ जा